participants from various colleges, my colleagues, and students and scholars from KFRA. Warm welcome to the third day of the technical section of the Tropical Forestry Conclave. So this particular session is on advanced tools and techniques in forest research and management. And the session aims to expose the participants on recent developments in tools and techniques employed in forestry and environmental research. This includes GIS and remote sensing applications for environmental monitoring, AI, ML, and IoT-based forest management tools, and the emerging role of next-generation sequencing technologies in forest science. So we are privileged to have with us today two eminent speakers in the first subsession on GIS and remote sensing, technology shaping environmental and forestry research. We have with us Dr. Sudhagar Reddy, Head, Forestry, Scientist SG, Forest Biotechnology and Ecology Division, National Remote Sensing Center, Hyderabad. Welcome, sir. Due to some unavoidable official commitments, Dr. Chandrasekhar sir, APCC of Kerala Forest Department, was unable to join us today. And we have the second speaker, Dr. Gopakumar S. He is a professor and Dean, College of Forestry, Vellanikara. He will join us shortly. So with great pleasure, I welcome Dr. Krishna Kumar sir, former PCC of Head of Forest Force, Tamil Nadu Forest Department, former Director, 𝐓𝐑𝐿𝐷 Coimbatore, and former Chairman, State Environmental Impact Assessment Authority, Tamil Nadu, as well as he is our former Chairman, Research Council, KFRA. So, who has kindly consented to moderate this session. So, welcome, sir. Over to you, sir, for the conduct of the session.
Thank you, Dr. Suma. Morning, friends. I find a lot of young, smiling faces here. Not that maybe a good breakfast in the morning or happiness, because the subject for the whole day is going to be very important for us. Young friends who were here yesterday, or those who could not be with us the last 2 days, I should tell you that we did a lot of deliberation on tropical forest ecosystem, the significance of it, and how important it is, just not for Kerala. Tropical forest ecosystems for the Western Ghats, for the whole of the country, becomes important. So the conclave has been so well arranged that every day has a connectivity with the next day. And whatever has been deliberated has a continuum. And then the whole thing, we look forward to some excellent output coming out. And today's deliberations are very important because we are talking of advanced tools and techniques. I only go back to the days of 82 and 85. We have to always remember good old days. I remember the type of surveys that we used to do in the forest, the Gunter's chain and the pole and posts that we used to carry for surveys. And then those heavy chain has to be dragged for, all along the ground. Doing some surveys was so very difficult. But then, 1990, when we really were working for Hill Area Development Program in the Nilgiris and the whole of Tamil Nadu, etc., I remember me going with the district collector Nilgiris to meet the ISRO chairman, Dr. Rao. And then we had a deliberation at that time. And then he was telling us the significance of this subject of GIS and the sort of development that were happening in India. We gave them the work of doing watershed mapping for the Nilgiris, and then we got the macro, micro, mini watershed. We knew what are forested watersheds, non-forested watersheds, and then looking into our inputs for the next 10 years to come, how can we really saturate watersheds to see whether those watersheds are giving us the ecosystem services that we think of. With this experience, we also, I remember conducting, Dr. Kannan Warir is here in our own institute. We have an institute like this in Coimbatore called the Institute of Forest Genetics and Tree Breeding, where we had a tropical forestry workshop. And then that also gave us inputs. So, for students particularly, sitting in such conclaves and conferences becomes very important. You get exposed to the best of things happening in the country because very eminent speakers are coming up to talk to us. And that's a very great fortune today because the future lies with all youngsters in India, laden with youngsters, is the bright future for us. Now you go up and outside and just walk across, you will see out of the 30 or 40 posters or 70 odd posters, 70 odd organizations are joining us the last 3 days. And out of all those posters, I think some significant posters are from this area of GIS and RS, and the type of work that's going on today is mind-boggling. Awesome sort of work is going on in this area. You touch any field, for example, 𝐓𝐑𝐿, the type of work that's going on mangrove mapping, or the type of biodiversity assessment that's being done here using these tools and techniques is so interesting. And n number of other institutes are also working. We have very lead speakers today and 2 eminent speakers. In fact, I am very honored to present before you Dr. C. Sudhakar Reddy. Welcome, sir. And it's a pleasure. He's the scientist G and head of forestry from the National Remote Sensing Hyderabad. As a principal investigator, he has successfully executed 25 sponsored research projects, and as a project director, he led a major initiative on biodiversity characterization using Earth observation data. Dr. Reddy has been consistently ranked among the world's top 2% of the scientists. Now give him a big clap for that. And top % of the scientists for the past 6 years by Stanford University and Elsevier. He has made outstanding contributions to remote sensing applications in forestry, biodiversity, and ecology, playing a pioneering role in the development of spatial biodiversity databases, the first of the kind for India and South Asian region. I also understand Dr. Sudhakar Reddy has been spearheading and supporting KFRI's initiatives in very big way. We are all very keen on listening to him. I'm sure there will be a lot of messages and takeaways from his talk. Without much delay and me not going into the subject, I will request Dr. Sudhakar Reddy to kindly come and present your thoughts.
Good morning, all. I am grateful to Director KFRI, Organizing Secretary and Convener, as well as the entire team of KFRI for giving me this valuable opportunity. So before going to the presentation, generally like in politics, in Hindi, like you may be knowing word like roti, kapda, aur makan, like food, shelter, cloth. So now the technology, along with this roti, kapda, aur makan, you have internet. Now we are living in an era, we cannot live without a smartphone, without a communication. It may be a remote sensing, it may be a telecommunication, it may be a navigation, or maybe resources. So now the entire sphere of life of the human is revolving around the internet. So in my presentation, I'll be speaking about some of the known things, maybe by you, some of the new things, which are recently conducted with the collaboration of KFRI and many other academic as well as the scientific organizations of the country. So going to the presentation, I would like to highlight, so why we are worrying much. If your house is safe, no one will bother. If your neighborhood is also safe, you will not be much bothered. But your entire planet Earth, it is not by the one individual. It is the amalgamation of flora, fauna, microorganisms, many ecosystems, landscapes, resources, okay, whatnot. There is uncountable resources, and the humans are depending on all these resources. And if you see the recent literature or in any publication, you will be coming across with some keywords like the carbon sequestration, climate change, ongoing crisis of biodiversity loss, and also the increasing level of pollution in our environment. So this is like a kind of a triple planetary crisis. And now if you look into the aspects, the entire system is now revolving around the green leaf. So by protecting, by conserving, by restoring the ecosystems, specifically for the native species, you may be able to reduce or able to minimize the impacts which are currently faced by the planet Earth. So it is not for the sake of the flora, fauna, we need to see, it is for our own survival. So what is happening? What the science is doing for a long time? Is there any solutions came, or still we are doing some progress, or we didn't achieve anything? But if you see the policies, so the conservation policies or the many negotiations like CBD, IPCC, they are because of the efforts by the science. It doesn't mean that our publications are not going anywhere. No one is using. We are giving the inputs to the policy and also to the conservation strategies and also for the management plans. So if you see here how the chain is evolving or the evolution for the science of the biodiversity. Initially, it has very humble beginning with our traditional taxonomy, and everyone is aware of the father of modern botany or taxonomy, Carl Linnaeus, the binomial nomenclature, species plantarum. Okay? So now, in the 18th, 19th century itself, we achieved the good knowledge about the identification of the flora and fauna. And all our expeditions have contributed the enormous in situ observation data, very fine resolution, very fine scale information, starting in the last 2 or many centuries. So this knowledge is now growing day by day or decadal scale. And if you see this entire evolution chain from like 18th century to the recent period, how we are now studying the biodiversity. So if you see this, initially just like a checklist of species for a given area. Later, we are utilizing the like herbarium and museums as the resources for a proper identification. Then at the time, in the time of Linnaeus, there were no the concepts of like the ecology, evolution, or there is no proper concept of like the plant community, biological community, ecosystems or species. It is later, as the technology is growing, our science is evolving, slowly we are like moving from the simple identification based on the morphology to the current day like the integrative taxonomy. So in this, like the today's session is also, one more session is also on the advanced tools of the molecular biology. So it is now, it is not simple based on identification, is based on morphology or anatomy. It is now integration of the integrative knowledge from the ecology as well as the molecular tools. So then only we can able to give a precise identification. So it is not the expertise from one morphological taxonomy, it is now the integration of the multiple scientific disciplines. Then again, if you see this, the concepts of ecosystem, they might have like came in 1900, but now the science of ecology has grown very much. It's like you have now several ecological models, species distribution models. Now we can able to identify the impact of the climate change on the ecosystem as well as on the native species, or even we can able to predict the future biological innovations. So like we have large amount of data coming from the in situ field data by the efforts of the taxonomy, efforts of the ecology. And as now we have the advanced technologies like GIS. They came very late, but now they are becoming very prominent. Even GIS, remote sensing. You have multi-sensors. You have the data from like sub-meter resolution to coarse resolution. You can able to monitor the Earth's surface in near real time, daily basis. And now you have the last 5 decades of the time series data coming from the space-based or satellite-based remote sensing. So they are fulfilling large amount of requirements for the policy. Okay? Why I'm saying policy means it's not like we need to have the information only at the plot level. We need to have the understanding local level, like micro level to the global scale. So in this context, the remote sensing is one of the best suitable technology, providing the data required for the management and also for the biodiversity research. So then how to monitor? Whatever the advancement happened is based on the knowledge from the predecessors like Dr. Nandakumar sir. He gave way for the new generation. Dr. A.R.R. Menon, he helped a lot in the initial days of ecological studies through the KFRI. And now we have Dr. Srijit and many youngsters also. They are also contributing not only like in one particular biodiversity, they are now touching like ecological or ecophysiological aspects of biodiversity, mycology. It's like it is not limited to one just simple domain of angiosperm taxonomy or the study of higher plants. Now we are going in the approach of the like comprehensive. So try to add as many types of groups like the flora, fauna into your biodiversity research. Then that will give you the more precise or accurate information for the resource monitoring and also the biodiversity conservation. The problem is that the biodiversity is like a very complex subject. It is not like the one person who is like having a specialization in climatology, they can't do something. So there are experts in different domains. But now the biodiversity is like a one kind of umbrella. It will bring everyone together, like geneticists, cytologists, or if you take any field of life sciences, if you take any field of environmental sciences. So we need the collaborative efforts. Then we can able to assess the change in the biodiversity patterns, maybe at genetic level, species level, and at the ecosystem level. Then what have the, because again we need the standards. It is not that everyone is working on some silos or in some different concepts. Okay. So for that, there are like several influential publications came. One is by Noss, published in Conservation Biology in 1990s. So he identified the hierarchical approach. Okay, he suggested that we need to study the biodiversity from the bottom level to top level. So here the species will become the finest scale or the unit for the biodiversity. Okay, but later studies identified that we need to go for even biological organization, including the ecological organization. So the concepts are now going like the new way. Now the global community has an agreement that now we need to utilize the novel concept in the name of the essential biodiversity variables. So this concept came through the concept of essential climatic variables that is required for monitoring the climate, for the climate change purpose. So taking the concept of the climate, the experts in biodiversity from the multiple disciplines, they published the very influential research article in Science in the year 2013. And currently the GeoBon, it is a Group on Earth Observation, Biodiversity Observation Network, GeoBon, they are contributing and also supporting the standardized frameworks for the monitoring of the biodiversity at the various levels. So and they are also opened one, they have the web portal. So all you can even become members in the GeoBon, and you can also contribute from your side to this complex science. Then what is the remote sensing? Why I am giving the highest value for this? Okay. Here, none of the sciences are like inferior or superior. It is like a collaboration. We need the expertise of all the disciplines, even including geography. It is not that none of the subjects are like superior or inferior. It is like each subject will support in its own way. And finally, the policymakers or the resource managers need the information, because they will not do science like us for like 3 years or like 5 years project. They want the output easily readable, easily executable, and so they need the abstract way, the information. So we are summarizing that whatever the research we are conducting in the form of maps, in the form of the scenarios, in the form of the indicator development. Okay, so that is required for the precise management of the biodiversity and also like forest resources. And this is depending on like the subject. The primarily came from like physics. So everyone is aware of like electromagnetic spectrum. Okay, but why I am saying electromagnetic spectrum and why to use like remote sensing is our eye is able to see only the visible region, blue, green, red. Okay, we can't see any information which is available in infrared or thermal, because we can't say what is the room temperature by just touching any object. Okay, but the remote sensing, it will provide you the information of the thermal, like land surface temperature you will get. You'll also get the information, spectral information. So that is the proxy for the species composition. The remote sensing also, by having the time series data, will provide you the functional aspects of the biodiversity, like phenology, productivity, carbon sequestration. Okay, so you can able to address many of the things which are human cannot sense But the remote sensing, by collecting the information in the infrared region, thermal region, as well as in the microwave region of remote sensing, it will provide you the information on the three primary attributes like the structure, composition, as well as the function. So now coming to the ecological side. In general, our majority of our program is primary components. So our one of the mandate is to look at habitat. Is the habitat is intact or undisturbed? How to say it? By having the time series remote sensing data, you can able to map and monitor the changes in the habitats. So that will give a first-hand information to know that the species, the habitat of a given species is relatively safe, or it is shrinking, or the habitat is expanding, or habitat is getting restored through the management activities. So we can able to monitor through the various kinds of remote sensing data. Then also we need the land use, land cover information. Also we need the information on the aerial distribution of natural vegetation, human-made vegetation, like the cultural vegetation. And then also we need the precise information about the vegetation types, communities, and species. Okay, this is now possible through the integration of the samples from the field, and by using the machine learning techniques, AI-based tools, you can able to generate the very important information. Okay, the earlier days, like when Nandakumar sir is working, like we are at the time, like in 2001 time, we are doing like on-screen visual interpretation. Okay, so the only expert only can able to do the mapping. Okay, but now the people from the computer science background, geography, environmental science, so anyone can do. But the primary information should come from the people with the knowledge of the taxonomy and ecology. Okay, so then you will be using their data, and you are training the different kind of machine learning models. Okay, so why I am saying machine learning model is, it will be doing both things. It will be doing the classification of the land cover, or classification of vegetation types, or classification of different species, and it is also doing the regression. Okay, so it is doing classification as well as it is also validating the map itself. Okay, in the earlier days of like visual interpretation, we are only mapping through the expertise. Then we are taking some samples to go for the accuracy assessment. Okay, so there may be some subjectivity because you are taking few samples and you are saying that my map is very accurate. Okay, here it is a random choice. Okay, like a random forest model. Okay, it is like a completely machine dependent and unaltered. Whatever the product you are generating has the statistical meaning along with the biological significance. So now, what kind of data we required? Okay, it doesn't mean that it's like a simple observation data, like point location. We need the data in a quantitative form. So now, if you see this plot, the recently like we did with collaboration of KFRI also in the Western Ghats, and also collaboration of the premier institute like the French Institute of Pondicherry, MS University of Baroda, Andhra University, IIRS Dehradun. Like that, we are collaborating. Is the phase one of the study, and we felt that the studies which were done earlier really useful, but there is a lot of uncertainty in estimation of the biomass or in estimation of the biodiversity, because our small plots may not capture the information on the three fundamental aspects. Because your small plot, you have the overestimation of biomass or overestimation of species richness, or the vice versa. But by having the 1 hectare plot, it will be providing the information on the structure, the composition, and the function. And this 1 hectare will be plot is like a mini forest. Okay, and this is not came through my own wisdom or science. In the like there is one international body like CTFS, Center for Tropical Forest Science. Recently it became Forest Geo. So the CTFS protocol recommends that in a tropical forest, we need to go for minimum plot size of the 1 hectare. Okay, so then it will reduce the lot of uncertainty while estimating your carbon sequestration potential, or even you are going for any quantification of the ecosystem services. So this is like an ideal plot. Okay, and also it will helpful to the resource managers at the local scale, because here we are not simply doing the map, collecting the data of the plot. We are trying to georeference each tree location. Okay, by having the tree maps, You can able to use these plots for even future management plans as well as for working plans. So we need not to go every time to the new area. So we can use these permanent samples, and later stages we can able to study the succession, degradation, improvement, anything, and also the information on like the distribution of the invasive species, endangered species. So a lot of information is available by having the larger plot. Then why the remote sensing became very popular. Like the ecological information which is collected at the local scale, it can be able to generate information of forest type for the small areas. Okay? But by using the same data and using the multi-sensor remote sensing data, maybe the multi-created decision model, maybe by using the ecological principles or even using machine learning based tools, now we are in a position to integrate the information like the climate information that will help us in identifying the tropical, subtropical, temperate and alpine zones. And also we can able to integrate the DEM, three-dimensional information coming from the remote sensing. So you'll be getting the elevation zones information. So you can able to identify the coast, hill, mountain. Okay. So now you can also integrate the information coming from like the biogeographic zone classification, Western Ghats, Himalaya, islands. Okay. So now based on integration of this kind of information and also through the coming through the high temporal remote sensing data, we can able to capture the information relevant for classifying all the vegetation types. So as per the champion set, that is one of the standard framework in Indian forestry. So we can able to identify all the 16 major forest types by using this multi-source remote sensing data. Okay. Then it's not only like we are able to delineate the different forest types, and while doing the classification, you are working with the human or modified landscapes also. So then how to generate the information really required for the near real-time management. That is a challenge earlier. So thanks to the AI and ML kind of technologies, and thanks to the web portals like the Global Forest Watch, and many portals like from the Copernicus program, USGS, Earth Explorer, okay, like you have several web portals or geo portals, they are also providing a lot of open access data, okay. And like if you see the ISRO's Bhunidhi portal, now we made 5-meter resolution data free, and all the data like IRS list 4, list 3, all the data of the resource set, you will be getting free from the Bhunidhi portal. So now you can able to monitor the vegetation, not to wait for like 2 years, like the FSI is providing the assessment. You can able to see the vegetation at even every 5-day interval at a 10-meter resolution. You may be aware that there is one series of satellites from Sentinel-2 basically. You will be getting the 10-meter resolution data for a given spot and free of cost, and that is a analysis ready data. Okay, you need not have the expertise of computer science or the geoinformatics. So directly you are downloading the imagery and you are putting your samples and you are classifying. So then you can able to see that if any changes of like degradation or also positive side, like we can able to monitor the afforestation activities taken by the forest department. Then coming to the complex side, the mapping of forest types is relatively easy because we are using mostly coarse resolution and medium resolution. But coming to the community level, okay, here we need to discriminate the community based on the structure, phenology, and all the related attributes. Okay, so here is like first of its kind exercise in the world. Here is the first publication. We use the data from microwave remote sensing that will capture the information on structure, okay, like X-ray. Again, we use the Sentinel-2 series of data, high temporal for the 2 years. So it will capture the spectra information for composition and also the phenological information as the one key attribute to separate the different communities to one of the key. So by using the Sentinel-2 and also by using the freely available LiDAR data, okay, you may be aware of that JD, Global Ecosystem Dynamics Investigation LiDAR, that will give you the data for the tree height, okay. So there are 2 publications came, one is with 30-meter resolution, another with 10-meter resolution. So they using the data from the International Space Station, the researchers have developed the global forest canopy height map. So you have now large number of data sets. So we use the tree height map as one of the key to separate the community. So by having the information of these multiple resources and using the data fusion techniques, we could able to generate the community level maps as part of our collaborative project with the KFRA. Then what is happening at the global scale? So we need to see at the local scale, and also we need to have the understanding what is happening at the country or global scale. So there are a lot of developments in the technology, and the Europeans, if you see the Copernicus program, now they have generated like phenological maps. Okay, you can able to get now every year data freely. So if you are not even having any skills of image processing, you can even download this data, and you can able to study your vegetation types and communities. So this is like peak of growing season for the entire world. Okay, and this data is freely available. So you can make use of such type of data sets in your ecological research. So here I'm showing one of the effort for the initially for India. Later we extended to the neighboring South Asian countries. Okay, here we have mapped the forest cover for the different time periods. Okay, and then I'm showing also map for the 1930s. The satellite imageries are available from 1972. Okay, but there is large amount of survey data is available in the form of the topographical maps. So we have mapped the green cover using the topographical maps, and it will be like a proxy for the forest cover because we don't have any other alternative. So that will give you an idea that how much of the habitat got lost. If you really want to establish a framework for the red list, so this kind of database are very, very important. Even if you are going for any kind of ecological restoration, if you want to identify the areas for improvement. So these kind of spatial databases are useful, and some of these are available in the Bhuvan web portal of NRSC for the free download. Now, like we're now living in an era of IT, information technology, AI and ML. So people may not even want to go to field, but still they want to protect the resource and conserve the resource. And also there are many indices, and one I'm showing the most important one is the DHI, Dynamic Habitat Indices. I am giving here one example of Sentinel-2 data. You will be getting data at every 5-day interval. Okay. So if you use a time composite of monthly, seasonal, or year, or decade, because this data is available from the last 2016 onwards, you can able to see the trends in the greenness. So DHI is also helpful to the managers to see that is there any early warning, is there any improvement or any degradation or no change. So like DHI greenness trend. Here I am showing one example for the Simlipal Wildlife Sanctuary in Orissa. So it shows that the changes are very, very insignificant. If any change happened, they are like changes in the croplands. Because we have the crop layer, we can able to see that whatever like negative is coming, because from the land use map you will be getting the proper identity. So that will help you to that see that okay, this is the change in the cropping pattern. It is not the loss of vegetation from the native ecosystem. And then also overall also it will show that how much productivity is getting increased. Okay. And also if you want to use this data in a long term, you can able to see the like impact of climate change on the vegetation. So the seasonality map will give you that the change in the seasonalities of or phenological cycles of the flora. So you can able to find out through this kind of indicators. And you can use in many places like identifying the areas of high species richness, areas of many like habitat condition. You can use many types of applications. Then like AI and ML era. See, no one is now waiting. They want immediate. So the global community, both science as well as the policy, they want the information with less uncertainty, more accuracy, and with the systematic observations. So there is one web portal with the collaborative efforts of the global agencies. Now we have the Global Forest Watch. So they are saying that at the global scale, we are like almost we had last like around 12% of the decrease, 12% decrease in the tree cover in the last 23 years. Okay, so this is one indicator, and where it is happening, you are able to see it there. Are they in the scrublands, forest lands? Okay, The problem here is that everyone has their own definition. FAO has its own definition. FSI has its own definition. I have my own definition of natural forest. You have your own layman definition. The problem is that while estimating, we need to utilize one global, that still we need a lot of consensus. If you see the literature, for the forest, you have more than 800 definitions. Even some countries are having more than 5 definitions. So that is also one of the problem while reporting such kind of changes in the vegetation. This is a global forest canopy height map. It is also freely available from the University of Maryland website. So if you want to have, for example, KFRA tree height map, you need not to have the expertise of laser sensing or any remote sensing knowledge, or you need not to have any software now. So by using open source like QGIS, you can download this data and you can able to study, and you can use this as one of the input in your ecological model. So forest canopy height information. Biomass estimation. This is again very challenging. Okay, it is very difficult to see the biomass from the space. Okay, so it is only the contribution of the ecologists. They identified the different kinds of the approaches. Okay, later we applied the GIS, and later we applied the relevant remote sensing data, and trying to use all the approaches which are valid. Okay, as of now, if you see the FSI based biomass or forest cover kind of assessments, they will be integrating the approach called stratify and multiply approach. So here, they are collecting the samples, field samples from very dense forest, moderately dense forest, and open forest. Okay, so they are making one mean value for open forest. So based on that, the entire Kerala open forest will have one value. Okay, but as an ecologist, you know that there is large amount of variation even in the open canopy of even 10 to 40% of canopy closure. You know that age of the plantation or age of the tree, height of the tree, wood density, many things, they are going to affect your map. Okay, so then what we have to do now? We need to go for the better one, like direct remote sensing. So here we'll be depending on LiDAR data that will be giving the actual volume, tree height, or another way you can use the data sets like microwave remote sensing. But the limitation was we don't have the appropriate bands for the biomass estimation, like C band, which is saturates, that is only good for the low biomass vegetation. It may not be good for the high density forest. But currently, with the collaboration of ISRO as well as NASA, they collaborated, have one new mission recently launched in the name of NISAR, NASA ISRO Synthetic Aperture Radar, L band, and also S band from ISRO. Then we have the one more band, biomass, P band. So those will be going to help us in the accurate estimation of the biomass of any kind of ecosystem. So this is based on the stratified multiply approach. So here I have integrated the information of the forest cover, forest canopy density, and also the mean biomass values of the different vegetation strata. And then we are able to build the historical biomass changes in the native forests. Here I have not included any orchards and plantations. So we are trying to see that is there any change. Okay? If you see the graph, it is clear that the forest cover is relatively stable at the national scale. Okay? I'm not saying that no deforestation kind of thing. It's like national scale, it is stable. But the level of biomass getting decreased in the native forest. Then the forest fragmentation. Okay? Specifically, while you are studying the biological innovations or plant innovation, and also while you are studying or you want to address the wildlife-human conflicts or the wildlife-related studies. So the fragmentation as well as the connectivity maps are the prime input. So as the technology grown, a lot of open source tools are available, like FrogStat, landscape fragmentation tool, MSPA, morphological spatial pattern analysis. Okay, they are giving you the information not only for the fragmented landscapes, they are also generating the connectivity information, like the bridge forest, loop forest. Okay, so you can make use of such kind of open source tools, and the input is your forest cover map. This is the one decadal long project. At the time, we had collaboration with 48 institutions across the country. And KFRI was one of the prime agency institutions supported us for the Western Ghats of Kerala. So then Dr. A.R.R. Menon was our principal investigator. He supported us with a large amount of field data, and also he has a lot of knowledge in the GIS and remote sensing. So based on his knowledge and the expertise from the other people, we developed the model called SPLAM, Spatial Landscape Modeling. It's the multi-criteria decision model, helps you to identify the areas of high biological richness. Okay? So here, like, the experts from the field are giving information on species richness, economic importance, endemism, ecological uniqueness. Okay? People from, like me, even though I am botanist, since I am working here, I'll make use of the computer-based technologies. So then we had a multi-criteria decision model that was built based on the expertise knowledge from the multiple people. They are coming from the different disciplines. And we also shared this database with the National Biodiversity Authority and all the state forest departments in the country to be utilized in their plans. Then the technology is growing up. No multiple experts are not required much. You can use the new hypothesis, like the spectral variation hypothesis. So here, again, in our recent project with the collaboration with Dr. Srijit, here we try to generate, not with the multi-criteria decision model, because sometimes it's like subjective. The expert may be biased towards remote sensing side or ecology side, so there may be a problem of different weightages. Here, we are using the satellite imagery, high resolution, and hyperspectral as a proxy. So they are shifting the concept like the biological species to the virtual species. The virtual means the spectral species. And the actual data is coming from you people only, the ecology. So we are using the tree mapped plot data. And then initially we are calculating the Shannon-Weir index alpha diversity for the plot. And also we are calculating the alpha diversity for the spectral species. Then we are relating a few minutes, sir. Yeah. So this will not only generate the information for the required plot, you can able to upscale this information to the entire sanctuary or the district, okay? And also it will give you the accuracy, like regression value, how much it is relating with the actual data. So this will be useful for the management to identify the areas of high productivity and high biomass. Then another important application is the forest fires. The remote sensing became quite popular because it is addressing the many natural disasters, and also like including the floods, landslides, and then like forest fires. So this map shows that the fires are happening throughout the year, may not be in the same place, but somewhere or other place. Then what the data will helpful is, now you can use this open source data. You can download into your QGIS, and you can use the open source tools like space-time pattern mining technique. You can able to generate not only for your Kerala or Western Ghat, you can able to generate information for the global scale, because data is free. Now several cloud computing platforms also coming. So you can, your laptop may not be having sufficient disk space. So you can depend on the cloud computing also. So that will generate the patterns for the like fire regimes or any kind of application like habitat suitability, and you can able to generate for the global scale. And like if you are working on species, you can make use of the data from GBIF portal also. Nature Conservancy portals, they are giving data, map of life. There are many data sets are available. And you can able to see that management effectiveness in the declared wildlife sanctuaries or national parks or the Ramsar sites. So here I'm showing that the area growing up for the Prosopis juliflora in 𝐂𝐍𝐂𝐃 National Park. So this was done by one of my MSc student. She was from Kerala only. And I fortunate to guide 29 students from Kerala. I guided 66 students for MSc dissertations. Almost like 30 people are Keralites. Yeah. So like when we are now going to the new era, we have the access to the Google Earth images, Bhuvan high resolution, and also commercial platforms also providing the sub-meter resolution data. So you can able to map the features which are not possible earlier. So now again, now we have to increase. Here it is like a human-made landscape. And we want to link with the ecosystem services and the demands for the local people. So now we need to have reference landscapes, not only for the forest, like Dr. Srijit is doing for the native forests, evergreen forest, by establishing the large plots with CTFS protocol. But now we need to go for the larger plots, even including even KFRA campus. That will give you the actual information. You need not to map the trees inside forest. You also map in the human-dominated landscapes. Okay, that also will give you the changing patterns in the resources. So then, as the resolution is growing up, you can able to extract the tree crowns, and you can able to estimate the biomass at the tree level also. Then you can use the two types of data. This is an endangered chimpanzee species. So here, chimpanzees are living on the nests of the trees. Okay, they will build their own nest like birds. So once the nest will be only like branches, it is collecting the leaves and the branches, they will become dry. Each chimpanzee is living on one tree. Okay, now because it is an endangered species as per the IUCN, I can't able to count the number of chimpanzees because the salinity measure is good for discrimination of plants, not for the animals much, except few studies for like elephant and few birds, but mostly it is for the plants. So now I can use the UAV-based remote sensing data or very high resolution data like a SkySat 30 centimeter. So I can able to detect the number of nests of the chimpanzee, so that is equal to the number of the chimpanzee populations. The species distribution modeling, I will not go into the time constraints. Sorry for I thought I took more time. Yeah. Then the models. So now it is an era of collaboration. Like you know, GBAF, Global Biodiversity Information Facility, they are giving some data. You may not get the comprehensive data. You have to use it as a additional data only, not like prime input. So you collect the prime data from the field and some data from the botanists. Then try to generate the species distribution models. Okay, it is for both science point of view and also for the ecological point of view. It will give you a lot of knowledge that what is going to happen to your species in near future. Okay, like the prosopis is going to expand. It means it is going to replace our native species. Okay, like that you can able to model. So this is my last slide. The future directions. The biodiversity is not like not from the expertise of only taxonomy. It is the people from the like molecular biology, or like I said, like integrative taxonomy concept, and also climate. We need the expertise from the multiple disciplines. Okay, so now we recently published an article with the name like essential variables. It is not that only essential biodiversity variables of structure, composition, phenology are required. We need to integrate environmental impacts. So we have to try to, in the very near future, we need to study all the essential climate variables, okay? Essential geodiversity variable, essential water variables, and how they are affecting the biodiversity, okay? So this is one of the challenge. Then the GeoAI, now the new term came as a buzzword, geographical or geospatial intelligence, AI, artificial intelligence. It is nothing but your remote sensing, GIS, and artificial intelligence. So they are going to suffice and provide the requirements both for science and the resource management. So thank you very much. Thank you very much for your time.
Thank you, Doctor. In fact, we have time for 2 questions. It is a very elaborate lecture. We'll make a comment on it later. But anyone, quickly? Because look at the fund of knowledge available with him. I think youngsters everywhere should make use of him. Afterwards also, after he comes out, he says so many PhDs guided by him. So some of you should think of joining him and getting knowledge out of him. But any question? Yes. Youngsters, then we'll come to Dr. Nandakumar later. Are we to point out who should ask the question?
I think everyone understood, sir.
Yeah, they understood very well, but yes, thank you.
So I'm basically from a molecular biology background, so I was really interested in when you said about the plotting forest fragmentation and connectivity plots. So I would like to know which would be the connectivity indices that can be used so that, like from the data available, so that we can correlate it with the genetic data, which would be the connectivity indices that
Yes,
can be.
yes. Like I said, like evolution. Initially, we are only like identifying the plants based on morphological features. Now, like you are asking an appropriate question of this time, that now even the landscape ecology is also gone beyond. We have like the concept like phylogenomics and studying like landscape genetic diversity. Okay, there are few studies by even CCMB, Center for Cellular and Molecular Biology. They are also trying to utilize the GIS and collecting the genetic data or some kind of the appropriate data for like elephants, tigers, like they are saying like the tigers in Simlipal are like inbreeding depression, some kind of, I don't know.
Yeah,
But yeah,
they'll be like
yes.
estimating the gene flow,
Yeah,
and
yeah.
then they'll be plotting the connectivity.
Correct, correct.
Yeah.
So such kind of, you can make use of same principles.
Yes.
The principles remain same, but you can make use of these advanced tools which are available in open source.
So
And,
you
yeah,
said
you
something
can,
like one database like FracStats. So I would like to know whether there would be these indices values in that database.
No, the FracStat is only for numbers mostly, like MSPA, morphological spatial pattern analysis. It is available through the Guidos toolbox. Okay, so you can download that. You will be giving your data. See, computer or the image cannot identify itself something. We have to tell that it is an elephant, it is a tiger, something.
Right.
Then the appropriate ecological attributes. Say, for example, like I'm an ecologist, I'll use the information of elevation, phenology, species identity. So you can give any kind of attribute to the system. It will read and it will generate the hotspots. Okay, hotspots of connectivity or hotspots of degradation. Anything you can generate in a very less time.
Okay. Thank you.
Yeah, if you need any collaboration, you can contact me.
Sure.
I'll definitely help anyone, everyone. Yeah. Yes, sir. Yeah, thank you, doctor. Yeah. Yes, sir. Thank you, Chair, for the time. Yes, sir.
So, thank you, Dr. Sudhakar Reddy. That was a brilliant talk in the morning. It pepped us all. And now we have another excellent friend and speaker, Dr. Gopakumar. I think many of you may know him.
Let me start by thanking 𝐓𝐑𝐿 for giving me an opportunity to speak my mind on this. But let me say, I was just thinking what I should be talking about. But my basic training in forestry led me to just think, because GIS, remote sensing and all, I think we collectively call it as geoinformatics, like it is a technology. It has pan-science application or pan-social science application everywhere. It's a technology. Whatever data you have, you can use it, and you can use it wonderfully well to the betterment of science, any applications, governance and all. I think as youngsters attending this program, understands this applicability more than my generation and all. I am reminded of my remote sensing classes way back in 1989 and all, when they were showing just false composite pictures, black and white. All we understood was we were just sensing from a remote area and just talking about that. So so that led me to this particular title. I thought we should go beyond mapping, how we can use geoinformatics to drive forest strategy and action. So that is the title I want to say. I have structured this presentation like this. So when I was given this challenge, I thought, okay, what are the current challenges in forestry right now? So I'm flagging that at the start. Then I also thought, what could be the future strategies? So we are aware of all these things. So I thought I will talk about that briefly. I know I have 20 minutes time. And then I picked up a few case studies highlighting the applicability of this science, which I believe, to the best of my understanding, which I believe that there are some directions or takeaways from these kinds of published literature which I am going to show. Then I thought I'm always a person who believes that technology should be helping the people. And forestry is a land use science where we manage land, and there is involvement of people. So how best this technology, the science or the data connected with forest can be used to make people more aware and get them involved in the management process and generate the best forest management decisions. So this is how I have structured my presentation. So I was talking about the forestry challenges. I don't think there is nothing new in here. Most of you might have actually understood this. We are talking about climate change, and we know that there are a lot of issues connected with unprecedented rainfall, unannounced rainfalls of huge nature. Then there is a drought. Our rainy days are changing. There is no pattern now happening. So there are a lot of shifts happening. And GIS can actually, geoinformatics or geomatics can actually sense that and use that information for better management decisions. Now coming to the invasive alien species, I think all of you are aware. We have all sort of classical examples. Some of these species we know. Some of the species are waiting to be discovered. They are actually doing a lot of things. We cannot understand what is below the ground. We are microscopic organisms, all that. We are talking about carbon markets, green credits. A lot of awareness programs are happening, and there is a lot of information there. And what actually is happening, how can we account, how can people benefit out of this? All these can be actually addressed by geoinformatics, and that is happening also, but there needs to be better decisions. That is what I understood from my readings and all. Then the eternal question of development versus conservation and all. We need to strike balance, and for that we have been using geoinformatics, but always with refined technologies. Dr. Reddy was talking about the I forgot the term, some AI-driven geoinformatics and all. So better information will be available there. And finally, erosion of genetic, we know that forest is a gene pool reserve, and we are losing that. We are identifying some of the losses, but majority are being lost. So that is one thing that so coming to the future challenge, again, people will be more interested. I have planted a few trees. How can I get carbon credits? And also better accounting and better, showcasing and payment. All these can be done using carbon finance. Then again, another interesting thing I thought was because in governance also this geoinformatics and AI. As an administrator, I know the power of AI in helping me to take decisions. Means like several other circumstances I can use this technology. So why not in forest governance? Why can't we use that? Then we are also seeing urbanization happening even in Kerala. Recently, the government has received the report on the urban commission. There is a lot of urbanization happening. So how urban forestry has to shift to new level. We are still talking about the social forestry of the 1980s, but now it is a time of smart cities and all, where we need to have geoinformatics here. Then species range shifts, that is an area that we always are very interested in this. And again, institutional capacity, you can read that along with the number 2, how we can use that. Now, I move to some of the case studies where I think some of the information provided here could be useful for young researchers. I found this work by this Chinese workers very interesting, because the novelty is, instead of just detecting what is the change, they were accurately trying. They claimed that they were actually trying to understand what kind of disturbances were happening. Like if the pest occurred, what kind of a kind of a defect has happened, and how it is happened. A sort of a dendrochronological assessment of the situation you can. So this is what they say. So we can actually use it to detect illegal logging, fire and pest incidences and all. Rather than just focusing on change, we can identify the change agents also through application of this geoinformatics. Now again, this is a work that was done in France, and where they used information, classical physiology plus geoinformatics to understand the urban tree management or urban tree health. They used several indices like leaf chlorophyll, leaf area index, and canopy chlorophyll content as key indicators. I think this is just an indicative list. We can expand these kinds of indicators, and we can use how we can understand, because as I already pointed out, urbanization is a reality in everywhere. By 2050, most of the world's population will be migrating to the cities. That's already happening in Kerala. So urban forestry in Kerala also had, the forest department can actually use this information. So this is one example. I think all those geoinformatic information which I've highlighted in blue, the GIS researchers might be able to understand better than me. Now, this is again another study done which the researchers claim that they have used the P-band tomosar. I just heard that we have the B-band, P-band, L-band, lot of new technologies, satellites have been launched and all. So they use this kind of data and use it to calculate better the above ground biomass and all. So this is one application where instead of just trying to understand the basics, we can go for refine our approaches and bring in more clarity and quality to the data that we can use. So that better data means better management decisions we can use. And again, one of the after effects of this development and all is the loss of habitats, fragmented habitats, small habitats, all those things. And we are talking about lost corridors and all. So this study actually was trying to integrate the multi-species movement, habitat suitability, and restoration simulations using RS and GIS to identify and prioritize functional forest corridors under habitat fragmentation and climate change. So the point here, the takeaway point here is that we have small fragmented, especially in Kerala, the debate is usually like we have a combined spatially explicit framework. I think this is one area where if you are in wildlife or conservation planning, please read this particular work. So that can be, that I think that has got a lot and all. So these are some of the classical works which I wanted to just highlight. As I already mentioned, my idea is to draw your attention to these possibilities. You can scale it up, especially with the latest technologies and all. You can actually use it to take better management decisions. Now, from these theoretical works and all, I just moved to what I believe that GIS and AI and all these geomatics combined with AI can help in taking the findings to the actual users, including the common people. So this is one thing which I thought will be useful, like a GIS-enabled dashboard where actually people can use it like a sort of a Google Map-like situation, which can be used offline and all, where you share basic information like the forest finch communities, if they are using it, they can access these maps and say, understand whether there is a fire alert there or some actual fire is happening, or if there is a wildlife crossing happening, all those things. This is basically a database that is supplied from from the department side or the government side, while this one, this is again also offline, where this is like a Google map where the people can actually input data. Like if there is as we move in the forest fringe areas, there is a wild animal crossing. Immediately they can enter the location. You don't need an internet. You just leave it there. When you reach a place where there is internet connectivity, automatically the map gets updated. So where are the NTFP zones? Where are this particular fruit has become ripened, so it is ready for harvest, and conflict zones, wildlife movement, all those things. So this can be used by the forest fringe communities. And taking it a little bit further, like in the forest range offices, why can't we go for, again, real-time data rather than restricting all these findings to our boardrooms? Perhaps this is happening. I'm not very sure about it. But if you can give real-time data to their desktops, even in remote areas where they can use a mobile phone and get a connection and use this kind of information, they can actually, like in my range, if there is a activity change, there is a logging happening, or there is an elephant movement, the satellite can input and give those information on my desktop. I can tell my men to go there and take some good decisions. And as I said at the opening, as an opening statement, I always believe that you should take people along with you. See, you want people to get involved in conservation and all, and you say that the forest fringe communities are always at loggerheads. Not exactly. If you can rope in their expertise information by sharing information. So this model I am talking about, sharing information like a kiosk or something like that, where you can give information about the forest nearby. See, I am a person in the forest fringe community. How can I know with my layman's knowledge what is the wealth? Is it a tropical moist deciduous, whether it's a tropical evergreen forest or it's a shola forest, any type of forest. But if you can provide such information, I am more aware. I know that I am also inclined to give in my energy to support this very valuable information, because these are all the information connected with it. So this is one thing I thought will be interesting. And again, another type of information, like when you are having all this color-coded information being shared in your JFMC meetings, BFM meetings and all, people will be able to understand that. See, in a participatory approach, basically these PFMs and all are participative, but what exact participation is happening? when you are wearing a uniform and going there and talking to people, they will not participate. They will not open their mind or get involved in the participation. But when you share these simple color-coded maps and share it to them, you can actually get their support and get their inputs, which you can use actually for resource planning and community-led management. So that is what I thought about this. Then leaving the forest and coming to the city forest, I believe that the social forestry concept of the 1980s has changed, and urbanization is a reality. The urban population is going to stretch and expand to the forest boundaries. So what is going to happen when we have a large-scale migration or urbanization? Easily geoinformatics can predict and be ready with all the information. That is what I wanted to throw to you through the slide. Now again, as a fallout of the same activity, you can expect that this is already happening here or elsewhere in India, that there is a lot of landscape changes happening. Like there is a demand for more building materials. And where are these going? They are going to the forest fringe areas, not exactly to the forest, but to the forest fringe areas where immediately real-time data you can get and be aware of what could be happening, like there is a spread of invasive alien species. If Senna has moved to a new place or Hypostus have moved to a new place, we can easily detect or predict, simulate or predict, and be ready with, armed with all this information. Now my final slide would be like, again, we are talking about smart, resilient cities. Urban planning is a reality. UN has got a lot of online courses. Some of you might have already done that. There is a big talk about urban planning and all. So we should be aware of the possible urban heat islands happening. What should be our strategy for that? What could be the green spaces that has to be sacrifices when we build our cities or expand our cities or modernize our cities? All those information can be taken. Again, if there is link it with that study in France where they were monitoring the urban tree health and all. So a lot of things. All these are not standing, all these slides I believe are not standing in isolation but are well connected, and this geomatics being an information, being a management tool, everything, all those, it's a multidisciplinary area where you can actually bring in all these experts and work for the betterment of forest management. I am sure that we can bring a lot of better forest management models through this integration. Thank you very much.
Thank you, Doctor. It was very wonderful. Some of the components that you spoke are very important, particularly when you spoke about governance and urbanization. We'll come to you, but we would definitely like, I think already there's one question that has come up. I'll read out the question. Can remote sensing data be used as in detecting and monitoring invasive species? I think Dr. Reddy also had spoken about it, but yes, they want your thought also.
I think it is possible. I think he has already shown that it is possible.
Yes.
All these things are possible.
I think, yeah, but they wanted this question to go to Dr. 𝐓𝐑𝐿𝐾. Who's that? Anyway, they both concur that it is possible, and I think a lot of work is already happening in the area. Yeah. Any other questions from the?
Sir,
yes.
may I?
Yeah, please.
Sir, how effective is mulberry leaf trees? Like, how effective will be the mulberry leaf, growing the mulberry leaf trees in cropland area, the border areas, in mitigating wild boars and elephants?
Yeah, that's a different question. Frankly speaking, I haven't come across any literature that if you grow, I think I understood your question correctly. Like
Yes,
if you
sir.
grow mulberry plants on the forest fringe areas, wild boars will not come. That is your question, isn't it?
Yes, sir. Like I heard like the smell of those mulberry leaves are so bitter. Like the taste of leaves are so bitter, so the elephants won't able to have it as food.
Is it elephants or wild boar you are talking?
Sir, I'm talking about both of them, sir. Like if they are put up in the border areas as a like gateway, so they can't able to trespass within the region.
To the best of my knowledge, I haven't come across any literature or any news suggesting that if you plant mulberry, this will act as a deterrent. I am not aware of. I'm sorry about it. Somebody else may be able to answer if they have come across it.
Any other questions?
Thank you, Doctor. Thank you, sir. Thank you.
Thank you, sir.
Friends, we are coming to an end of this session. Of course, I should say both the speakers gave us so much of thoughts for the morning and for our future. And Dr. Reddy has probably shown the scope that GIS, RS has so much of potential, both for urban planning, rural planning, forest biodiversity, tropical forestry conservation. He started off by telling that our research should not end up in just publications, that there should be support to policy also. That's a very brilliant statement. I think we've been always craving for that, because let not publications be confined and locked within an institute. It should come out for policymakers, because I know during the time when we used to have budget when the budgets are going on, or when the parliament is in session, or when the assembly is in session, the type of questions that are posed to managers. So this interaction between policy, managers, governance, publication should all come on the same platform, so that we are able to work as a team for the best of the country. Then of course, Dr. Reddy highlighted all the areas where today ecosystem structure, function, and composition, the way that GIS is being used, and biodiversity as an umbrella, the type of work that's happening in these areas. And he went on to show us certain brilliant areas that you should look for. And the geobond is an area. And then look for free sources from which immense work can be tapped. He went on to tell us about tree mapping, and then the community diversity, forest fire. I remember today in almost all the forest departments and almost all the sectors, rural department, everybody has got a GIS wing and a GIS platform. But yet there is no time for them. They are only firefighting. But good work has to come from people like us, who have to probably support the GIS labs, etc., of the departments. So it is just not the forest department. We may have to help a lot of other departments when it comes to landscape level planning for biodiversity conservation. And then the type of resources that we are, may it be lakes or ponds, wetlands, marshlands, in all these systems today GIS and RS is getting applied. And but one thing, what we really require is ground truthing. And this is where probably the youngsters should put your land on. Forestry today needs what are called as forest foot soldiers. Technology is available, but I'm also afraid that your reliance on ecology will be too much on GIS base and RS base. Me as a forester, I have always said, you know, we talk of a bedside clinic. I was talking to an old-time doctor. He said, the best of the doctor is one who will always go around the patient. Today's doctors, you know that the moment even before you step into the room, he starts writing his prescriptions. But I have seen old-time doctors. They just don't give you a prescription like that. The patient is on the bed. He takes his students. They will just walk around the patient at least 5 to 6 times to know from hair to foot what sort of problem the patient is suffering, and before he comes to any conclusion. I know a doctor who was silently watching his students. And then after looking at the patient for a long time, the students could not say what the problem was. The body was yellow or it was bluish. They kept on telling so many things. Then the doctor said, you foolish guys, so many of you are young doctors, and tomorrow going out, you don't know what the problem is. It's a snake bite on the leg. Look at the blue color that is happening. So, this becomes clinical investigation. What is called as clinical doctor becomes important. So also, we call ourselves as food foresters. So you may be in biodiversity, you may be in bioprospecting, biotechnology, whatever it is. This tool is available to all of us, GIS and modern tools, AI, etc. But do not forget that you should breathe forest, you should smell forest, you should touch forest, you should feel forest, and you should put your foot in the forest. So going in and understanding, so then becomes the right mixture of RS, GIS, and your food forestry. And therefore, that's what is called as ground truthing. Brilliant was the talk of Dr. My friend, I told you, sometimes, as age, we forget names also, Dr. Gopakumar. And I know his transition, he's touched almost every field and now come to even discuss on this. But his emphasis very on governance, I think this is very much required. I really didn't want to go into the length of the talk by Dr. Reddy. Please look at the slides that he gave us. You will all have to go and talk to him whenever you get a chance and pick him for very good things. But Dr. Gopakumar is right close to us. You can always make use of him. I like some of the slides of Dr. Gopakumar also, like the village level kiosk that he was talking of. And, and particularly, urban tree management. Urban forestry is a very big challenge. Often we think that, I know when I come from Chennai, and a place like Chennai, most of us keep discussing today about rooftop gardens. We talk of, how in Singapore, they have what is called as pollinator pathways. Every road is lined with those trees and, multiple bushes, etc. So they connect. Today, yesterday there was a long talk on corridors. How do we really connect the rural forest with urban forest through pollinator pathways? So design your trees, bushes, shrubs, herbs such a way so that the rural-urban connect happens. So all these tools and technologies are available. Youngsters know the strength of these tools, but they should be rightly utilized, channelized for the best cause, so that tropical ecosystems, that's the idea of this conclave, very well thought out. I always keep telling KFRA, who else can do this? And now we have this, our Manuti, Agriculture University also. Two huge giants in the area of tropical forestry. And then across we have the Institute of Forest Genetics and Tree Breeding. All of us should be working together. And we should be, yesterday somebody was talking about empire building. Empire resources for tropical forestry can only come from this institute. I really do not want to talk much. My organizers sharply looking at me. So thank you very much for this wonderful morning. And I should say both the talks were brilliant. We all benefited out of it. Thank you so much, sirs. Thank you.
So with this, we are coming to the end of this session. So I thank both the eminent speakers for their thought-provoking speeches. And I also thank our Krishna Kumar sir for the very efficient conduct of the session and for the excellent concluding remarks, sir. So now that I invite our director, Dr. Kannan C.S. Warrior, to present the mementos to the speakers. I invite Dr. Sudhagar Reddy sir for accepting the token of appreciation. Next, I invite Gopakumar sir. Thank you, sir. So with this, we will break for tea, and we are supposed to assemble back by 10:45. So the tea is being arranged just outside this auditorium. So all of you can come back and assemble maximum by 10:50 so that we can have the next sub-theme session.
I would request everyone back in the auditorium for our next session. Kindly requesting everyone to be back in the auditorium for the next session. So welcome back, everyone. We'll be moving on to the next session. The second session is on the next generation genomic tools in forestry. This session focuses on the emerging role of NGS tools in forestry research and management, highlighting how advanced genome-based next-generation sequencing technologies, genome editing technologies such as CRISPR-Cas9 systems, RNA interference, and molecular diagnostics are transforming tree improvement for biodiversity conservation and climate resilience. So we have with us today 3 distinguished speakers in this area. Dr. Matish Nambiavital, who is the Chief Scientist and Head of the Plant Biotechnology and Cytogenetics Division at the Institute of Forest Genetics and Tree Breeding at Coimbatore. And we have with us Dr. Madhumita Dasgupta, Chief Scientist, Plant Biotechnology and Cytogenetics of the Institute of Forest Genetics at Coimbatore. And we have Dr. Manju Elizabeth, who is an assistant professor at the College of Forestry, Vellanikara. So I'm extremely happy to welcome now the moderator of this session, Professor A.V. Santosh Kumar, who is Professor and Head, Department of Forest Biology and Tree Improvement at the College of Forestry, Kerala Agriculture University. Over to you, sir.
Good morning, everyone. During the course of the previous sessions, what we have seen, this conclave has actually taken us through a very coherent and a timely journey through contemporary forestry science, grounded in things like ecology, climate change, land use, biodiversity conservation, declining ecosystem resilience, human-wildlife conflict, while basically grounding these discussions in the realities of forest management and policies that exist today. Subsequent sections, if you have seen, expanded these perspectives by examining ecological process, restoration strategies, productive enhancement, integration of traditional knowledge with modern scientific approach. And the previous session, we actually flew over the skies. And talked about applications of modern technologies like remote sensing and GIS. Okay, now we have a felt need to move and have an understanding of more predictive, precision-driven, scalable solutions for forest management. And that brings us to this session, which talks about the next generation genomic tools in forestry. Forestry, as you know, provides very unique challenges because of its very long generational gaps, highly heterozygous genomes, complex genomes, complex pollination mechanisms, very strong environment genotype interactions. Where in the conventional breeding and improvement program, although they are successful, often are very slow to respond to rapidly changing climatic and pest pathogen regimes and all. Advances in genomics now offer us tools to fundamentally alter this trajectory. And in this session, we will explore how modern technologies like next generation sequencing, genome-wide association studies, genomic selection, transcriptomics, other omics technologies, emerging genome editing tools, they all reshape tree improvement, conservation genetics, and the adaptive forest management. The speakers today bring in expertise spanning molecular biology, quantity genetics, and applied forestry, and will bring us understanding both the promise and the practical constraints of deploying genomic technologies in forestry research and management. With that, it's a great pleasure to invite our first speaker, Dr. Matish, who will be online. Is he online?
Sir.
Yeah.
Namaskaram, sir.
Yes, yes. I'll introduce
I
you.
hope I'm audible.
Yeah, you're audible. Dr. Matish is a chief scientist at ICFR Institute of Forest Genetics and Tree Breeding, Coimbatore. He is a recipient of several prestigious international awards, including the Fulbright Nehru Academic and Professional Excellence Fellowship, DAD Fellowship, and DBD Young Scientist Associateship. He has pursued advanced research in France, Germany, and USA. Dr. Matish is a pioneer of genome engineering in forestries in India, with landmark contributions in RNA interference and CRISPR-Cas9-based gene editing, particularly in eucalyptus and casuarina, aimed at climate resilience and improved pulp food. Matish sir, you have 20 minutes for presentation, and let's hear him out.
Very good morning to all of you, respected Director Dr. Kannan Chandrasekhar Warrior, my dear friend. Distinguished luminaries there. I'm sorry that I'm not there. I had a broken back due for yesterday. My apologies for not being there. I know Dr. Krishna Kumar sir is there. My respects to you, sir. My respects to the KFRI scientists and all of you there. Dr. Suma, who invited me, thanks to you. James, happy to see you. So I will just share my presentation. I hope it is audible. I mean, visible there. Give me a quick second. Is it visible to all of you?
It's coming up.
Yeah. So I am from the Institute of Forest Genetics and Tree Breeding, and I would like to share my perspectives on engineering genomes in forestry. I've been given 20 minutes, and that is a small time. Anyway, I will just try to encapsulate what I have to share as quickly as possible. So Bill Gates told that the most complex genetic program on planet is the genetic code, and it has been organized, and we have the promoter, the coding region, and the terminator, which we call it as a gene. There are regulatory sequences as well, and we know that we have the RNA, and the RNA produces the protein. So if we have to engineer genomes, then we can degrade this mRNA, and then we call it as RNAi. I'm not sure if the whole screen is visible to all of you. Yes. Okay.
Yeah, now it's visible.
Yeah. So we have the RNAi tools that target the mRNA, and we have the CRISPR editing tools that target the genome sequence. So we could target the promoter, and we could target the coding region as well to alter the genome. And this could be a transgene, and then we call it as transgenics. So to go about this, we recruit the help of nature's genetic engineer, which is the Agrobacterium tumefaciens, and that has the TI plasmid, in which we carry the payload in the form of the tDNA molecule, or the transfer DNA. And that transfer DNA can carry the gene of interest or the silencing construct. When we use a silencing construct, we mean silencing of the RNA molecules through the RNA technology, or the gene editing constructs. So we launch, we use the agrobacterium, we put this binary vector, and the binary vector has the tDNA containing the reagents that will do our RNAi or CRISPR-Cas editing. Now the critical part is exactly this. We transfer it into agrobacterium, and then the tDNA moves into the cell, and this regenerates into the plant, into a plant which is transgenic, or which expresses the RNA construct or the gene editing reagents. And the tDNA carries the selection marker, which is the plant antibiotic selection. And that makes us helps us to regenerate only cells which have been transformed. Now, if for all genome engineering techniques, the bottleneck lies here. It is not easy in forestry species. It's very difficult. So recently we have published an improved transformation protocol in eucalyptus. So we use sonication, used biochar, vacuum infiltration, and we could get a transformation efficiency of around 3.9%. So if you see the whole flow chart, it takes around 320 days just to get the shoot alone. You can see that the roots are not formed. And here you can see the cells that are in antibiotic selection media. Many of them are dying because there is no antibiotic selection marker, and some of them are regenerated. So this is the bottleneck in forestry species. Now, transgenics are important not only for modifying a desired trait, but to understand the function of a particular gene also. Now, if we knock out a gene, then it has an effect on the phenotype. So we need systems that can help us to quickly regenerate a plant expressing a foreign gene, and then we need to study the effect on the phenotype. So our lab has been instrumental in developing such a protocol in eucalyptus for the first time, and that is being used in other labs for lignin, understand the function of genes involved in lignin biosynthesis. So here we use agrobacterium rhizogenes, and that is used for generating roots quickly from an already formed shoot. So this sort of reduces a 2-year time period to generate a normal transgenic plants to 6 months. So we can quickly evaluate a composite plant for its function, for the gene's function, and the gene expresses in the roots. So we see the green fluorescent roots because our tDNA has the green fluorescent protein expressing. So we can remove the roots that are not transgenics, and then we have a whole plant. And this can go for evaluation, phenotypic evaluation. So this is in eucalyptus, we were the first to do this. Now here I gave an example of tDNA expressing a foreign gene, a GFP gene. How do we introduce a gene silencing constructor? Here, suppose, let us say that this is the mRNA and we want to degrade this. What we do is we take a portion of the gene that codes for this mRNA, put it in inverted orientation, and when it gets transcribed into a single-stranded mRNA, because of the inverted orientation, we have a double-stranded structure that forms because it complements on itself. It forms a hairpin-like structure. And when the cell detects a double-stranded RNA, it has its own innate mechanism to sort of degrade it into 20 nucleotide siRNA molecules. And because this mRNA has been derived from parts of the mRNA that we have designed to degrade, it goes and binds to the mRNA and causes degradation of the mRNA. So it is, I put this in a very simple language. Now, how about gene editing? So what will the tDNA carry for us to create gene edits? Now, I told you an important requirement of RNAi is to introduce a double-stranded RNA. If you introduce a double-stranded RNA, we have RNA interference and gene silencing happens. In case of gene editing, we have to introduce a double-stranded break. So this is made possible through a endonuclease that cleaves both the strand, which is called as the Cas9. And this Cas9 is guided by a guide RNA that goes and finds the complementary region to the guide RNA. So the guide RNA is designed to target this DNA, and this has complementarity. So the Cas9 will make a double-stranded cut, and the cell doesn't like a broken DNA. It immediately repairs it through a technique called as non-homologous end joining, and we have a mutation there. So it is possible to generate a mutation precisely in the gene, in the region that we want to. Alternatively, if we are able to introduce a transgene at this break, we can create transgenics also. So now the point is that the advantage of gene editing is that we have introduced tDNA to introduce the CRISPR-Cas reagents into the plant cell. If that is there, it is a transgenic plant. But because the gene edit happens in another chromosome, while the tDNA is integrated in one chromosome and the gene edit happens in another chromosome, in the subsequent generation, these chromosomes segregate, and we can get a plant which doesn't have the tDNA having the CRISPR reagents, but which has the gene edit. So that is the whole point. So after flowering, after getting a new plant, we have a plant which has only the gene edits and not the tDNA. So that's the beauty of this approach. And of course, in tree species, this is not easy, correct? Because flowering takes time, and when will we get the next generation, next progeny, it is time. And so our lab is working on working out these issues through enhanced flowering and various other techniques like ribonucleotide proteins. Here, instead of introducing a tDNA or through agrobacterium, we have the Cas9 protein and the guide RNA that is complexed in the lab in tubes, and they are then introduced into the plant cell. So this has been successful in wheat. In our lab, we are just starting this work. There are many other approaches, but but the possibility is there. There are a lot of issues like chimera that happens. So that is a separate issue for discussion. So essentially, gene editing can help in creating mutations. We can correct the DNA also. So these two are called as SDN1 mutation and SDN2. We can also introduce a foreign gene, and the government of India has relaxed rules for the SDN1 and SDN2 type mutations. So that is something that's a breakthrough. They have launched rice varieties also. Now you can see the process of regulations of transgenic crops. It is my friend Dr. Kumar at TNU says that it is almost like releasing a drug for human consumption. So that has literally killed the field of transgenic research. It has been very tough for researchers to get funds. With meager funds, we do it. Now, what the 2022 guidelines have done is, if you are able to get a transgene-free plant, my director can tell that the plant is without transgenics. It can go for multi-location field trials and commercial release. I don't need IBSC or RCGM. So that is a breakthrough. Thanks to the government for doing this, because it has been a struggle in transgenic crops. So I think it's the right time that the forestry scientists invest in this technology. And now coming quickly to the applications in forestry, we work on salt tolerance. We started on pulping traits. I'll be covering on all these things. We have aims for pest and disease control, invasive weed control, biomanufacturing. But I'll be talking about a few things. And of course, James told about the diagnostic purpose. I'm not touching that. So in my lab, the focus is on salt tolerance and can we reduce sodium uptake at the roots. We are hell-bent on finding methods to do that. That is the approach. I would say that we need technological solutions for problems that don't have solutions at present. We need high technology only for this purpose, and I believe that climate resilience is in one such area. This chart showing how the species, the plants and animals vary in their salt tolerance. You can see Rhizophora mangrove species can tolerate relatively high. We have the higher plants like Atriplex and Saliconia tolerating up to 1.2 molar salt. And we have Artemia saline, the animal, the prawn-like crustacean animal, small 1.2 mm size, most tolerant animal on the planet. So they have adaptation mechanisms. Why not we engineer them in the roots? So here we just show a project that we had. We can see that we have synthesized the sodium potassium pump. This pump is not there in the plant kingdom. It is there only in animal kingdom. So it is two genes. We have fused them. Synthetic biology here. Fuse them, express them in the composite transgenic plants. See, technology has to give a very significant improvement in salt. Only then we can apply it. Our results have not been so spectacular. You can see here the NACA expressing composite transgenics, but it shows that the roots are white, especially in alkaline conditions with NaCl. This work has been done by Manoj. He's there. He's presenting another poster on base editing. And using the chloride channel gene also, we see some improvement in salt tolerance, not spectacular. So our search continues for new genes, and so we expect some breakthroughs. This is another interesting work on mangroves. We know that rhizophora restricts sodium uptake in the roots, so we wanted to know the secret. So we compared a relatively susceptible Rhizophora apiculata and mucronata. We compared the transcriptome. We identified the genes that are highly upregulated that can go for transgenic approach, and we identified genes that are less upregulated that can go for gene editing, stuff like that. One gene was the peroxidase gene. It's not a normal peroxidase gene. This uses the lignin monomers. So we feel that maybe the lignification that is happening in the roots of Rhizophora mucronata may be a reason for ultrafiltration of sodium. So right now we have cloned the gene, and we have put it in a transgenic tobacco. We have to see what will be the effect. We expect that there will be less of sodium uptake. Now, when we are using a whole plant transgenic, we need to use a promoter that will specifically express the gene in the roots. So we have tested an MSPRP2 promoter that is already known to be roots preferential and salt inducible in Arabidopsis and Medicago. So we have tested it for the first time in eucalyptus. Promoter directs the transgene. So it's very important to work on the promoter also. So this, we saw that it is expressing in the veins in the xylem phloem. So it can be used for wood engineering as well. Now, there is another gene called as the 𝐓𝐑𝐿 gene. It's a sodium transporter. Can the silencing of it improve salt tolerance was another question that we asked. We generated the whole plant transgenics. We silenced the gene using the promoter, the root preferential promoter. But to our disappointment, we couldn't see neither a salt tolerance or a susceptibility in this. But anyway, the plants that we used for generating transgrafts. So this is another technology that can come in future. We have the transgenic plant as a roots stock, and we have the breeder's plant as the scion. So we are very happy with this. So this can reduce the biosafety concerns also. So here you can see the RNA plants in the greenhouse. Some GFP, MSPRP to GFP plants, the transgrafts. Srija is working on these things. And we have deleted a portion of a genome. I talked of single mutation, but our lab has generated 1,400 base pair deletion in the HKT1 gene. That's a very recent thing. And we use the MSPRP2 promoter to guide the guide RNA to the promoter as well as to the exon sequence. And the idea was to remove this part of the gene. So this part of the protein has the sodium transport properties. So remove that. So we designed the guide RNAs and cloned it into the vectors, generated composite transgenic plants. And we were clearly able to see that there has been a deletion of 1,400 base pair. Most of the articles in gene editing, you find small mutations. Here we have used 2 guide RNAs, and we have seen that there is a deletion, and then we got a downregulation of the gene as well. So I'm very happy that this happens to be the first report on gene editing in any woody tree species from India. So I'm not going, but when we tried the salt tolerance aspect, we didn't find that the plants, gene-edited plants, had an improved salt tolerance. We saw that this was, we saw that our analysis showed that it is a heterozygote. We are generating more plants. We have all heard of COVID mutations. One single mutation causes the virus to become more virulent. So the gene functions, and yet there is a mutation. So I was looking for some genes in the HKT gene which causes improved potassium uptake and improved salt tolerance. So erstwhile studies have shown that just a single change in amino acid from asparagine to serine improved tolerance at 300 millimolar. Another mutation in the serine to glycine improved potassium uptake. So can we, instead of knocking out, can we change the amino acids so that the gene remains functional and yet the sodium potassium uptake properties are not affected? So Manoj is there, he will be presenting on this. So he has generated a number, we have made the construct for this. For this, in addition to Cas9, we use a TAD-A protein fused to this Cas9. And the plants are regenerating, and hopefully we have something. Due to limited time, I'm flying. I hope during discussions I could clear out many things. So this is another interesting approach using RNAi in insects. Hibbertia is a major pest in teak. We have used RNA. We showed that there is some mortality, but later on when we were doing, we didn't know. Lepidopterans are not very good material for RNAi. In leptocybe invasive, we have generated RNA plants. We targeted 5 genes of leptocybe. We did the transcriptome analysis. And interestingly, 6 out of the control plants have been infested with the leptocybe, but none of our RNA plants have the infestation. Leptocybe lives within the gall tissue, and we have made the eucalyptus to produce the RNA constructs. So this is some light that we are hopeful. Another project that we have is on improving growth. There are some reports already by others. They used knockouts and that is improved biomass. Our lab tries to actually improve by improving GA20OX. You all know that green revolution happened in rice because of a mutation in the GA20-oxidase gene, which reduced the gibberellic acid and made the rice plants dwarf. But here we want the plant to grow taller. So our aim is to improve GA accumulation. So we feel that the GA inactivation enzymes could be targeted, could be inactivated by gene editing. So that is something that we are trying right now.
Dr. Matesh, 20 minutes are up.
Okay, I stop quickly.
Yeah.
Please give me 2 minutes. We are working on pulping, and gene drive is another approach, very interesting approach, and we intend to do that. And some perspectives are that we need to invest a lot in these technologies. We had the minister visiting us. I had proposed that we should have a genome engineering center. He has agreed. Our Minister of State wanted that we work on invasive weeds. So we put that in that proposal. Heartfelt thanks to many people here. My guide who recently passed away, Dr. Gurmurthy sir, Claudine Franch and Didier Bogus, from whom I learned RNAi. Krishna Kumar sir is there. My thanks to you, sir. You have been the most supportive person for this field of transgenics. University of Wisconsin, professor, where I did my Fulbright. I learned a lot about chemistry. My students were with me because they were going through all the difficult times because of low funding. Thank you. I stop here. We could have discussions on many other aspects. I will stop the presentation here.
We can take a few very quick questions. None?
Hello, sir. I'm Donald James, as you mentioned. Sir, I'm also working on similar lines, so I was wondering, what do you see for genome editing in the next 5 years? How far are we on lines which are possibly we can release given the fact that in 2022 there was this guidelines from DBT which has allowed us to release SDN1 and SDN2 genome edited. So what is the perspective from your point of view on release of lines, especially in eucalyptus or teak? Can you hear me, sir?
Yeah, thank you, James, for the question. I forgot to thank our moderator, Dr. Santosh Kumar, as well. My respects to you. Now, that's a very important question, James. See, in agriculture crops, the crops are already out. Okay. Now, you know that SDN1 and SDN2 should be transient free. The most important thing that any researcher should do is not focus on gene editing, but to develop protocols wherein we can have transient free gene edits. So the next 5 years, we'll have gene edited plants. Now, you see, you can hasten it through enhancing flowering. When we quickly have the flowered gene edited plants, then we have the progeny that will be gene edited. Now here again, you should understand that it is, when we go for flowering, it is not the original plant that we have. It's a new genotype, but with the gene edits, but that can go for crossing. In the next 5 years, I see a lot of gene edited plants, at least from our lab, we already have gene edited plants, of course, but they're not really good to go for. But we'll have some gene edited plants showing at least fast growth. That is what I believe. And we need to take it up for making it transient free. I hope I have answered your question. We need to work on, you see, if you're working on sandal, we need to improve the transmission protocol. That's a bottleneck. Editing anyone can do, but doing it in a particular system is the crux.
Hi, sir. So, it's a continuation of what you said. In your study in eucalyptus, when you did the salt tolerance study, you have transformed the genes from, I think it's a shrimp, Artemia is a shrimp, right? So, despite all the difference in the membrane composition, so what all challenges you faced, like when you transformed a gene from an animal to eucalyptus, how hard it was to standardize that protocol? So,
I'm really very happy with this question. This problem was very passionate. Why not we take adaptation mechanisms from animals? So this actually is 3 genes. So we compromised on certain things. It has an alpha protein, beta protein, and a zeta. So zeta is not very essential. So we had 2 options. One is to fuse it using a GSG linker, or to use a viral F2A linker. So viral F2A linker means releasing it to farmers may face a lot of opposition. So we went in for fusing these 2 genes using the GSG linker. So what has happened is, instead of a free 2 proteins, we have fused it. Maybe that is the reason why it is not that effective. One thing that is in my mind is to express it separately. Another question is, yes, this protein, we are expressing it in plants where there is a cell wall. So is that a reason for the not so spectacular salt tolerance? So certain questions are there. We have to put the gene in the plant and see the effect. We are not worried about whether it works or not. The thing is, we advance to a certain level. A number of other technologies, we improve the transmission efficiency, and we have better ways. You see, the first train or plane was not the plane that we see now. We try various iterations, and ultimately, someone will be able to do it. That is the idea with which researchers work. Thank you. Thank you for that question.
Thank you, sir. I would also like to know, what's your idea on gene-based, these kind of approaches for invasive weed removal? What is your idea on such kind of approaches for invasive?
Yes. Thanks again for asking. In mosquitoes, they have done this. Okay, they introduce gene drive, which spreads in the population, and slowly there is a lethal gene which causes the mosquitoes to die. So recently, I had this interest to work on it for the past 10 years. But now we had a minister, he told Matish, can we do it in invasive? Which I told, sir, there are biosafety concerns in that. No, Matish, India should work on this. So we have put a proposal. The whole idea is that, we use gene editing here. And the idea is that the gene drive should spread quickly to a number of plants. So they have been successful in this only in 2024. The recent papers have come in Arabidopsis. So they have used a gene drive which uses gene editing to knock out a pollen germination gene. So when the pollen germination is knocked out, the pollen is not able to germinate. Now in those pollen which has the gene drive, there is a recoded version of this particular gene which makes the pollen to germinate. So that means in a population, only the pollen which has the gene drive is able to spread to the population. You got it right. So we have a lot of population which has the gene drive, and we will have another guide RNA that will make the plant to be susceptible. Okay, susceptible means a resistant gene can be there, and that can be mutated, and the plant suddenly becomes disease susceptible. So we slowly wipe out a population. That is the idea of gene drive. In plants, they have successfully tested gene drives. They are still not used a deleterious gene for wiping out. All this has been done in Arabidopsis. We want to do this in Parthenium and Chromolinea odorata. So that concept has been submitted. So we are waiting that we get some funds in this area.
What about passing it on to further generations, like as you said, the gene drive is happening like in current? Will it be passed on to the next generation also, so that?
Yes. It doesn't follow the Mendelian inheritance because the pollen that is not having the gene drive does not germinate. There is a favor for all the pollen that has the gene drive to pollinate other species. That means the gene drive spreads to the next generation. Only when it goes to the next generation, you can wipe out a population. That's what they have done in mosquitoes.
Yeah. Thank you, sir.
Yeah, sorry to interfere because we have a limited time. Main problem is the next session has to start at 12:30.
Thank you, Dr.
A very interesting session. I'm sure that most of the young crowd has a lot more questions. Probably you can directly ask Dr. Matish. His contact details we can provide. We move on to the second speaker, Dr. Madhumita Dasgupta. She is a person with a vast experience, 27 years of experience of working in genetic engineering. Most of you sitting in the audience wouldn't be even born. So she has such a great experience. Her work has been including molecular tree pathogen interaction, marker-assisted selection, water stress tolerance, molecular karyotyping, genome and transcriptome analysis. She has received advanced training at Texas A&M University, USA, under the DBT, CRUST, and ITTO fellowships, and has been recognized with a large number of awards, including the ICFRE Women Professional Award and Plant Science Innovation Award. She also contributes actively to the national and international scientific committees, including IFRO and DBT task forces. Madam, the floor is yours.
Good morning to all of you, and at the outset I would like to thank Director KFRI, Dr. Suma, my good friend, for giving me an opportunity to be in this very important event. So today, what Suma was suggesting is if we could talk on what are the different tools available which could be applied in tree sciences. So I would say that my talk would be very generic, like I'm not going to talk on much what we have done, but I'm open to questions. You can always query me on that. So today we are looking at this particular what we call as omics technology, how that has already impacted and is going to impact the future of tree sciences. I think, after COVID, COVID was, of course, a pandemic, something which we don't want to remember. But one good thing which COVID has done is it has brought this science to the common public. I think all of us now understand there is a virus mutation, there is a surveillance, there's a qPCR, there is a CT value. I think before COVID, no one even cared about what was genomics. So today, genomics has become a common, terminology with all laymen, and they understand the power of this particular technology. So sans any biological system, this technology is considered a very powerful one. So of course, it is going to have impact on the tree sciences also. So what actually the technology does is it brings in multi-level data sets, It is multi-tier data. It brings in all the data together. It tries to explain the interaction between the different data, and ultimately what it allows us to understand a phenotype or whatever we want, a long-standing questions. It allows us to answer that. So our answers are not based on only the genome, the transcriptome, the metabolite, or anything. It is based on all the informations put together. So that's why we consider it as very powerful in terms of understanding, any questions. It could be adaptation, evolution, it could be anything. So when we consider trees, most of the time, even when I joined, probably after my MSc, when I joined into the Institute of Forest Genetics and Tree Breeding, we were all under a false notion that, working in molecular biology will be as easy as working in crops. No. So anyone who comes from a crop background gets the first shock when they enter into tree sciences, because we are, facing something which is very, very unique. Of course, later we understand it's a field which gives you a lot of information. So the first thing, uniqueness, is you have the same genus which can grow in heterozygous environment. That's not possible in crops. They are very, very restricted most of the time, defined ecological niches. But you will have tree species. You'll have the, sister taxas itself coming up in different niches. There could be some endemic, some could be broad-based. You have every possibilities in tree species. The next thing is the long generation time. That none of us, all of us agree with this. And another powerful thing is a tree species, if you see, predominantly they are outcrossed. So you have lot of progenies, lot of seeds. Now these progenies in nature face a selection pressure. And the local adaptation is a very, very powerful concept in tree sciences, which you don't essentially find in crops. So that's where we have resilience, we have climate smart forestry. So all can emanate from this concept of local adaptation, or as we say, like, the adaptation to any given landscapes. Then when we talk about the evolutionary processes, of course, it's a very complex. The genome is, most of the genomes, if you see tree genomes, are said to be, they would have duplicated. There would have been a duplication event sometime. There would have been a triplication event somewhere. What we consider as diploid, actually the genome would have duplicated sometime, millions of years ago. And sometimes a part of the genome would have duplicated. There'll be tandem repeats, like the R gene, the disease resistance in trees is tandem repeats. The lignin modification genes, some of them from the phenol-propanoid pathway, you will see tandem repeats, because those are important for the sustenance of the tree species for millennia. So they're very unique in terms of the genome too. So, considering what actually we are facing when it comes to trees and annuals, the difference is very huge. First thing is, we are looking at a very, very heterozygous genome. Like when we say eucalyptus and poplar, we today call them as crops, tree crops. I think Krishna Kumar sir knows very well. We call them all as crops, 3-4 years of breeding. We have brought down the generation time, but still they are just 3 to 4 years away from the wild. So you can understand, where is the genome? The genome is still heterozygous. It's not homozygous to make genomic studies easier. Then the evolutionary plasticity, as I said, the genome, the trees survive for millennia. Hundreds of years they may be standing in a place, so the genome has to be plastic. Its reaction time is much, much more faster than a crop, because a crop size you can see, and a tree size you can see. The signal transduction is very fast, absolutely dynamic in case of tree species, not that very robust in case of short-lived crop species. But we have bottlenecks. even full SIPs today, not very many full SIPs are available. Very few hybrid programs are there. Full SIPs itself we don't have. So where do we think of near isogenic lines, recombinant we don't have any of the inbred lines. So the genetic material with pedigree is most of the time not available with us. The genome size sometimes are very big, like in gymnosperms. And the generation time, like I would join and probably I would retire to see the first or the second generation of the breeding program. That is something we are looking at. And all the traits we look at, the breeding traits, they are all at the mature age. None of them are like in the first year we are going to get any of these traits. And for me, I would say that the biggest challenge is the funding. I think Matish has also told it. Who is going to commit a 50-year of funding for a teak breeding? Who is going to commit as a 20-year funding for sandal breeding? It's impossible. So that's where the challenge lies. Opportunities are huge. Challenges are equally, overwhelming in case of trees. So now, in spite of it, for the past, I think, 2 decades or 3 decades, there has been a phenomenal input into the tree sciences because of the advent of next generation sequencing. Because the cost of sequencing has gone down, even small laboratories today are able to take up, genome sequencing or genotyping to answer the questions much better. So we say that tree science today is more on a transitional stage. We have a lot of unanswered questions. And these long-standing questions, it will be possible if we bring in this particular technology where we can understand adaptations, evolutions. At the same time, we will also be getting what we call as stringent predictive tools, whether for conservation, to understand adaptiveness, or it could be to even improve breeding programs. So it can assist in both. So the two main research tools is when we are talking about a short rotation plantation crop where we have a well-laid breeding program, there this particular technology can be used to accelerate the breeding and breed for better traits, whatever are the target traits. And the second is when we are looking at a natural populations where conservation efforts are required, there these tools can be used for monitoring, management, and conservation. Rather, it will give us a scientific, a way of looking into conservation and resource management. So the main objectives, first is to understand, I said tree genomes are very plastic, so try to understand the genomes. Try to find the bridge between phenotype and genotype. Ultimately, what interests us is the phenotype. So we look into the genotype to understand the phenotype, look into how adaptive diversity is there, how much of efforts we need in conservation. We can have genomic tools for accelerating breeding. And also, like Matish was talking about transgenic programs, they are considered as warehouse of very novel genes, the tree species. So can these novel genes be harnessed for any transformation programs? We could work with that also. So now let's look into how it can help in breeding. See, most of the time, we say that molecular markers can be used in breeding only for marker-assisted selection. Actually, that's not the case. Marker-assisted selection is not the only way of using markers in breeding program. Of course, that's one of the main thing in accelerating breeding programs, whether we do it through QTL mapping, we do association mapping. Today, we are going to look into genomic selections. So these are all technologies which can accelerate a breeding program. But other than that, there are other ways of using the molecular markers in a classical breeding program. Okay, so they're like if you have some clonal orchards, we can identify if there is any mislabeling of clone. If there is any misidentified clone, that can be removed, because the output, the seeds from these clonal orchards are something we are looking at in enhancing the productivity. So it can be used to identify the genetic purity. If we are talking about a strictly hybrid population where both the parents are known, so we can look into the I would say, unintentional pollination by unknown pollen. We can look into that purity also in a hybrid orchard. We could look into parentage reconstruction. Now, this is becoming very popular. 𝐓𝐑𝐿-Kasabi's breeding without breeding is a concept which came in trees long back. So here you don't essentially do a breeding program, but you harness the power with the existing trials. So it allows us to do a pedigree construction, pedigree reconstruction. So you would know how much of which family is contributing more towards the pollen, which families are not, into pollen contributions. So that actually talks about the output from an orchard. So we can actually modulate it through these markers. And finally, use of these for identifying the genomic matrices. Again, this can help in accelerating breeding. So we'll look into one example. This is the one in white spruce. So here they had 2 progeny trials, 15-year progeny trials from 10 families, you can see. So they did a marker analysis and they were able to say which family actually contributes more, where there is a pollen contamination. And if we do a silvicultural thinning, what happens before the thinning and what happens after the thinning? Because when we do silvicultural thinning, it's more phenotypic. We don't consider the genetic, I would say the genetic makeup of the individual. A crooked tree doesn't mean that it is not genetically robust. We always say that in genetics. So it allows us to, modulate even a silvicultural thinning program. So these are certain areas where markers can help in a classical breeding program. Then coming on to marker-assisted selections, of course, these are the traits which are generally considered like growth, adaptability, straightness, disease resistance, insect resistance, wood quality, very, very well worked out since they are a source of timber and wood. This is one which is used. But what has happened over the period of years is that we started with QTL mapping. Then we felt, no, it is not defining properly. The phenotypic variability it defines in the field is very low. So we moved to association mapping. QTL mapping uses 2 parents with known traits. You hybridize them and then you do the analysis. Then it turned out that QTL mapping is not very successful because it's parentage based. So we moved to a population based strategy of association mapping, whether it was genome wide or candidate gene based. But even that in case of tree species is not very successful because the phenotypic variability with which it can say the seedling is going to have this trait is as low as 20%. 20% is the highest, I would say, in case of, I think, eucalyptus. So there also there was a failure. So what came next was to use another advanced technology, which is called as genomic selection. So this is the accelerated domestication model in trees, which is called as genomic selections. So what happens in genomic selection is it reduces the time for evaluation. It reduces the progeny trials, right? So you do the selection right in the seedling, you do the evaluation trial, and you straight go into propagation because trees, we have this advantage of vegetative propagation, so your clonal material is available. So we can see that a classical tree breeding, if it takes 28 years, the genomic selection is going to take 10 years. So this is the power today. Most of the researchers feel that this is one technology which can actually accelerate breeding programs. So in fact, the first example of genomic selection came from trees. It never came from crops because in crops, they don't have a time. They are not bothered about the time much. They are all short rotation. For trees, land locking for 28 years is a huge money. So genomic selection has the best impact and the significant impact in trees only. So this was the first example in 2012, rather, by Resende. The first proof of, I would say, a concept of genomic selection was given in case of eucalyptus, where they have said that they could come out with elite clones by 9th year, whereas a classical breeding, even for a short rotation crop, was 18 years. So this was, and here the advantage was the selection was not based on single trait. GS selections are usually multi-trait selections. So it would be for growth, it would be for wood properties, it could be for disease tolerance, it could be multiple, it could be for fiber biometry. So multi-trait selections are done. And they say that GS actually, the missing link in the genome, which is there in QTL and association, this uses the whole genome data. In GS, there is no, partiality that I will use only this marker, I will not use that marker. No, they use the scan, the complete genome, and then do the analysis. So the analysis is robust. The predictive models are very challenging to generate, but the results are, extremely encouraging. So that was in 2012. You see the example in 2024, they had implemented a GS in an existing breeding program of Thuja. So the Thuja's first generation breeding takes 27 years. Okay, the first breeding cycle is 27 years. So what they did was, they did a multi-trait selections for growth, heartwood chemistry, foliar chemistry. And they used the GS model, selected about 119 individuals which had these traits through the GS model. They integrated into the breeding program. So the first cycle of breeding program was over by 5th year. So this is what is, the marker technology is moving towards. So based on this, what we did is, sorry, I think I need to go back. We at 𝐓𝐑𝐿𝐵 Suma is here very much a part of our All India Coordinated Project. We had implemented the All India Coordinated Project on sandalwood. We started on 2020, just completed it in March. Now sandalwood, I don't need to introduce the species to Kerala. So our species is very close to our heart. But if you see the breeding program, even though it started way back in 1997 by IWST Bangalore, today we have no information on these breeding populations. Either they have been cut and taken off, or the data has not been collected. So we have absolutely nowhere to start the sandalwood breeding program. So when we started this program in 2020, we again started from selections from natural forests. We had a bottleneck. We couldn't do the phenotyping, sir. Because no forest department, no farmer is going to allow us really to take the core samples or heartwood or do the oil estimation. So the selections were more on phenotypic parameter, which is not good enough for a species like sandal, where the worth is only the heartwood and the oil. So, but of course, we have done the selections. At least we have the populations. 𝐓𝐑𝐿𝐵 Suma has done the assessment of the base population. We have ample of diversity in the base population. We have now created at IWST and 𝐓𝐑𝐿𝐵. But then we said, how do we go forward? We have nothing to start with. So that's where we came out with this model, which we have presented as the second phase of sandalwood, to bring in GS into the sandalwood breeding. Now we are able to do this because of few facts. The sandalwood genome is extremely small. It's just 230 million bases. You can understand it's just a couple of 2-3 times more than Arabidopsis, smaller than rice, because it's a parasite. So genome is very small, so sequencing is very easy and cheap. That's one thing. Second thing is, now we have a technology. You have a standing tree. We have a technology to assess the heartwood content with 90%, 95% precision developed by IWST. The technology is called as electric resistivity tomography, ERT. It's something like a CT scan. So you could just go, take the data, so you are able to do it. You can see the photograph there, the ERT being taken. The third technology we have is, with a 100 milligram wood sample, we can now say what is the oil yield. I think that's available even with 𝐾FRI, where a spectrophotometric method has been fine-tuned, so that with just a small piece, we are able to at least say the oil yield is high or low. So with that and the genome-wide data, so we are now positioned to implement a GS model or a GS program into the sandalwood breeding program. So this is what we are envisaging, taking the power of genomics. So where we are moving, the future of tree breeding, we were still in the genome, then we moved little to transcriptome. But what's going to happen in the coming years is that all the omic technologies would get merged into what we call as OAS, or the omics-wide association studies, where we'll be looking into epistatics, epigenetics, we'll be looking into environment interactions, try to have better predictive tools to define a trait in the field. So the second most important application of genomics is in climate resilient, in generating climate resilient trees. So all of us know that trees are essentially, as a natural populations, I think very rarely we would know that there has been a wipeout because of this disease, or usually it happens in plantations. But in a natural forest, that's very rare, because usually the diversity is very high. The intra-population diversities are very high in case of trees. And also the I would say the resilience or the adaptation is also very high. So, but still, if there is a, you can see the scenario there, there's historical climatic conditions and we have a future climatic conditions. When there is a fast change, there is a mismatch of genotypes. So how does the tree handle this mismatch? How does a population handle the mismatch? One thing is, it can migrate. The seeds, because it has the wherewithal to move the seeds to farther places, it can go to a niche which is more conducive. It can migrate, but not all tree species can migrate that far. Or it can become extinct. There are certain populations and species which cannot adapt. So in that case, they would become extinct. And the third is in adaptation. So this is where we are looking at the conservation aspect, looking into the adaptive population. So how does genomics help in it? It can help in, hybrids are known to be very adaptive. So do we have any closed taxa hybrids? Do we have putative hybrids? It allows us to know. It allows us to monitor the movement of allelic, adaptive alleles. Monitor inbreeding. We know that when there is a climate change, a population is going to fragment. It fragments, it will inbreed. So we can monitor that. And then come at scientifically informed management decisions. So it could be a decision like it allows us to unravel genomic basis of local adaptation. We know the population structure and demographies of populations. Is there a bottleneck? Is there a barrier? Is it an ancestral population? Is it an admixture population? A lot of information can be gained. And then, is there a genetic load? Is there any recessive allele which is getting piled up in a population which in future can become extinct? So all these informations it can give. And this is one technology. I think 𝐓𝐑𝐎 is also working, Suma is working, Sharath is working. This is on landscape genomics where we try to, here the selection is by the environment, not by the scientist. So we use the environment data, we use the genomic data, we do an association and say probably these are the alleles which are adaptive, and they can be traced when we do a breeding program or a restoration program. Okay, so all right. So so how does genomics, how do you consider them as more advantageous than the present existing manual, or I would say the common garden experiments? First thing is they give faster results than field trials. A common garden experiment will take us 15 years to know which is adaptive. Or it can allow us to differentiate, is it because of an environment or because of the genotype? It can allow us to break down the two major effects on the performance of a population. Identify candidate alleles. It can help in assisting migration of gene pools. Like I think there are a lot of studies of Suma which says that these are the populations where there are unique alleles which need to be conserved. There are populations in sandalwood, whichever she has identified, where there is a loss of gene pool and which will require an assisted gene flow through bringing in populations from other places. Again, when we are doing it, it has to be, just 1 second, sir, I'll finish it. So when we are doing it, we also have to see a seed sourcing, that the seed sourcing should not be from a maladapted population. That's very important in conservation. So what omics is expected to do in the coming years is we should have T to T genomes, means telomere to telomere, chromosome level genomes in species which has ecological significance. We only work on economically significant species, but we need to work on ecologically significant species. Have pangenomes. Today, pangenome has become a reality. Pangenome will allow us to know actually how much diversity is there in a genome. Use transcriptomics, epigenetics, metabolomics, and microbiomics also to understand resilience of a population. So this is the future of all tree genomic programs. Essentially, we need to come together. Individually, it's very difficult for all of us to come together, integrate, and work towards achieving both accelerated breeding and conservation. And main thing is the cost of sequencing has come down, but data storage cost has gone up. So we need to invest on the database resources. And another thing, as geneticists, genomic persons, we avoid, we don't think is very important is phenotyping. But I would say that the results of genomics can only be seen if robust phenotyping is done. A deep, in-depth phenotyping is very essential whether we do breeding or we do conservation. In conservation, you can do the leaf trait phenotyping, root phenotyping. They are very important to harness the power of genomics. Yeah, thank you.
We can take one quick question. Anyone? Dr. Madhurita will be available here. If you have questions again, you can ask her.
I'll put two important aspects. You were telling about the challenge.
Sir.
Not losing the data and maintaining the data, funding the thing, that is one thing. Secondly, again, different markers, all these things. The technology revolution, how you can bridge the gap. Well, here, particularly research managers have got a key role. That is where we are not making effort. Corpus fund has to be there because, for example, just like KFRI, 𝐓𝐑𝐿 also have got years, university has got years of experience, but there is no corpus fund. No corpus fund. So these things, when a scientist goes, finished. When an officer retire, that is the challenge we have to address.
Yes, I mean, that's a concern for all of us. We do not have this second generation. We have a second generation breeding program, but we don't have the second generation scientists to take over.
Tell you the type of work that has gone on in genomics and what Dr. Matish's area, I think it's tremendous. The House should applaud because KFRA also doing all this, and then we have our own, professors also. So all of you, I think it's a very big strength for field foresters. We feel so happy that when you told us that the, rotation time period has been reduced, all that field evaluations can be done away with. Now, these are all very happy news for us, how much of time all old foresters have spent on this, and they've gone so sad that in their generation they could not do the best of selections. Now, when you tell us all this, I am very sure, even if they're dead and gone, they'll be happy to hear all this.
Thank you, sir. Thank you. Thank you, sir.
It feels so sad when you have to cut very good speeches, very informative talks. But unfortunately, we are rushing because we do have the next speaker. The next session starting at 12:30. That's online. The speaker is from UK. We need to keep the time. The next speaker, Dr. Manju Elizabeth, is a colleague of mine. She is an assistant professor with Kerala Agriculture University. She specializes in plant biotechnology and molecular biology, with research experience in plant molecular biology and genomics, non-coding RNA and RNA interference, stress physiology and different signaling in forestry species, and molecular approaches for disease tolerance and sustainable forestry. Please restrict yourself to 20 minutes. Thank you.
A warm good afternoon to all distinguished scientists and the audience present here, and thank you the coordinators for inviting me to the talk. Okay, so I would like to compare the trees with ourselves. So like trees and us, both are facing stresses. So what are the stresses the plants facing? Mainly drought, cold, then salinity stress, and biotic stresses like pest and diseases, and anthropogenic stresses like pollution and fragmentation, etc. So when a stress comes to the plant, it tries to adapt to it or manage it. Like us, what will happen when COVID came? We will adapt to wearing mask. Before that, we didn't use that. But when the next flu come, we used to have a habit of wearing mask. Like that, plants also do. And another thing is that we are spending much if the money is with us. But just imagine a situation where the government stop the salary for 1 year, then we will adapt to the spending pattern and we will learn how to spend. So the better we adapt, we can survive further. So like that, the plants also better adaptation succeed to the next generation, and everyone not succeed, someone will fail also. So what we can do is that with the help of genomic technology, we can accelerate the adaptation or accelerate the stress management. So what is the use of, what is the need of genomic technology in these days? Because the olden days, it was the breeding program. Not like the crop plants, it is long living organism. So the breeding cycle is too long. And the genomics will help us to bypass this long duration of the breeding, and we can identify the stress tolerant genotypes, and we can predict the performance and informing both conservation and plantation forestry. So now with the help of genomic tools, we are moving from a reactive forest management to a predictive data driven forest management aspect. So why we need to manage? Because the mortality rate of trees is much more due to the stress. So we have to manage or overcome this by, we have to predict the susceptibility or detect the susceptibility before the symptom appear, because infection can happen but the symptom will take time to express. So if we can detect the things whether the plant is susceptible or prone to stress, we can manage we can start the management strategies earlier. So I'll rush to the genomic technologies or tools which are already dealt before. So advanced whole genome sequencing, because earlier crop plants were sequenced, but now forest trees were also sequenced. So this whole genome sequence technology will help us to identify resistance genes and the copy number variations and the structural rearrangements linked to the stress physiology. So the sequencing only will help us to identify what are the stress related genes or the genes which are responding to the stress. And the third generation sequencing will help us to resolve the repetitive regions, because the genome is very large. So many repetitive regions are there, and uncover the methylation patterns. Methylation pattern is also important in the stress management, and it will distinguish paralogous gene families from the common trees. Paralogous gene families means there will be duplication of the genes in the same genome. And a combination of this triad, that is sequencing triad like high fidelity sequencing, Oxford Nanopore technology, and high-seq sequencing will help us to get the uniting the accuracy, continuity, and chromosomal scaffolding. So now we will be able to identify the 3D structure of the chromosome, and the transcriptomics will help us to identify the stress response. So in transcriptomics, what we are studying, which gene is transcribed. But transcriptomics may not give full idea, because when the mRNA is produced, we are analyzing the mRNA. But in the next step, during translation, we need to know the proteomics, whether it is converted into a protein. So we have to identify the protein. So after that, with the protein or the enzymes produced, whether any metabolite is produced. So metabolism also is important. And QTL mapping is also important, because the trait related to stress resistance can be identified using the QTL mapping. And as Madam told, genomic selection is the game changer. So it will reduce the selection time. So this genomic selection actually based on a statistical model. So based on the physiological and marker data, we are training a model, and it will predict based on the genomic estimate breeding value. It will predict which population is better, or which seed. We can predict, select the seed, thing in the seedling stage itself. And as I told, apart from the single omics, multi-omics technology is important. And another booming area is the CRISPR-Cas. For CRISPR for activation of the gene, and CRISPR can be used for inactivation of the gene. And this is happening, as the first speaker told, using the agrobacterium. And prime editing and base editing is also done in the recent days, because in CRISPR-Cas9, we are making a double-stranded break and introducing the new sequence into that. But in case of base editing, if you know any mutation is responsible for the undesirable trait, we are changing the base. So base editing can be done. And in case of prime editing, what is doing is that we are associating a RNA polymerase enzyme along with the good sequence or the edited sequence, which should be created inside the system. So along with this, the guide RNA, the RNA polymerase will create the sequence which are correct in nature. So without breaking the DNA, this can be happen inside the cell. And another new advance is that ribonuclear protein based delivery. So apart from the agrobacterium mediated delivery of the CRISPR-Cas system, this is the transient delivery, and it will be having precise genome editing and offering quicker action because it is transient in nature. This ribonuclear protein will degrade faster, and the CRISPR, the sequence which we need to introduce usually will be there in the system. And as I told, this sequencing technology is from telomere to telomere. That is, the full-length sequencing will be helpful, and the high-fidelity sequencing and haplotype resolved genomes, and like the pangenomes. So pangenome is the full sequence not of an individual, it is of population. So from the population, there will be some core genome and the accessory genome. Mostly the accessory genome will contain the genes for the stress resistance or stress, any good traits. So another area is epigenomics. So epigenomics means there is no sequence variation, but there will be modification within the genome by the different methods like methylation or histone modification. Usually histone acetylation and deacetylation happens. And one advantage is that there is no sequence change is happening. The system genome is as such, but only the modification. This modification will help the plant to decide whether the gene should be expressed or not to expressed. So this will help to modify or manage the stress. So one advantage of this thing is that there will be transgenerational memory is there. So once these modifications happen, in some cases, it can carry over to the next generation. So this memory will help to manage the stress in the next generation also. So in case of this epigenomic modification, this microRNA, siRNA and long non-coding RNA have their own role in modification. So this miRNA will adjust the gene expression by targeting to the mRNAs, and these small interfering RNAs will detect the methylation and to the site-specific DNA sites, and the long non-coding RNA will organize and recruits the chromatin remodelers. And coming to the memory mechanism. So in some cases, it will be somatic memory. That is, means it will be exist only in that particular generation. And some cases, it is transgenerational. So usually histone modifications are short-term memories, but DNA methylation can have the possibility of long-term memory. And the modification by non-coding RNAs can be variable, either short or it can be sometimes transgenerational. And not only the genomic tools, another technology is arising is artificial intelligence. So we have to combine that artificial intelligence with the genomic tools for the most understanding of the genes. So machine learning models will be helping for the genomic prediction, and digital twin for forest health is important. That means it's a virtual replica of the forest land, because we are feeding the data of the forest into a system. And when we are feeding the genomic, climatic and physiological data of the present situation, it will predict what disease can be occur, or what is the impact of the drought. Like that, we can predict. So that will be helping us to predict or take a action before few other data here. And the big data pipelines for the genomic database is essential for this mapping and all. And today I am concentrating on the stress management based on using the defense hormone. So actually defense hormones are present in plant, but they are getting activated when the plant is exposed to a stress condition. So when the plant is exposed to stress, these hormones will activate and they start their function like stomatal closure, production of antioxidant production and metabolic adjustments. So the major key players are abscisic acid, salicylic acid, jasmonic acid, ethylene, auxin, cytokinin, gibberellins and brassinosteroids. These are the major players in stress response. So they are not acting alone. They have a crosstalk between the signals or the defense hormones. So there will be crosstalk between the abscisic acid ethylene pathway. So these are antagonistic in nature and they are during the germination period of the seeds. But in case of senescence and stress adaptation, they are cooperative in nature. And coming to jasmonic acid and salicylic acid, they will balance defense against the different pathogens and it will manage growth defense trade-offs. And coming to brassinosteroids and gibberellic acid, they act synergistically. So what they do is, it will degrade the DELA protein degradation. So DELA protein is a growth retarding protein. So it will degrade the DELA protein, it will promote the growth. And this abscisic acid, auxin will coordinate the root architecture and stomatal function under what stress condition. So during stress, it will enhance or produce more roots to get more water and it will cause the stomata to loss of water, reduce the loss of water. And this is one study we did in College of Forestry for evaluating the role of jasmonic acid and salicylic acid in alleviating the drought and salinity stress. So this study was carried out in mahogany seedlings. So the stresses given are drought and salinity. So 100 millimolar sodium chloride is given and drought is given when until the watering was stopped until the drought symptom appear. And after drought, exposed to drought, the plant is sprayed with salicylic acid and jasmonic acid. The foliar application is done and the observations were taken 8 hours, means 8 days after the application. And you can see that after the drought and after the application of this salicylic acid and jasmonic acid, the treated plants are recovered compared to the control. So both salicylic acid and jasmonic acid at specific concentration give the result for the adaptation, that is the acceleration of the stress management by the plant. So the control plant failed to regain. And these are the data for the number of leaves after the drought regain when these plants are treated with the salicylic acid and the jasmonic acid. So both cases, the treated plants will be having much more leaf production compared to the control. And these are the number of shoots produced and salicylic acid and jasmonic acid, and the two stress conditions give better result compared to the control. So apart from that, we did the proteomic analysis after the treatment of the defense hormone. This analysis are done after 24 hours of the application. And all these conditions were analyzed, normal condition, stress condition and regain of the drought and regain of the salinity stress at different concentrations of the salicylic acid and the jasmonic acid. So this is the SDS page gel analysis. You can see that the normal plant have a different pattern and the regained plant, salicylic acid and jasmonic acid treated plants have the different pattern of protein gel. So this is for salinity, the other was for the drought. And also we analyzed the fold change increase in the proteins after the treatment of the defense hormone. So we can see a considerable difference in the applied treated plants, both the salicylic acid and jasmonic acid compared to the regain and the normal plant. So these are the proteins identified for the drought condition. And for the salinity stress, these are the proteins identified and the fold change analysis was carried out. So from this study, we find out that there will be a unique expression profile for each stress response. So for each for stress, drought stress, the proteins expressing are different, and for salinity, the protein expressed is different, and during treatment, the protein will be responding in such a way that. So here we need the climate smart forestry genomics. So what we need to do is that genomics is the transforming forestry from slow breeding to rapid predictive and precision based management. So we have to use the multiomics and the artificial intelligence and this genomic editing tool to offer the unprecedented opportunities. And the integration of these things, genomic tools into forestry, must be in a systematic manner such that it should be linking labs, nurseries and field trials, and also policy framework is needed. Even though so much of technologies are there, it should be applicable in the field. So policy regulations are also important. So I would like to conclude that we have the tools, we know the science, and what we need is that we should have a collective will to do it. And let us work together with the forest that can survive the next century. So I'm not telling it will replace, but it will empower the forestry. And by moving from the sequences to the functional understanding and from understanding to application, we can design forests that are resilient, productive and sustainable. Thank you.
I guess we can conclude this session. That is quite heavy. 3 sessions describing how planned next generation technologies can actually transform the entire forestry sector. We started off with Dr. Matish, who talked about functional genomics, editing, gene silencing. I think he opened up a whole new world for us. I mean, we have been actually struggling with abiotic stress tolerance breeding, especially an area he has been elaborating on salt tolerance. I think in agriculture, we have absolutely no clue in conventional breeding. If you look into, say, crop like rice, almost all the stress-tolerant rice varieties have come from the Pokali area. If you look into wheat, everything in the world has come from Karnal. Look into the salt tolerance mechanism, we find a complex mechanisms moving from anatomy, morphology, physiology, biochemistry, everything. There are plants that actively take up salt, throw it away. There are plants that actively take up salt, keep it aside. There are plants that refuse to take up salt. There are plants that take up salt and degrade it. So complex mechanisms, and now we find that 𝐓𝐑𝐿 has been making some headway in this. I think about most of the things he mentioned, I think they are very, very promising, including transcraft, CRISPR-Cas9 technology in eucalyptus, RNA interference, gene silencing. I think we could not do justice to what he wanted to do in terms of time. I think we had to rush. Probably you can get in touch with Dr. Matish for more information on this. He has a lot more to say on this. Then Dr. Madhavida took us to the, I mean, she told us that it with 27 years of experience, 27 years back, she was shocked when she entered into this field from agriculture. I think we are still shocked. Shocked not by the problem, shocked by the opportunity that these tools are now giving us. Now the breeding time could be halved, more than halved. I think from 18 years to 9 years, from 27 years to 6 years. I think huge opportunity being presented by the genomic technologies, the omics that is developing. I think huge, and we have taken a full circle. We started off by when Lamarck said inherited characters cannot be, I mean, acquired characters cannot be inherited. Now we talk about epigenetics, where we say that it could be. Okay, Lamar could be wriggling in his grave. So we have taken a full circle. I think Dr. Madhumita took us through the entire gamut of technology that is available, and some of the technologies, you know, I mean, when we started off tree breeding, agriculture people are so lucky that they can come out with pedigreed population. They can talk about mating designs. You know, we can never imagine that, and we have the tools like breeding without breeding, wherein pedigrees can be constructed today. And I mean, those are things. I mean, we never imagined. We never imagined that we could work with a pedigreed population. Now we don't really need to develop a pedigreed population. We can walk into a forest and construct a pedigree and work on that. I think the opportunities that is provided are huge. I think Dr. Madhumita, again, to her also, I'm very sorry we couldn't do justice to what she has actually come prepared to deliver. And then we were introduced to a new area by Dr. Manju. She gave us an overview of the entire functional genomics and omics technology. And finally, she talked about one of her favorite, because I know that she's working in our lab, which is basically on stress management through defense hormone. Again, maybe one day we need not have to work through stress breeding. We could actually manage it through defense hormones and things like that. Probably that is also a huge area, because personally I am working on stress tolerance in teak, especially for drought tolerance, using most of these technologies. But then we have new tools in terms of defense hormones that again offer us a great opportunity to overcome these challenges. I think Dr. Manju concluded with a slide that aptly describes where we are going. She concludes saying that genomics will not replace traditional breeding, but it will empower it. I think that's a final take. I mean, these are going to make our world less complex, more manageable. Traditional forestry breeding will be empowered by all these tools. I thank all the speakers for their excellent presentation, and I'm sure that most of you would be having a lot more to discuss on these things. Please feel free to contact these people. They are very approachable people. Please feel free to contact them and clear your doubts, and thank you again.
With this, we are coming to the end of this session. On behalf of the organizers of Tropical Forestry Conclave, I thank all the eminent speakers of the session, and I also thank the coordinator, Dr. Professor Santosh Kumar, for very excellently conducting the session. So now I invite our director, Dr. Kannan C.S. Warrior, to present the mementos to the honorable speakers. I invite Dr. Madhumita Das Gupta, Chief Scientist at 𝐼𝐹𝐺TB Coimbatore, to accept our token of appreciation. Next, I invite Dr. Manju Elizabeth, Assistant Professor at College of Forestry, to accept the memento as a token of appreciation. Thank you, sir. Next, we have a very special lecture from a distinguished professor from University of Oxford, Professor 𝐓𝐑𝐿𝐵 Malhi, who will be joining us online. And he's giving us a lecture on energy flow view of the tropical forest. I invite Dr. Srijit K.A., Principal Scientist, KFRA, for introducing the speaker.
Good morning, all. Good morning, 𝐓𝐑𝐿𝐵𝐿. 𝐓𝐑𝐿𝐵𝐿 Malhi is a pioneering scientist working on challenges of understanding and restoring ecosystem across the globe. He is a professor of ecosystem science at the University of Oxford, and he was also the director of 𝐓𝐑𝐿𝐵𝐿 Center for Natural Recovery. He has worked on these topics for over 30 years, conducted challenging field research in some of the most remote regions of the world, ranging from Amazon to Siberia, and founding and nurturing global networks of tropical forest research. And he has extensively published in leading global journals with more than 600 publications and more than 1 lakh citations. And his studies are on ecology and biodiversity and various other spans such as remote sensing, machine learning, all these aspects he has been used. And he is a recipient of Marsh Award for Climate Change and the Patents Medals for the Royal Geographical Society, and most recently, the Ramon Magab Prize for Ecology 2025, one of the world's premium awards for ecologists. He is also a trustee of the Natural History Museum of London and a fellow of the Royal Society, and was appointed as CBE in 2020 for his services to ecosystem science. We are fortunate to host 𝐓𝐑𝐿𝐵𝐿, a global leader in tropical forestry research, the most appropriate person in such a global conclave. And thank you, 𝐓𝐑𝐿𝐵𝐿, for accepting our invitation. I know that you are so much busy with the British Ecological Society annual meeting. Still, you make it. We are really honored and grateful to you. Over to you, 𝐓𝐑𝐿𝐵𝐿.
Okay. Good morning. Can you hear me okay?
Yes, we can.
Okay, great. Okay. Thank you for inviting me, and congratulations to 𝐓𝐑𝐿𝐵𝐿 on your Golden Jubilee celebration. And it's a pleasure to come to be here virtually and to be with you and to congratulate you all on your achievement. And I was asked to talk about tropical forests for this tropical forestry conclave. And so what I am going to talk about is a relatively unusual view of tropical forests, which is through the context of energy flow. And I'll explain why energy flow is a useful way of thinking about the health of our tropical forests. And so energy is something that unites all of life. The capture of sunshine by plants and its conversion into organic matter, and the use of that energy by plants, and then the cascade of that energy through herbivores and detritivores into animals is what powers almost all of life on Earth. And so therefore it can be a unifying way of thinking about ecosystems that combines trees and animals and fungi and bacteria into a single viewpoint of the functioning of an ecosystem. And it has the study of energy flow has a long history in ecology. It starting back in the 1940s with some of the work by Lindemann and MacArthur, some of the founders of ecology, had this idea. And another significant person was Ramon Margalef, the Spanish ecologist, who talked about ecology as a study of organization of the biosphere, the patterns that emerge as energy and matter cycles through ecosystems. So it's a very old unifying concept in ecology, but one actually I think isn't used enough in our thinking about ecosystems, where we tend to narrow down to particular organisms such as trees if we're thinking about tropical forestry, or animals if we're thinking about various aspects of zoology. And we don't often integrate across plants and animals and microbes as a single perspective. And so in this talk, I'm going to apply this ecological energy lens to thinking about tropical forests. And why is energy so useful? Firstly, it's something real in an ecosystem. There's not a metric that we're making up to describe something. There is actual energy flow in an ecosystem. So it's physically a meaningful thing that's happening in a forest, just as much as biomass or other measurable quantities. And it can be considered, unlike I think that other metrics that tend to be concentrated on one taxa, it can be consistently applied across all taxa. So you could look at energy flow in birds or in mammals or in particular functional groups like elephants, and you can look at energy flow in trees. And the physics definition of energy is the ability to do work. So when you describe the amount of energy flowing through trees or through a particular functional group, such as herbivores or seed-dispersing birds, you're describing that ecosystem function, how effective that function is. So it can integrate across multiple ecosystem functions. And firstly, we can think about the energy flow of the biosphere, or the land biosphere in particular. And this figure shows you a figure that is based on data that's probably very familiar to you. It's satellite data of net primary productivity, gross primary productivity of the biosphere. Using satellites and field data combined, it gives an estimate of productivity. But unlike most conventional maps, what we've done here is produce a cartogram. We've expanded or contracted the size of each pixel according to the amount of photosynthesis going on in that pixel. And so areas that have high biological activity, high photosynthesis, are expanded in size, whereas areas with less activity are shrunk. And this is for the month of July, and it looks vaguely familiar, but you see the desert regions such as northern India, Sahara, the Middle East are shrunk away to low productivity, and as are some of the southern hemisphere, like Australia. If we do this for January, however, the picture looks very different. You can see that the northern hemisphere has shrunk away into deep, dark winters, which is where I am now in Edinburgh, in Scotland, and you can see that the tropics are dominating the biological activity of the biosphere. So just look at the size of India there compared to all of Europe, for example, biologically. And so this is looking at the Earth through biological eyes, and you can see how much the metabolism of tropical regions, such as Kerala, dominates the metabolism of the entire planet. And this is why tropical forestry, tropical ecology, I believe, is so important. It isn't some peripheral area of the ecosystem. It's actually a core of the metabolism of our planet. To understand and manage tropical regions is an important part of sustainability of our planet as a whole. And we can do this for every month of the year, this calculation, and you can see this relative pulsing of the amount of energy going through the tropics and the extratropical regions, and how I like to think of this as the great green heartbeat of the Earth being visualized, that this power of energy captured by plants and cascading through ecosystems is the core heartbeat of our living planet that maintains the webs of life that maintain our civilization and all the rest of life on Earth. And maintaining this heartbeat, keeping it alive, is an essential part of anybody concerned about the sustainability of our civilization and the sustainability of our planet. So in a lot of my work, as was mentioned in the introduction, I've been looking at tropical forests in particular around the world. And about 15 years ago, we started a network called GEM, the Global Ecosystems Monitoring Network, that tried to understand in detail the flows of carbon and nutrients, but you could also say the flows of energy in different tropical forests, in the vegetation, focusing only on the plants for this initial work. And we established a protocol and a manual called the GEM manual that we applied, that was freely available, and we encouraged others to apply it. And you can see we worked in many regions across the tropics in South America and Africa and in Southeast Asia. And and just to summarize some of the key components of energy flow in a forest, or carbon flow, you can think of carbon and energy as relatively equivalent. It's because the carbon flows in the form of carbohydrates, which capture energy, so represent captured energy. So that the numbers are different, but the ideas are very, very similar whether you talk about carbon or energy. So plants capture photosynthesis, and this is called the gross primary, they capture energy from the sun through photosynthesis, and this is called the gross primary productivity. Now, some of that energy is used in the metabolism of the plants themselves. This is called the autotrophic or plant respiration, and the rest of the energy is used to build biomass in the plants. The captured frozen energy is used to make leaves and flowers and fruit. It's used to make wood, often the main focus of forestry activity, or it's used to make roots, coarse roots, woody roots, or fine roots that capture nutrients and water. And that's called the net primary productivity. So the photosynthesis is sometimes called the gross primary productivity. Now, all of that biomass eventually ends up as dead biomass, either because when the leaves are shed from the tree after a few years, or when the roots die, or eventually when the tree dies and falls as dead wood and is decomposed by fungi and bacteria. Now, that dead material is consumed, as I said, mainly by microbes, and that is called the heterotrophic respiration. So there's this flow of carbon dioxide out of dead material and the burning up of that remaining energy in that material. Now, a small fraction of the biomass is captured directly by herbivores, whether it's insects or by mammals or other herbivores, and then a smaller fraction of that cascades up through the food web to predators, carnivores. And so the main focus of traditional ecosystem ecology has been the plant and microbe part of the cycle, but later on I'll also show you how energy flows are a way of bringing in those last bits, the herbivory and predation, into our understanding of the health of a tropical forest. And so this is another way of understanding this flow of carbon or energy in a forest. And at the top left, we have things, we have the gross primary productivity, the photosynthesis. And this is something that's often monitored by optical remote sensing, satellite data, or often in models. There's a lot of focus on understanding and predicting photosynthesis. And the bottom right, we have the standing biomass of a forest. This is often the focus of forestry in terms of tree diameters and structure. We can use forest plots to understand standing biomass, or we can use some of the newer technologies such as LiDAR remote sensing. And those two are the most common parts of this chain that are measured. It's much less common to measure the parts in between that connect. And so sometimes we can make the mistake of assuming there's a simple direct relationship between photosynthesis and biomass, or if you measure biomass, we're saying something about the photosynthesis of a forest. But in fact, there's a decision in a plant, there's a certain amount of that photosynthesis that goes into respiration. Of that net primary productivity, only a small fraction goes into wood, the rest goes into flowers and fruit and roots. And then the relationship between woody growth and biomass depends not only on the growth rate in a mature forest, but also on the mortality rate, the rate at which trees are dying, which can be thought of as a residence time. And so understanding all of those processes is important if you're trying to understand the link between energy capture and the biomass of a forest. And so in our protocol in GEM, we try to measure those components in between. We try and measure the growth rate of trees, often with high precision, with dendrometers, although you can do it with conventional forest censuses. We try and measure the canopy productivity by measuring litterfall, the rate of production of flowers and fruit and seeds and twigs. We try and measure below ground productivity by putting in in-growth cores where we have mesh bags with and without roots, and then we measure the rate at which roots grow into those bags and take out the bags every 3 months and weigh the new roots that have entered the soil. and measure root productivity. We try and measure the flow of carbon dioxide out of the soil through soil respiration. And we also try and divide that into soil with litter, without litter, with root mycorrhizae. And we do the same with stems. And with in campaign mode, we might try and do leaf respiration and photosynthesis at the canopy. And we couple this with climate data. So we have a good relationship between the carbon cycle and the climate. And this is an example of the types of diagrams that emerge once we do this work. It requires monthly measurements over a period, so it's quite a little bit of work. But now we've created diagrams like this for about 60 or 70 tropical forests around the world. And I won't go into detail in the numbers there, but I'll highlight one key thing, and that is in the bottom there, inside I've circled, that's the woody productivity, the rate of woody growth, which is often the focus of forestry measurements. You're looking at the rate at which trees are growing. And the top number is the total photosynthesis of this particular forest. It's very typical for many tropical forests. And you can see that the woody growth is just a few percent, less than 10% of the photosynthesis of a forest. Most of the energy captured by a forest, the carbon, is used for other things, for the canopy, for respiration, and other components. And so to understand the full carbon cycle of the forest, it's important to understand how, say, woody growth and production might be affected by climate change, or by rising atmospheric CO2, or other factors of change. And so, as I said, we've done this work in a number of sites right across the tropics. I'll give you some examples of some of those sites. In Ghana, in West Africa, we've applied this approach to a precipitation gradient going from wet rainforests through to dry rainforests, to dry forests and then to savanna. In Gabon, we've looked at the effects of elephants and large animals on forest dynamics and ecosystem cycling. Gabon is one of the few places in Africa where the forest elephants and gorillas and other large animals are still found at natural high densities. So they're shaping the forest and structuring the forest and seed dispersal in a way that's only possible in a few forests around the world, including some in India where this is in principle possible. In Peru, we've looked at the elevation gradient going from the lowland Amazon rainforest at 200 meters elevation all the way up to 3,500 meters elevation in the high Andean mountains. In Brazil, we've explored a logging gradient of intact forest, logged forest, and then bio-affected forest to understand how they affect ecosystem processes. We've done the same in Malaysia and Borneo, and I'll return to this site for later on in this talk, and looking at a logging gradient. And in Australia, we have a similar elevation gradient. So this gives you some examples of the types of sites where we're working. But what I've described thus far is something that I've talked about quite a lot, which is the flow of carbon and energy in vegetation in forests. And there's a lot to be said there. There's still a lot to be discovered, understanding the patterns. And I could easily spend the rest of this lecture just talking about some of the findings we found about vegetation. But what I'm going to do this time is actually take this work in a slightly different direction and talk about the other parts of an ecosystem that are often missing in our understanding of tropical forest functioning. That is the role of animals in those ecosystems. And when we think of the health of an ecosystem, it isn't just about the trees, it's about the mammals, the primates, the insects, the birds that make up a healthy or an unhealthy ecosystem. And energy flow can be one way of bringing animals into a more holistic understanding of ecosystems. So the question we can ask is, how does this sunshine spill out beyond the plants into the rest of the ecosystem? And so the key question practically we have to ask is, how do we calculate the energy use by animals? For some animals, like insects, we could do this directly by looking at how a fraction of leaves are consumed by insects. But for most of them, we have to use allometric approaches. And by allometric approaches, I mean, we look at the relationship between the mass of an animal and the energy requirements of that animal. So the larger an animal is, the more energy is required to maintain it, but it isn't a simple linear relationship. But we can use those relationships to say, if we know the mass of an animal, how much energy is required to feed that animal to maintain it. So if I take one example, this is a European badger, a 10-kilogram omnivore predator we find in the UK. From its body mass and the fact that it's a mammal, we can calculate its energy expenditure around 5,000 kilojoules per day. And then talking to experts, we can look at how its diet is partitioned between earthworms and arthropods and fruits, and calculate the daily food energy intake as this energy expenditure divided by the assimilation efficiency, which can vary between those different food items, but typically it's around 80%. And therefore you can calculate the amount of food energy required for each of those components. So how many earthworms need to be consumed on average, how many arthropods, and how many fruit. And then to go from the individual badgers to the population, we need to know, the main thing we need to know is the population density, how many badgers there are per hectare or per square kilometer. And then we need to know the body mass, and we need to know some basic behavioral characteristics. Is it there all year? Does it hibernate? Does it migrate? And we need to know the assimilation efficiency of the food. But the main information we need to get from field data is the population density of those animals. So that's data that can often be difficult to find in the data set. So I'm going to show an example of the application of this to the tropical forest system, which are the tropical forests of Southeast Asia, of Borneo, of Sabah in Borneo. And I'm going to compare old growth forests there with logged forests, which have been repeatedly logged for timber, often in a very non-manageable, non-sustainable way. And these forests are often called degraded forests, and in some contexts they are degraded in terms of biomass, they and carbon stocks. They've certainly lost a biomass. But I want to question this idea of degraded by looking at the ecological health. And degraded can be a mixed term. So sometimes a forest, even a well-managed logged forest, can be called degraded. And if the word degraded is used as a way of conserving or restoring those forests, it can be a useful concept. But sometimes it means that the forest is of lesser value, it's ruined. So it's not good for conservation, and it's okay for conversion into agriculture or to other land uses. So degraded can also be a dangerous term if it gives a lower value to a system. And what ecological analysis can do is ecologically, how degraded are these ecosystems? Should we think of them as lower value systems than old growth forests? And in terms of biomass, they are certainly degraded. So these are LiDAR images, airborne laser images of these two forests in cross-section. On the left, you can see these old growth forests in Sabah, magnificent, amongst the tallest tropical forests on Earth, 60-meter tall canopy height on average. And on the right, you've got this heavily logged forest. You see you've lost most of the biomass. It's less than 20 meters tall, what's left, and there's more of that concentration of the biomass near the ground. So in terms of carbon stocks or biomass, they are certainly degraded. But how are they in terms of ecological function? So we applied the GEM protocol that I described earlier on and measured the productivity of these forests. And what you see is that productivity is maintained in the logged forest. In fact, it's slightly higher than the old growth forest as there's more light gaps and the trees are trying to capture that light. But it's changed in allocation. There's much more growth in wood biomass, less in the canopy in the logged forest, recovering from disturbance, than in the old growth forest. But once you go to the oil palm, you see very different. You have lots of oil palm fruit canopy productivity, but the rest of the productivity is greatly reduced. So productivity is more or less maintained, or perhaps goes up. So how much of this energy ends up going up through key vertebrates, such as birds and mammals? And so what we were able to do at this site is measure the total populations of all the birds and all the mammals. This was an interdisciplinary project with lots of specialists. And so we were able to measure the population densities of the birds and mammals. And before we did this work, my expectation was that we'd show that the energy flow through birds and mammals was maintained in the logged forest and then drops off in the oil palm on the right. But actually our surprise was that we found 2.5 times as much energy flowing through birds in the and mammals in the logged forest than in the old growth. This means the population densities of most species were 2.5 times higher. And it was actually similar species to that found in the rainforest. It wasn't generalist and new species coming through. And so this was quite surprising. But is this just a few generalist species taking over? If you just find one or two species taking over, even though there's more energy flow, it doesn't mean that it's necessarily healthier. And so what we did is also look at the diversity of species. So the size of the circles in this diagram are the amount of energy flowing through that particular group, like insectivorous birds, for example, in the center. And you can see most of the circles are larger in the center than in the left, so there's more energy flowing through. And the other numbers that you see, the S is the number of species in that group, and E is a metric that we made up called the energetic Shannon-Wiener index, which is a measure of the spread of energy across different species in that group. So the larger the number, the more the energy is spread across multiple species. And you could argue that maybe that's more of a sign of resilience, because if one particular species declines, other species are performing that function. And we see those E numbers are also maintained in the center. And so therefore, there seemed to be more energy flow, but maintenance of energy, the spread of species. So why do these species, the logged forests, have more energy flow? It's mainly linked to plant traits. So the plants become more prioritized rapid growth after disturbance. They reduce the amount of chemical defense in their leaves, and there's more fruiting as well. So there's more food available for herbivores, and if there are insects also eating the leaves, there's more food available for insectivores, and so therefore the energy flow cascades through the ecosystem. There's also more vegetation near the ground, where large animals like deer and elephants can eat the food. So the forest becomes more salad-like in some way. And so when you think of a forest like this, you should also think of this as a site of intense chemical warfare, where most trees have had a long history of chemical evolution to battle insect herbivory, and so have very advanced chemical defenses that limit the amount of herbivory. This is why this forest is green and not consumed by insect herbivores. And logging or disturbing shifts that balance a little bit, makes the forest a bit more palatable, that increases food supply to animals. And so what I'm doing here, I wouldn't argue that this is a reason to log protected areas or old growth forests. We need to value them for many, many reasons. But this does challenge this assumption that modified forests, human modified forests, are ecologically degraded in a way. They often can be as ecologically vibrant as old growth forests. And so one other thing in this diagram, as I said, we were talking about the flow of energy multiplied by the spread of energy. And since this work, I've been thinking whether I use the term ecological vibrancy informally as a metaphor in this work. And I was wondering whether that actually is a useful idea that could be formally defined as a metric that focuses on the vitality of ecosystems. So this is the one slide with a bit of mathematics, where we are now trying to formally define this the spread of energy in an ecosystem. So if you look at any group in an ecosystem, it could be all the birds and mammals, it could be all the animals, or it could be a particular group like herbivores. You can describe the total metabolic power as a sum of the energy flow through individual species. So by summing up over all the different species in the energy flow, like I did in Borneo, I've got the total energy flow, the metabolic power. You can also describe the spread of energy across different species, and this is where we use some physics and some ideas from entropy theory, and this equation described as a way of describing the spread of energy. And then we can define vibrancy as this ecological power in a particular group, whether it's birds or mammals, and the spread of energy in that group. And this is called the vibrancy of an ecosystem. And here we see an example of this data. So this is the Malaysian data again, and here in this lens of vibrancy. So you see at the top, the figure that I've already shown, the higher spread of energy in the logged forest, both in birds and in mammals. And then in the second one, I described this dispersion, this energetic Shannon-Wiener index. You can see it does go down a little bit in mammals in particular in the logged forest, And then the vibrancy is the multiplication of the top two graphs, the energy power times the spread. And you can see this higher vibrancy in the logged forest before it goes down again in the oil palm plantation. So it's another way of showing those same results and proposing this metric of vibrancy. And we can apply this metric to other human-modified systems. So this is a shifting cultivation system. And this is a smallholder farm in Nigeria. And so it's a way of understanding how human-modified systems, whether through logging or through other traditional indigenous management systems, can often enhance the ecological vibrancy of forests. And it's a way of looking at those processes in a more integrated way overall. So this is certainly an open area of research of how human activity can often build the ecological health of ecosystems. And it also can analyze systems that are diminished in vibrancy. As an example can be a monoculture plantation, which may be absorbing carbon, maybe producing timber for wood supply that we have to think about, or it could be absorbing carbon for climate change mitigation. But in terms of ecological vibrancy, this is not a vibrant system. It's all the energy of life channeled through a single species, none of those rich food webs that I described. In many ways, you could argue this is ecologically, this is not a forest, this is simply a tree crop. And so you can describe those differences in vibrancy that helps us distinguish between different types of human activity. So we can also compare different across ecosystems. So in some more recent work, one of my students, Indian students, Bhavya Palgudi, has been looking at the biogeography of different forests, looking at Borneo and comparing that with a tropical forest in Peru, in the Amazon. And these are results from her work recently. And so what we've done here is compare the Borneo old growth forest that you saw earlier with an Amazonian forest in Peru, both of them very faunally intact, very little hunting pressure on the forest. And you can see some interesting differences. The bottom left chart in the middle here shows the amount of energy flow through birds in Borneo and in Amazonia, and you see much more energy flow in Borneo through the birds. And the right, we see the energy flow through the mammals, and there's more flow in the Amazon than in the Borneo site. And so what's going on here? Well, this is where biogeography gets very interesting. The Asian forests in Borneo are dominated by dipterocarp trees that are wind dispersed and they don't produce fleshy fruit. Whereas the Amazonian forest has many more animal dispersed seeds that produce a lot more fruit and food supply for primates and other tree-dwelling mammals. And also when the fruits fall, you have pigs and peccaries eating them as well. So the biogeography of the trees ends up shaping the energy flow through different parts of the ecosystem. And so we can do this similar analysis across these sites. On the left, I've got the Borneo site and the Peru site. In the middle, I also include a British woodland just outside of Oxford called Whiteton Woods, where a lot of ecological work's been done. And on the further right, I included a tundra in the Arctic, just as a contrast, a non-forest ecosystem right near the North Pole, where we've also done similar work on birds and mammals. And on the top, we see the number of species. We see much higher diversity of species in the Amazon than in Southeast Asia, but both the tropical forests are much richer than the British woodland and the Arctic tundra. And then we've got the energy flow, and we see, despite the differences in birds and mammals, the overall energy flow through birds and mammals together is very similar in Borneo and in Peru. It gets less in the UK, but it's still quite high and dominated by mammals, in particular deer are there as a large component. It's interesting to speculate why a temperate forest has so much energy flow compared to productivity. And then you have the tundra on the right. And so what we see is quite high but similar energy flow in both Borneo and Peru, slightly reduced in the temperate, but much more than we expect. And I think the seasonality of the temperate forest means that you often get an understory that's relatively palatable that large animals can eat, which you can't do in an evergreen tropical forest where it's always dark and shady in the forest. And we see lower energy in Greenland. And so then we can do these calculations of energy dispersion and the kind of the metric I showed before, and you see higher values in Borneo and Peru and a gradual decline in the UK. And then we can calculate this ecological vibrancy, which is the multiplication of the energy spread and the energy flow. And you see the total vibrancy is highest despite lower species diversity, the vibrancy in Borneo and that in Peru is very similar, maybe slightly higher in Borneo even. And so there's a difference in species richness and ecological vibrancy, and it diminishes gradually as you go to the other ecosystems. And so in our recent work, we've also gone beyond individual sites to start applying this to the scale of whole ecosystems. And this is work that was published just in Nature the last few months. So one thing we do is then take a metric that's quite widely established, the Biodiversity Intactness Index, which is available globally as a product, which looks at the relative decline in animal populations relative to the baseline that's found in protected areas. And so our colleagues in Africa have produced this Biodiversity Intactness Index for sub-Saharan Africa that you can see in this diagram. Areas of green have relatively intact animal populations, and then areas in orange are where the animal populations are greatly diminished. You can see West Africa, bits of East Africa with greatly diminished populations. And then we used a similar approach that I just described at individual sites, but this time we applied it to the whole continent of Africa, and you could see how the energy flow through birds and mammals historically was much higher, and at the bottom you see how that's been diminished by land use change and by hunting pressure. And you could talk about the ecological power of West African wildlife having diminished by a third compared to its baseline values. And we can do it for particular ecosystems, we can do it for forests. So this diagram, the outer circle shows the original energy flow through that group, and the inner circle is the current energy flow. So you see at the bottom left, for example, large terrestrial herbivores have been greatly diminished. You see a big reduction in energy flow through that group, whereas other groups such as the primates, the frugivores, are less affected, and the smaller animals, the rodents, are even less affected. They're very relatively resilient. And you can see how the energy flow has changed over time in the forests. And if you compare this to our Borneo forest, you see a similar result to what I showed with Amazonia, that there's more fruit available in African forests than there is in Bornean forests, fleshy fruit, and therefore you support more primates. So there's a difference between these two forests. And then we can look at how much energy flow is going through different functional groups. So grazing or browsing animals, or nutrient dispersers or seed dispersers. And the light column there is the original energy flow, and the black column is the new reduced energy flow. And then you can see on the right, the proportion of the energy flow that's changed. And you can see some functions like grazing and browsing or nutrient dispersal are greatly reduced because they're dependent particularly on large animals that are most affected by human activity, whereas other functions are less affected because they're dependent more on smaller animals such as soil formation that provide have been less affected by human activity. This is a way of combining our understanding of multiple animal functions, look at how those functions are changing over time. So I'll just conclude by a figure that tries to place where this idea of vibrancy of energy flow fits into our understanding of the future of tropical forests. So this is a figure that I produced over 10 years ago for a review on the future of tropical forests that I still like to use and to motivate my students as well. So on the left, we have the tropical forests of the past, where we've got not only intact vegetation, but we've got rich animal populations, we've got all of these complex ecological interactions. And those tropical forests have been increasingly under pressure for multiple reasons. One is around local land use and forest use associated drivers. And secondly, there are global changes, climate change, and other factors. And you could think of those as two belts that are tightening this image, which are in red. And this could be a one-way path to decline, but I think there are reasons to believe that this is a particular pressure that these forests are facing in the early 21st century, that over this century, we might expect some of those pressures to reduce as forests, as land use pressures decline as far as human populations stabilize, and also macroeconomic pressures change. So we're seeing this already in many forests in Europe and North America, now in China, where the pressure on forest land is diminishing over time as populations urbanize and stabilize and the economy shifts into other sectors. But what comes out of this bottleneck can either be very vibrant systems full of ecological function. All these systems will be different from the ecosystems of the past because of climate change, because of invasive species and all these factors. But they could still be very ecologically functional systems, or they could be greatly diminished systems. And what determines that is often how tight this bottleneck is, which is why I have those blue hooks there as determining how our actions that we can do to make that bottleneck as wide as possible to maximize the chance of these vibrant ecosystems. And the vibrancy metric that I was proposing earlier in this talk is one way of measuring the ecological integrity of these new forests that are emerging in the 21st century overall, affected by human activity, but in many cases still rich and vibrant forests. So thank you. So I'll stop sharing there. Okay. I can't hear anything if someone's talking to me. Okay.
Thank you, 𝐓𝐑𝐿𝐻𝐑𝐿. It's a wonderful talk, and it's a new perspective, actually. I have a question before that. Anybody?
So, Professor 𝐓𝐑𝐿𝐻𝐑𝐿, can you point out any relevance for the so-called old, natural, untouched forest over logged ones or the degraded ones? I strongly disagree with the concept of sampling and extrapolation for such conclusions, particularly on ecological functions you are pointing out. And my argument is very simple. So-called similar or so-called global studies that such high institutions are doing are indirectly supporting logging. For example, in the case of degraded or landscapes which are under restoration are heavily affected by invasive species. Such aspects are not well, we also have points like birds and other communities are adapting to such invasives, but these hypotheses which you are placing can directly impact on the untouched natural reserves that we are having. Thank you, sir.
Thank you for that question and that challenge. And I think you're very right. There are, and I was very worried when that paper came out that it would be used as an excuse for logging companies to say, oh, well, the professors in Oxford are saying that logging is healthy for forests, so therefore it's fine to log them. And that was definitely very, very clear. That's not our message. There are lots of important values in having forests that are untouched, and not only in terms of biomass and carbon stocks, but also particular species that are very specialist and that forest the resilience of those forests. Our focus more was on the lower value that logged forests have, given that some logging is happening because the timber is required, or whether it's in plantations or secondary forests, or whether it's logging of old growth of more intact forests. Given those, often those logged forests were being assigned lower value ecologically, and that would be fine if those forests were being restored. But often then, when we were seeing many companies and others talking about those forests, they were saying, well, now they're degraded, so therefore they're suitable for conversion to agriculture. They're already lost forests. And so therefore, there was a danger that by focusing only on protecting the highly intact forests and dismissing all the logged forests, that those logged forests were being further converted into agriculture, and you were losing a lot of ecological function in the landscape. Because ultimately, to protect forests, we have to think not only about the protected areas, we have to think about the wider landscape outside the protected areas and what their ecological functioning is. So that's the key message that we want to get from this, not an excuse to further log protected areas overall. And it's an important message to highlight. In terms of extrapolating from single studies, I fully agree. We need to do more of these studies. As you see, we're beginning to start doing these in Amazonia. I think it'd be magnificent to see similar work happening in India, maybe with 𝐓𝐑𝐿 or others, to try and look at similar questions. But so I don't disagree with your points there. I think we need to protect what intact ecosystems we have, but we also need to pay attention to the modified landscapes that we have and to ensure that they are also valued for their ecological services. And even sometimes non-native species can be a part of that story. That sometimes native species are damaging, undoubtedly, but many non-native species are not damaging and are often labeled together with invasives as all a problem. Whereas actually you should look at the ecological functions and to then decide whether they're a good or a bad thing for a forest.
Thank you. Any further question? I have one query. What about the grasslands, the energy dynamics and productivity comparing to this log forest and primary forests?
Yeah, so grasslands is something which also this vibrancy approach can be very useful in. And the nice thing is it doesn't focus so much on biomass. We're obviously forests have more biomass, but on ecological dynamics. So we're doing some work in South Africa and in Kenya where we're trying to describe the similar process of productivity and the flow of energy and finding that often the grasslands are as vibrant, as full of ecological function as forests. And this is a way of understanding and valuing their ecological function. And in many cases, what we find in many grasslands, you're facing this challenge of woody encroachment where higher carbon dioxide in the atmosphere or the loss of fire management is causing tree cover to expand in the grasslands. And that can often diminish the ecological vitality. It provides less food for large herbivores, and that cascades through an ecosystem. So again, energy flow is a way of understanding the ecological value of grasslands, rather than perhaps a rather traditional forestry view has been to see grasslands as degraded forests or diminished forests. That ideally, the ideal state of nature is to have a forest, and then somehow grasslands are of lesser value. So this is a way that you can show that grasslands are of equal value to many forest ecosystems.
Thank you. Any question?
Thank you, Dr. Srijit and Professor 𝐓𝐑𝐿. Now that I invite Dr. N. Krishna Kumar, former PCCF and head of Forest Force, Tamil Nadu Forest Department, former Director, Institute of Forest Genetics and Tree Breeding, Coimbatore, and former Chairman, State Environment Impact Assessment Authority, Tamil Nadu, for the concluding remarks.
Professor 𝐓𝐑𝐿 Malhi, thank you, sir, for being with us. In fact, a whole lot of us enjoyed your talk. In fact, I'm a forester with 40 years of experience from the Forestry Service in India. We've been dabbling with all sort of activities as far as forest is concerned, trying to look at various ways of management, trying to handle plantation forests, natural forests, wildlife, today even zoo management, etc. All that complexities. In fact, I'm really very, very thankful, and on behalf of the house here, would like to express our sincere thanks to you. And in spite of, I understand you're very busy this morning, yet you took some time off to come and give this insightful talk. And we appreciate your valuable time. Your presentation was wonderful. There was so much of clarity in energy flow. Energy flow is something that we all as students, trying to understand food web, food cycle, and energy flow in the schools and colleges. And when you look at it in the forestry dynamics, it's different. I think the catchword today is ecological vibrancy, what you said. Because today, increasingly, when it comes to restoration, biodiversity, when it comes to climate change, etc., we have to convince a lot of people outside the area of forestry, like the planners, the politicians, or the so-called, the public. When it comes to convincing all of them, we probably need to take new ideas across to sell our efforts of conservation. Tropical forest ecosystems, the last 3 days we were talking, doctor, and it was nice that you could come in and give us the importance. Of course, we have a small little doubts on what you told about the old forests and then the energy flow in different ecosystems, but yet I think these are concepts that need to be taken, because particularly I, for one, feel that energy pathway understanding in various ecosystems becomes important. It could be model for us to talk to people who matter and then try to convince them, when you just talk as to why we need conservation forestry, why we need to restore our forestry, why we need funds to protect our forests. Because as has been discussed the last 3 days, investments into forestry sector and continuous investments become very important. So therefore, on a platform like this, what we try to do is how best we can take good ideas forth across to people and then market our ideas for consistent fund flow. I think your talk was excellent. We all enjoyed it, and there is so much that we could, because I think you talked on gross primary productivity, energy transfers at various trophic levels, energy flow models you were talking of. You gave us various, insights in Borneo and other countries, trying to draw parallels from Africa and other countries that you work with. I also understand you are having a collaboration here at KFRA with Dr. Srijit. You had also visited our laboratory. In fact, if you were here, I think there could have been much more questions to you. Probably for want of time, students are restricting, but anyway, your presence even in the forum through this platform becomes very encouraging. Thank you so much for your valuable time. We look forward to more sort of collaboration, understanding, and gains from you. Thank you very much, Doctor.
Thank you. It's been a true pleasure to talk.
Yeah.
Thank you, Krishna Kumar sir, for the very excellent concluding remarks. So with this, we are coming to the end of the forenoon session. I hope the young aspirants, the students from various colleges are really enlightened and motivated from the lectures in the forenoon session. So we will break for lunch and we will assemble back by 2:15. Yeah, thank you all. Thank you all the speakers. Thank you for the patient listening. Yeah. The lunch is organized
by University of Fisheries and Ocean Studies, Kochi. And Dr. Annie Elias, she'll be joining online, and she is a Ramalingam Fellow, postdoctoral fellow at 𝐓𝐑𝐿𝐷 Coimbatore. And she's also founder of 𝐓𝐑𝐿𝐷 Analytics. And we have Dr. Magesh NS from CWRDM, Center for Water Resource Management, as moderator. And talking about Dr. Magesh, he's a multidisciplinary expertise in hydrogeochemistry, geospatial analysis, and environmental contamination, which integrating remote sensing and GIS data is working on environmental monitoring. He has significant field experience in polar science, particularly in Antarctic systems, and serves as an editorial board member and reviewer of many national and international journals. And he has been recognized among the top 2 scientists of the world scientists by Stafford University and elsewhere for the last 3 consecutive years. Now I request Dr. Mahesh to conduct the session. Dr. Mahesh, please.
So a very good afternoon. So we'll start this session 3, that is digital forest, artificial intelligence, machine learning, Internet of Things, and remote sensing applications for forest monitoring and management. So this topic is more or less related to computer applications, managing big data at global levels, as well as the data which is used for managing forest in a more precise way. So for this, we have three eminent speakers, that is Sri Shafiq PM, Dr. Girish Gopinath, and Dr. Annie Elias. So let me introduce our first speaker, that is Sri Shafiq PM. He's an embedded systems and IoT professional with over 13 years of experience in embedded hardware development, IoT solution design, and system integration. Currently serving as a manager, projects and technical coordinator at 𝐓𝐑𝐿, he leads IoT drone and software teams, drives LoRaWAN network implementations, and contributes to open source hardware and open IoT initiatives. His experience spans embedded platforms, wireless communication technologies, and end-to-end IoT architectures, complemented by strong project management and team leadership skills. He's also actively engaged in technical training workshops, mentoring, and speaker engagements. So let me introduce Mr. Shafiq PM. The stage is yours, sir.
Yeah, good afternoon, all. First of all, I would like to express my sincere gratitude to KFRA officials to invite as a speaker role in this session. My name is Shafiq. I am in Manager Projects and Technical Coordinator in 𝐼CFOSS. 𝐼CFOSS is an international center for free and open source solutions under Department of Electronics and IT, Government of Kerala, situated in Trivandrum, in Karivattam. Today, I will deliver one. Actually, I will deliver 2 or 3 use cases. Actually, we have deployed some applications across Kerala, outside Kerala also. So I will deliver that. What are the contents we have engaged it. These are the items I will handle by today about 𝐼CFOSS and basics of IoT and LoRaWAN and its role, LoRaWAN applications and microclimate monitoring, 𝐼CFOSS activities and live demonstration. Actually, we didn't attempt or deployed in forestry applications till now, but related to that, some of applications we have deployed in across Kerala. So I will explain it later. So starting with 𝐼CFOSS, I already told that 𝐼CFOSS is under Government of Kerala, and which is started on 2010 for promoting open source software promotions across Kerala, mainly across all departments. And we delivered all the department, force related activities in all departments across Kerala. And also we handled the training sessions and supporting sessions for the departments. And from 2017, we thinking about the hardware also. We need hardware development in open source area. So we have started a Center for Development of Open Hardware in our 𝐼CFOSS for promoting the hardware solutions. And we convert our 𝐼CFOSS name to International Center for Free and Open Source Solutions. Till that, 2020, the name is International Center for Free and Open Source Software, and we converted into solutions because we handled software and hardware related activities in a single umbrella. Mainly the open source related activities we have handled, open source tools, open source related things we have handled. And we have a nodal agency to promoting open source technologies in Kerala IT, and a total solution provider, and JNU affiliated for some courses we have offering that. So these are the core team, which is CDOH. Under this, open hardware team, open drone, open IoT, and some GIS and ERP teams are there. And these are assistive technology, language technology. These are the another teams working in the 𝐼CFOSS team. So coming to our title, which is IoT. Have you heard about the IoT? Have you heard about I think all are here in the IoT. What is IoT? Yeah, that is the Internet of Things, which means, which is a collection of sensor network or a physical. We need to monitor a physical parameter in our cloud platform. So one example I will tell you. These are the chairs. So I need to know which chair is vacant or occupied. So we can integrate sensors in these chairs and get the data from that things. So the sensor will give the exact information of the chair status, and we can publish it or we can analyze it. The chair is vacant or occupied, something like. This is the one example, typical example for that. So anyway, all things are connected to the internet or intranet part. So that is called actually Internet of Things. Any things, any devices, any human non-living things or any type of sensors we can integrate to the corresponding server side. We can collect the data from the field. That is called actually IoT. That may be a sensor network thing, or a single sensor we can integrate it, or multiple sensors we can deploy it in a field. All things are under this category that is called the Internet of Things. I hope you clear. And this is the IoT ecosystem. If we need to develop an IoT ecosystem in our own, so these are the parts, which is there is an edge node. Edge node means which collect the data from the field. That one example is temperature sensor. Temperature sensor which collect the data, field temperature data, and that transmit to the corresponding wireless network. Wireless or wired network we can use. So the second part is the communication network. Actually, first is the edge device, and second one is the communication layer, and third one is the database or cloud server platforms for for the analysis part. And we can visualize the data to the concerned user clients, something like that. Okay, this is the IoT architecture part. So we have multiple applications, means several applications are there. I will explain it once. I will explain it later because I need to convey what are the communication technologies in IoT. So have you heard about the Wi-Fi, Bluetooth? Okay, these are the communication technologies we have listed here. There are several communication technologies are there, which is BLE, NFC, RFID, or cellular connectivities. There are 3G, 4G, 5G, and one is LoRaWAN also there. LoRaWAN is the long range wireless area network, which is, I will coming to that point because I need to convey why LoRaWAN using for IoT communications in this time. Actually, we can develop a temperature sensor using communication technology as a Bluetooth, and through Bluetooth we can collect the data to our mobile platform. That is possible, but that is limited to some range. That maybe Bluetooth can have a 3 or 5 meters, up to 10 meters range only for that. So we can do it in a single room or nearby receiver stations only. So we can't use the Bluetooth as communication technology in a forestry or large area based applications. Like agriculture farm is a large area within 2 to 3 acres or in hectares area covered. So we need a stable long range based communication technology we required. We can use the cellular networks. Cellular network, we have a cellular network available here. 3G, 4G networks are available here. So we can utilize that also. But the power requirement will be the more, because The cellular network depends on the cellular service providers. We need to pay them. That one is there. And the RF controllers are power hungry. So we need to take care of the battery backup also. So coming to this LoRaWAN, there is one main advantage is low power, long range communication technology. That is also a wireless area network related communication technology also. So IC4 is mainly utilizing the LoRaWAN network because of another criteria also we have, because the LoRaWAN is similar to, means aligned with the open source technology, because the LoRaWAN utilizing the spectrum 865 to 867 megahertz, which is open. We didn't pay for the spectrum bands. Cellular networks using some spectrum, that is not a free band. That is some vendors will be paid for the spectrum utilization. So that will be a chargeable basis. But in LoRaWAN in India, 865 to 867 megahertz is a free band. So this is one criteria, and we have server systems in open source platforms like 𝐓𝐑𝐿, Grafana. These are the server platforms and the visualization tools available for the LoRaWAN communication technologies. So in these requirements satisfied with aligned with the open source technologies, we use the LoRaWAN as the communication technology in IC4 applications, IC4 deployment applications. We did several applications in LoRaWAN. So these are the reasons to select the LoRaWAN as a IoT communication technology in this type. Okay, and this is the LoRaWAN architecture right now. This is the edge devices for smoke alarm, water meter, trash counter, vending machines, and temperature, humidity sensors, automatic weather stations, some metering solutions like energy meters, water meters. All things are we can integrate to the LoRaWAN communication technology, and these edge devices transmit the data to that wireless spectrum. Just chirp the data to the spectrum, and the nearby stations will collect the data, like that's a concentrator or gateway. This concentrator gateway collect the data and send it to the network server. Because the concentrator gateway have internet provision, or we can handle it in intranet provision, because we can develop a intranet based LoRaWAN network. So the gateways are connected to the server platform through the intranet. Like we can utilize the K-phone network as well in this backhaul. This mentioned the 3G Ethernet backhaul there. We can utilize the K-phone fiber network backhaul also. So we can prepare a private government-owned LoRaWAN network for the government of Kerala. This is the aim for IC4 to promoting the LoRaWAN network ecosystem in Kerala. So this is the main LoRaWAN architecture. Coming to IC4's LoRaWAN ecosystem, we have different activities. LoRaWAN gateway. We have LoRaWAN gateway devices. We have ultra low power edge nodes and LoRaWAN relay nodes. These are the main things we need to develop a LoRaWAN ecosystem in this area. So I will go into the this is the implementation model. Like we will deploy one gateway in the up terrain, in a tower or highest position, and we can collect the data from the 2 to 5 kilometers range, something similar to the mobile tower. That is the case. Okay, this is the deployment pattern of the or implementation plan of the model of the LoRaWAN network in field. So then coming to the application side, there are multiple applications. I have already told you several applications are there in smart homes, smart cities, and smart villages. We focused aligned with the forestry applications right now. So I already told you we didn't use the forestry applications till now, but we have aligned some applications. I will demonstrate some of the applications right now, which is LoRaWAN microclimate stations. We deployed LoRaWAN-based microclimate monitoring stations, which is integrated with temperature, humidity sensors, pressure sensors, solar radiation, wind speed, wind direction, rain gauge, soil moisture. These contents are in automatic weather stations were handled. And we deployed 14 ARG stations in across Kerala, which was in 2022, and that funded by Government of Kerala. And that was the data was handled to the KSDMA. So Kerala State Disaster Management Authority need ultraviolet sensors data for them. So they provided us the UV sensors, and we integrated these sensors to the existing ARG network and collect the data and to provide it to the KSDMA as well. So I will show the data pattern. This is the data, live data from the Kerala IoT-based microclimate monitoring systems. This is one exact microclimate monitoring station data pattern. So we can utilize these type of microclimate monitoring stations in our forestry applications also. Right now we utilize it in Kerala land. We implemented in one of the stations nearby in Pananjeri Panchayat office, and one in each district we have deployed this type of automatic weather stations in across Kerala. And one of we can take one data. These are the detailed version: temperature, humidity, and UV sensors. Like right now, this is 3. This is in Munnar. This is a live data coming from, and last 24 hours you can select the 30 minutes or 3 hours data, and there is no rain. And you can select 90 days data. There is a huge data is available. In last 2000 from 2023 April, we deployed these instruments in the field, and you can get the data from that 2023 onwards. So for the study purpose, this is an open platform, so you can utilize this data for your studies. These are the this is visualize.openiot.in. You can note down it, and you can check the data in your needs. Okay, this is one application. Then coming to next. And these are the implemented microclimate monitoring stations. 14 stations for KSDMA, 23 stations for LSGD name and block. We deployed 23 stations, 23 ARG stations. ARG means automatic rain gauge stations. And 8 stations for Gram Vikas Odisha, which is outside Kerala, which is in Odisha, for their village monitoring application purpose. And they utilize the data to predict the localized weather predictions. That also possible, because right now the predictions are making some Google data and satellite informations only. If we put the local data into that model, then the prediction results will be more than 80 or 90%, because the current data is 70% accuracy. Then we can improve the accuracy from if we put the data, local data, into the same model. That is the main advantage. And the IoT enabled name and block, I will show this data also. This is one name. We deployed the actually IoT enabled name. I will tell you one history. In 2020, we have commissioned IoT enabled Grama Panchayat at Katakada, Trivandrum district. So that is the one first in India. Actually, that interested to the Nema Block Panchayat. So Nema Block Panchayat have 7 to 8 panchayat internally. So they funded us to implement all the panchayat in a IoT level. So we have deployed 15 gateways. I will show we have deployed 15 gateways for the entire panchayat, which is around 140 square kilometer area. This is the panchayat area. Means Nema Block Panchayat area. It contains 7 to 8 panchayats. So we deployed 15 gateways for the coverage of the LoRaWAN network, and 60 more devices for collecting the data, like rainfall, temperature, humidity, pressure, wind speed, and wind direction, and all the data are. I think this is in. Yeah, these are the parameters we have, temperature, humidity. If you click the temperature, then you can see the temperature information in the map. This is the geospatial based application, and this is the humidity. Humidity, we can collect the data, and we can show these details, and the rainfall also there. And the last 24 hours data also shown here, because some errors are loading data. Yeah, these are the last 24 hours data here. You can see the data. And this is the color indicators we provided. That is not exact because we are not a domain expert like for this part. Actually, we are a technical implementation team only. So this is one, and the air quality devices are there. IAQ means Indoor Air Quality Index. We have put some air quality devices here and collect the data from there. CO2, water level data is also there. In Nema Block Panchayat, we put air quality devices, water level and water quality devices, water meters, energy meters, automatic weather stations, automatic rain gauge stations, temperature, humidity stations. All the things are deployed for the Nema Block Panchayat for their requirement, as per their requirement. So in these, all the data handling to the same LoRaWAN network and in a single dashboard. That is actually, you see this. Okay. So this type of applications we can utilize in the forest related applications also, because another thing I show you for the flood monitoring level, for flood level monitoring. That is also, we have one dashboard here. Yeah, this is flood warning system for, we implemented it for Technopark. Actually, this is the one replicable model for the Kerala. So we put it we take a small river, Tettiyar, which is in nearby Technopark. The Technopark affected flooded in the last 2023 time, rainy time. So they required some warning systems for them. So we deployed 7 water level sensors in that area, in a 15 to 20 kilometers area. And also we deployed rain gauge stations and automatic weather stations in the watershed area. So once the rain is happened, then we can predict the water level will be rise, and we can inform the corresponding authority. It may be flooded your the Technopark area. That's the main application for this work. Actually, this is the platform we provided to the Technopark authority, and they can monitor each green indicates the 7 water level data, which is in Karakutam to Veli tourist village. And this is the last, this is the warning levels. If yellow cross the moderate warning and red means the flood happened, flood started in their area only, means Technopark campus only. So we can easily replicable these type of flood warning systems in forest related applications as well. Actually, which is we can deploy the water level sensors in the forest rivers, then collect the data from that, and we can predict. Also, we can deploy the automatic rain gauge stations in the watersheds. Then we can predict if the flood is happened in the coming hours. Okay, these are the main applications. We deployed it in the in nearby. Coming to this, so LoRaWAN mesh, which is another, I mean, the continuing technology, because once you think about the LoRaWAN which gateway required a internet facility or an intranet facility in there. So in a forest related applications, we couldn't get the internet availability in the forest area, because which is a rural area, or which is which not covered in the internet facility. So one of the technology which is mesh gateways, we can put mesh gateways in the forest area, and the gateway communicated to another gateway, and the data will come to the final end of the border gateways. This is the pattern. Actually, these are this is for fire prediction, fire detection warning application. So these are the sensors. Fire sensors are deployed in the inner forest area, and this is one gateway which have no internet facility and no connectivity. So this gateway communicated to the next gateway through the LoRaWAN network itself. So the entire spectrum is acting as gateway to gateway communication and gateway to edge devices like fire sensing sensors, and we can communicate from one gateway to another gateway, and that gateway to another gateway, and we hop it 5 or more times. Then we collect the data in the border gateway. This green indicates the border gateway, and which have a internet facility or a intranet facilities. So the data will come to our server platform through that. So in the forest related applications, we can utilize this type of LoRaWAN network, and we can deploy the several sensors like fire monitoring sensors, or for the flood warning systems, for water level sensors, for climate monitoring we need automatic weather stations and rain gauge stations. So any type of sensors we can deploy under this umbrella, in this LoRaWAN mesh umbrella. We have tested this technology in our ICFOS office. And this one is actually implemented in Germany. One Dryad is the one of the private team. They are implemented a forest fire early detection system in Germany in a wide around 10 to 15 hectare area of forest. And we approach them, and we will get the devices from them, and we will plan to implement one POC in future here Kerala as well. So that is the plan, and this is another application for the forest related thing. So actually these are the main applications I convey to you in the forest related case, and these are microclimate monitoring stations, air quality devices, water level devices, soil sensors. Another research entity is acoustic based rain gauges in-house. Actually this is not a non-mechanical, this is a non-mechanical device. Actually all the rain gauges are typical rain gauges are tipping bucket based, because there is a mechanical movement, and irrigation department approached us, there is maintenance is more. So they if the non-mechanical rain gauge is available, then that will be the better option for them. So we have some researches happen in the last two years, and we develop a single POC, but that is not much accurate, which is around 70% accuracy we observe, and we need to work it more. So the acoustic based rain gauge is one of the new initiative from ICFOS also. These are the LoRaWAN based IoT applications from ICFOS, and we can utilize these applications in the forest related applications, forestry applications. Like these are the list of applications I just listed, and you can connect these technologies and sensors to this type of applications like forest microclimate monitoring, health and tree physiology, like all the applications we can utilize in the any sensors we can collect the data through the LoRaWAN network. That is the main part of the IoT, the Internet of Things case. So this is just a way forward of ICFOS. Actually, we working in the Edge AI, Open IoT, open source UAV also. We have a unmanned aircraft vehicles, and we developed open source based drones for surveillance and forest related monitoring, means photogrammetry applications like that. So we'll be releasing in 2026 itself. And we have collaborated with IIT Tirupati, IIT Bombay, and IIT Kanpur for research related activities, and KFRA for the forest related research activities. I think we will start a good project in 2026, which is for human animal conflict based or forest related applications as well. So these are from my side, and coming to over to you for any questions if related to this.
So any questions related to this topic?
Yeah. Thank you for nice presentation.
Thank you.
Just wanted to ask one thing, like when you're saying about the devices,
yeah.
Do you have the certification, like regarding AWS, you're mentioning it, but what about the accuracy?
Yes, sir. We have utilized the industrial-based sensors, which is certified from WMO or IMD. Which the sensors, like Davis is the one example. Davis rain gauges we have utilized their sensors, and we didn't manufacture any sensors in-house. We procured from the industry. I mean, Davis or another manufacturers directly from them. That's a certified. Okay, that is certified one. Certified sensors we brought, and we integrated in our LoRaWAN technology and collect the data through the LoRaWAN connection, network.
Okay, fine. And if anybody want to interest, like what will be the cost for AWS?
AWS is nearly, the last time we offered 6 lakhs for the IDRP.
Okay.
There's a different AWS units are there, DAV kits are there, industrial based sensors are there. What is the application? I mean, related, the cost will be different.
Okay, thank you.
Because several departments need just information about the rain, not much accuracy required. So they can utilize the DAV kits.
So any more questions from the audience side? No. So, sir, I have one question regarding this LoRaWAN. Per node, what is the range?
We get 27 kilometers in Trivandrum.
Per node.
Actually, LoRaWAN is a gateway. I mean, we have a wireless area network which have a gateway, and that gateway can accommodate 1,000 nodes in a single gateway. So the edge, nodes means edge device to gateway, the distance we observed 27 kilometers in Trivandrum. But that is not stable, but we recommend it in 2 to 5 kilometers. That is the stable network. We deployed, because we 140 square kilometer we have in Nemam Block Panchayat. We deployed 10 to 15 gateways in 140 kilometer square.
And second one is, what about the swarm robotics?
Swarm robotics, we didn't attempt that, but we supported with the IIT Kanpur. We prepare a small drone for them, and we assembled it and tested in Trivandrum and provided to the IIT Kanpur for the swarm drone applications. Not started in house because of some fund
Will be much
limitations.
more helpful in
Yeah.
forest research.
Yeah,
That's
yeah.
swarm robotics. Thank you, sir.
Yeah. Okay. Thank you.
So that was a wonderful speech. Next, I would like to invite the next speaker, that is Dr. Girish Gopinath. He is a professor and head of the Department of Climate Variability and Aquatic Ecosystems at 𝐓𝐑𝐿, Kochi. With over 25 years of experience in geology and geoinformatics, his specializations in geospatial applications for water resources, natural hazard assessment, hyperspectral remote sensing, and web-based spatial decision support system. Dr. Gopinath has international experience with Sultan Qaboos University, Oman, and GEMS International NV, UAE, and has led over 32 major research projects funded by agencies including MOES, ISRO, DST, UNDP, and KCSTE. He has published more than 125 research articles and serves on the editorial board of Scientific Reports, Nature. So I would like to welcome Dr. Girish Gopinath.
Thank you, Mahesh, for a nice introduction. So my outline of talk will be, I will start with a geospatial technology. Then I will spend some time on satellite remote sensing. I think already there is a morning session, it was there. Then maybe concentrated on UAVs, drone-based studies for a forest. Then there is what is AI, machine learning and deep learning. And if time is there, I will give us some slides about open source data also. So when you talk about geospatial technology, you are using this technology in your daily life, okay? Same as Swiggy or Uber Eats, Uber Ride, all the things. So when you see the lower part, there is a LiDAR, then radar is there. Nowadays, for a forest biomass, we are using the LiDAR technology. That I will show you in the later slides. Then if there is any monsoon is there, we are using radar. Then when you go the next stage, there is a lot of satellites are available in orbit that you are calling as the Earth observation satellite. More than 40 satellites belongs to India, like a Resourcesat, Cartosat, Resat, like that. When you go again up, you can see a geo-augmented GNSS, that's a global navigation satellite system. So each country develop our own system, like, so NAVIC is not fully operational. When you see, you start with a GPS, GLONASS, Beidou, Compass, Galileo, there's a lot of GNSS are available, but NAVIC is not fully operational. So what is our aim is to collect data. Okay, a piece of paper is a data. Suppose it is having a geographic coordinates, it's a data, right? So you are getting a data from your mobile, from a satellite, from an aircraft, or from a GPS. Finally, you are inputting a common platform you are calling as a GIS, right? When you start GIS in 90s, you are calling as a desktop GIS. Now you are calling as a cloud GIS, AI GIS, then the mobile GIS, internet GIS. There's a lot of development in GIS part also. So basically, when you talk about forest, we can apply, this is a tool only, right? The remote sensing is a tool. You can apply in any field, right? When you talk about remote sensing, whether it is a satellite or airborne, it is basically we are monitoring forest, right? You can map and monitor your forest, and also you can model also. Suppose when you come down, the resolution of data is very important. Suppose the scale of mapping is very important. You have to keep it in mind. Based on your project, the scale of satellite, the resolution of satellite, whether it is a spatial, temporal, or radiometric, it is very important, right? So when you go down, you need a high resolution data. There are a lot of high resolution data are available from US satellites like an 𝐂𝐻𝐿, Cookbird, WellView 3, 5, it is a 35 centimeter resolution. So when you talk about drones, drones will play an important role. I will show you we are is a part of project with ISRO. It will be helpful for the biomass mapping and also for surveillance of your forest. Then IoT is already explained by Shahir. So to study the microclimate, soil moisture, forest health like that. So AI will play an important role. Suppose you are getting a lot of data. Data means it is a huge, it's a big data concept or like that, whether your data may be a structured, maybe a non-structured or semi-structured. So you are getting a data from a drones, it is a very huge data, maybe 1 GB size or for LiDAR data is there. So how you can do AI and ML? Finally, there is a cloud computing and big data concept is there. Again, it is depending upon your data source. That is that thing. So when you sum up this remote sensing, basically we'll start with the black panchromatic photos. You are using a thermal photos, you are using a micro photos, or you are using a hyperspectral. Hyperspectral is a new branch of remote, not a new branch. It is again the big data concept is coming in hyperspectral remote sensing also. Suppose you want to study the species identification for a high resolution, you will go for a hyperspectral remote sensing. In your multispectral, you can see what is the type of forest. It is a degraded forest or evergreen forest or like that. When you go to a species level, you will go for a type of remote sensing called hyperspectral remote sensing. But the data set is very huge. So there is a new branch is called GeoAI, right? GeoAI is you are using daily in your life, right? For example, for a traffic signals, all the things. What it will do, it is basically used for a precision mapping. Or suppose you want to study the urbanized area, this algorithm, GeoAI is one of the algorithm, it can extract. It is just like your supervised classification of data. You can classify your data. And there is one more application for this for precision farming. A lot of countries are using GeoAI for a precision mapping. So when you go to the remote sensing, whether it is a spaceborne or an airborne or a ground base or a terrestrial, sometimes your airborne will not help in the forests because it is a very thick forest. Then you have to go for a terrestrial scanning. So that part is also you have to keep it in mind. When you talk about remote sensing, there is a lot of satellites are available. Maybe it is a medium resolution, coarse resolution, or high resolution. And based on your requirement, you have to select what type of sensor you need. Suppose it is a forest fire, you can directly go for a thermal remote sensing, right? Suppose you are studying a forest during the monsoon season, your optical remote sensing will not work. Then you will go for a microwave remote sensing. So resolution of data is very important. What is your type of investigation, right? Suppose you just want to study the forest, whether it is an evergreen or it is a degraded something, you just need a coarse or a medium resolution. Suppose you want to find what is a species in the forest, then you have to go for a high resolution satellites. So these are the data you can download free of cost. These are the remote sensing satellite you can download data free of cost. The Landsat, it is start from the Landsat 1 to 9. Then Sentinel, it is a very beautiful data. You can download from Copernicus. Landsat, it is a 1 to 9 series, like a different sensor like a TM, ETM, OLA like that. This data you can download free of cost from a USGS or NASA website or LPDAC. The Sentinel you can download from a Copernicus. Then there's ALOS data from Japanese. Then Indian remote sensing data you can download from a Bhuvan, or there's a new site called Bhunidhi also. You can download all IRS series of satellite, whether it is a list 1, list 2 or list 3, like that. So these are the broad application for remote sensing. When you see based on this, you can do for a change detection. What is the changes in your forest? For last decadal changes, last 20 years, you can do from a remote sensing. Then the deforestation assessment, biomass estimation, forest fire detection, drought assessment, all the things you can do from a remote sensing purpose. So it is a simple classification of image, right? This is a mangrove forest, not this one. Suppose this is an image you can download from free of cost from Landsat or Sentinel. Just you have to say classify means it will classify vegetation, water, and urban area. How it will classify? It is based on the clustering algorithm. Those pixels having a similar property will fall under the similar class. Then I will say classify. So you will get an image. It is a classified image. So you will get a what are the vegetation, what is the total area, what is the water body, all the things. It is based on the lot of algorithm is there. ISO data, iterative self-organizing data algorithm. So it will classify their own way, right? That you are calling as a unsupervised classification. So you will get an image like that. Suppose I want to classify mangroves in different classes, whether it is Agulacha or Marina, that also I can do from a satellite data. So once I will do this one, I will get an image like that. This is an image of Wayanad. Actually, it is a level 4 classification. If you go to NRSA website, there's a level 1, level 2, level 3, and level 4 classes are there. Level 4 will give a more detailed information of your terrain. So this is how from a medium and post satellites, how your land use can be obtained. So we are more interested, if we talk about forest, there is a lot of other technology like LiDAR, the RGB cameras, thermal cameras, multispectral and hyperspectral. So again, your type of investigation, you can select what type of sensor. You can put any sensor in drone, right? You can put a thermal sensor, you can put a hyperspectral sensor, you can put a LiDAR, or you can put a same as your mobile. There's a RGB camera also you can put. So this you can do based on, it is a purpose only. So what are the wavelength it is? I will not go to all the technical details, it will bore you. So this is one of the project we completed. We had a 2 project with the ISRO. This project is called NISR, NASA ISRO Synthetic Aperture Radar. It is a joint project with the CUFOS and CWIDM. This first stage we finished and submitted our report. And the second phase also we got. Second phase, CUFOS, CWIDM, and KFRA. That Dr. Srijit is also a part of this project. So our aim is to develop some 1 hectare inventory for northern. It is in Maharashtra, Shimoga, as well as Nilambur. That's our first prepare a virtual plots for a 1 hectare. So that's the aim of this project. And also we have to take a drone images, like a LiDAR images like that. So now NISR is already launched. I think it is last month it is launched, NISR. So it is the first global satellite having a dual frequency. It will operate both in L and S band. There's no other satellite in the world having a dual frequency, right? That's the importance of NISR. So these are some field photographs, Shimoga. So this is a field data we collected. It is just like an Amazon protocol. We take 100 by 100 meters in the forest inside. Then it is divided into 9 sectors. We took soil moisture. These are the plot we established already, but we already having a virtual plot also. So these are the parameters we took from the field, like a tree height, each plot. We are taking a tree height, the tree canopy density, that is a leaf area index, then soil type, soil moisture, then finally we took a LiDAR data also. So these are some field measurements, and these are the instrument we used, Luminous. So this is a plot of Shimoga. So we took 3 type of forest. One is a degraded forest. It is in Maharashtra. The another forest we took in Shimoga. It is both evergreen and degraded and mixed. Then we took Nilambur. Nilambur is consisting of plantation of teak. Then there is a mixed forest. That is why we selected these 3 for this NISR project. The aim is this data should be used to calibrate NISR sense data, biomass data. That is why we collected this first stage. So these are some plot in Shimoga. So these are the drone image. From this we are collected, you can see there's a canopy height. We got it from a drone measurements. Then we calculated AGB, that is a tons per hectare. The ground sampling distance is 2 centimeters. That is why the scale of survey is very important. Suppose when you talk about satellite, you are using a 1:50,000 or 1:4,000 for aerial photos. For LiDAR, the ground sampling distance is 2 centimeters. That much accurate you are getting this one. So this is a biomass of Shimoga. From this, you can see it is a tons per hectare. You can see it's a report we submitted for the first stage. This is a phase 2. The phase 2 is already on, and already we completed last week. Here our plot is 10 kilometer by 10 kilometer. We are using for a Maharashtra, Osangabad, Shimoga, and this one. We are taking 10 meters. Some are 20 by 20. Some are different plots to take measurements. So here this LiDAR will give you what is the height of the tree, what is the diameter of tree. Then we can calculate some volumetric equation. We can calculate what is the height of tree. So finally we'll calculate the biomass. So this is about the how the forest biomass we are using for different parts. So when you talk about, that is about LiDAR image, this is what is a RGB, right? The RGB means it's a high resolution data. This is one of the project we done in Dehradun. So you can use the different software, right? Whether you can use an ArcGIS or QGIS, there's a Microsoft, MicroStation. There is a lot of data are available, open source as well as this one. So first you are using an orthorectified photos from RGB. That means it is a vertically corrected. Then you are using subset image. Here classification different between coarse resolution and high resolution. Here you are using a object-oriented classification, right? For a high resolution and medium resolution, you are using a pixel-based classification. So those pixels having a similar property will fall under the similar class. That's the science you are using in the medium and coarse resolution. But for a high resolution data, you are using an object-oriented classification. You are classifying your image into number of objects. Then you are classifying. Then you can see the shadow is also one of the class. That means your data is having a high resolution. You can even classify the shadow of the region. So that is a basic difference between high resolution and medium resolution data, because your classification technique will be different. So I will just talk on machine learning, because you are getting a lot of data, right? Whether you are using for a biological conservation, you are using a forest landslide, you are using any type of investigation, because when you are using a LiDAR, you are getting a very huge data, right? Because even what our purpose of NISR project is that we will make a measurement from the field that should be matched with the LiDAR, right? We are making measurement in field, we will measure the height of tree, diameter of tree. The same thing should be happen for LiDAR also. We will fly the LiDAR, we will get some 90 to 95% accuracy. This model will be used for a NISR satellite. This is a global exercise. India is not only doing, Europe is doing, a lot of people are doing. So we'll develop a model, and the thing you have to keep it in mind, the NISR will give you the product. No need of processing your data. It will directly give the biomass. No need of any exercise, no need of downloading any data. It will give you directly biomass in tons per hectare. No need of downloading any raw data. So when you talk about machine learning, what is the importance of machine learning is that first you have to keep it in mind, what is your type of data, right? That also you have to go for any type of analysis. If you are having a lot of data, you have to keep it in mind, your data is a linear or non-linear. Suppose suppose rainfall will happen, your moisture will increase. That's a linear equation, right? First you have to see, then you have to see your data is structured or non-structured or semi-structured. Then these things you have to keep it in mind before going to for a machine learning and or a deep learning like that. So we are using 3 models. One is a standalone, the another is a hybrid, and 3 is the ensemble model. So standalone means you are using a single model, right? You are using a random forest or anything. Suppose you are using a hybrid model, means the output of one model will be input of second models. That means you are combining 2 models. You are getting an output from random forest. That output will be the input of support vector machine, right? When you are using an ensemble model, you are running all the models, you will take an average. That is why the difference between standalone models, hybrid, and ensemble model. And also we are done, one of the student is doing, a lot of things he is doing. I also started from his side only. I started studying all the things. So when your data set is low, no need of going for a machine learning. I think you heard about ASP, fuzzy, like that, that's the multi-criteria decision technique like that. When you compare, the result is same. Your data set is very less, no need of going for a machine learning. You can stick on your ASP or multi-criteria decision technique like that. So this deep learning is a game changer, right? When you see the deep learning and ANN, it is based on number of hidden layers. But again, you have to keep it in mind. The deep learning is a subset of machine learning, right? So again, if you are having a more data, you have to go for a deep learning. And also keep it in mind, for machine learning will well work for structured data, right? The structured means your date, time, the rainfall, all the things. If it is a deep learning can take care of non-structured data also, whether audio or video, all the things. That's a game changer because we are doing a lot of exercise. And this is one of the study conducted our student regarding, we are doing a lot of work, not only for a forest, we are doing for landslide, lot of other things for a machine learning and AI. So in this project, we have to find out what are the forest fire risks. We are also doing for a wildlife conflict also, that is also, it is finalized. To study Wayanad, because you know the Wayanad is a plateau, sometimes there is a frequent fire will happen. So our purpose is different, right? For this paper, you can use machine learning in a different way like that. So we develop lot of layers. These layers you are calling as a whether it is a distance to road, what is a slope, what is a alter aspect, all the things will elevation, all the things we develop. Then our aim is to run some 11 machine learning model, right? Our aim is to not to compare a machine learning, which learning is good, right? Our aim is to, a pixel should give the same result for all the 11 machine learning algorithms. That pixel is considered as the most vulnerable for the fire. It is a pixel based classification, right? Suppose you are converting your area into a pixel base, that particular pixel will satisfy all 11 models. That is the most chance for the fire, right? That we have done for entire Calicut. Sorry, it is a Wayanad. You can see these are the final maps. You are getting a high vulnerable, low, or a moderate vulnerable like that. So I will not touch this one. Is a very long back we are done, and also we are done for a biological conservation. One of the students he carried out. I think it is 𝐓𝐑𝐿𝐴 something. To study the redwood, how the redwood pattern is changing, because one student, she collected a lot of GPS value with us in the redwood area. So here also we have done a lot of covariate specialty, like these are the temperature rainfall dependent. If you go to B3, B11, all the things you can get it from the website. Then what is the clay carbon exchange like that. Finally, these two models we are done for random forest and BNN. For the prediction, whether in future, whether this sandwood, this habitat suitability for this 𝐓𝐑𝐿𝐴 for redwood. So it is already explained by Sri Safiq about IoT. I will not explain anything because he well explained, very simple. With a chair, he explained what is IoT, because I am not an expert in IoT. And you can see the most of data is free of cost. I put in the bottom, global observation of forest cover and land use dynamic. It is a part of NASA program called a GeoFC. It is actually Maryland University is take care of this part. So you will get an all fire data free of cost. And right, you can download from this website. And there is a new thing students are very well doing is a Google Earth Engine. Nowadays, no need of purchasing a satellite data, no need of purchasing any ArcGIS software. The Google Earth Engine will take care of. When you go to Google Earth Engine, just a search in Google, you can see you will get there is a 3 PDF file. One is a basic, the second is a intermediate, then advanced. Just you read it. Nowadays, you will get all the code from ChatGPT. The Python code or JavaScript you will get from a ChatGPT. Then you enter in Google Earth Engine, you can play, you can NDV. First you have to keep it in mind, you have a thorough idea about your science, right? Unless you will be, your final map will be very different. That is why nowadays the students will make a very good maps, But the final marks, we can find out whether it is correct or not. The ChatGPT will give you something, but you know what is NDVA, what is the application of NDVA, how it can be applied for forest, whether it is to study the forest health, mapping like that. So you know the science, then only you will go search for ChatGPT only. But thing is that now codes are available everywhere, right? No need of studying a lot of things. Then if you can select the data, the cloud-free data, you can what are the change detection of forest. It will do all the exercise. You need a good internet connection, right? You no need of any software or any satellite. Satellite data will take care of the Google. You can select whether mode is Aqua or Terra, like that. That also lot of papers are nowadays coming in Google Earth Engine. So these are the site you can download from your any type of data, the remote sensing or Landsat, like that. So this is again Copernicus, European Space Agency. The Sentinel data is very beautiful, right, than Landsat, because the spatial resolution is good for the Sentinel. You can do, in Sentinel also you can do some of the climate change, for it will give a carbon dioxide value, methane, lot of things. But the pixel size is, I think, 1 kilometer or 4 kilometer. I am not sure, 1 kilometer or 4 kilometer, something. I will show you two video. The one is about terrestrial LiDAR. The another is the processing of biomass. Only 2 minutes. Can you just show first video? No. Okay, for a final. LiDAR. അടുത്ത വീഡിയോ ഉണ്ട് നെക്സ്റ്റ് മീഡിയ പ്ലെയർ അല്ലാതെ വേറെ എടുത്ത് ആദ്യം ലൈഡാർ എടുത്തോ മീഡിയ പ്ലെയർ അല്ലാതെ This is a terrestrial LiDAR. It's a CWIDN campus, Calicut. We took some 10 years back. You can see this is a terrestrial LiDAR. So it will cover 360 degrees. So these are the cloud points, right? From a single seconds, it will take 10 million points. I think 1 million. I am not sure now. I am not remembering all the things. So this will give a cloud image, right? This is a institute main building, portico, all the things. Now I can take any type of measurement. I will get a measurement, both three sides, X, Y, and Z. The Z is very important for any type of forest hydrology or any type of flood mapping. The Z values are very important. So LiDAR will give you very good, accurate Z values. The second one is actually how we are processing our LiDAR data, because when you want to study the biomass, this is actually software called MicroStation. It is a very powerful software than ArcGIS. If you are going with some other type of scientific application, like any type of calculation like that. So actually, when you load LiDAR data, it is very huge. That is why it is taking time, right? Because it will below some 1 GB data like that. Suppose if you are taking the Nilambur, it is a tera data. We need a 1 TB data. So the images look like this. Wherever you are getting green color, it is a forest, right? Then you are getting lot of black dots. It is some space without leaf is there. So what this software will do, when I do this type of X and Y, it will make a tree pattern like that. It is from LiDAR data. Suppose I want to take what is the height of tree, it will take what is the height of tree. It will take what is the diameter of tree, I will get a diameter of tree. It is manual, but this software is automated, right? We are giving some algorithm. Suppose you know what is the difference between DEM and DTM. DEM is a bare soil. Suppose you are putting some plant above that one, you are calling as a DTM. From this, I can also remove the trees also. So it will give you them also. Suppose I can put the tree also, it will take the measurements like that. So you will get what is the height of this tree, then what is the diameter of tree, it will take automatically. This is only manually, it is calculating some 0.4 or something, meters something. So this is how LiDAR data is look like that. So fully what we are not doing any manual work. So nowadays there's a lot of algorithms are there, so it will take care of, right? So how we are checking our data means already we had a field values. We already having a field values, data is there. We know what is the height of tree, what is the species name, what is the diameter, what is everything we have. After that only we will do the LiDAR. So the LiDAR and this field survey should match about 95 to 97%. Then only that model will work for your satellite, NISR satellite. Thank you for a patient hearing, and my sincere thanks to organizers of KFRI for this digital for this tropical forestry conclave. So any question?
So any questions? Yeah.
Thank you, sir, for that wonderful presentation. I have two doubts. One is very basic. Actually, I am new to this. So my first question is, actually you have answered some of it, but if there is no ground data, can this AI detect this elevation? If it is a thick forest, can AI on its own, without any ground data, detect whether it is elevated or not?
AI is different, right? Once you collect data, AI will work, right? Once you are having a data, if you are having a big data only, we'll go for AI, unless no need of going AI. And also, I think you wrongly understood this thing. Suppose the canopy is high, the LiDAR will not work. It will not penetrate.
Okay.
I think you mix AI and LiDAR, right?
Maybe.
If the canopy is big, it's a very dense, the light will reflect from the top of it will not penetrate.
Yes.
That time we will use a terrestrial LiDAR to find out the biomass. But usually for Nilambur it is a plantation, no issue, because we have not faced any issue here for Shimoga or this one. So light will penetrate in between the trees like that. But it is a very thick forest, sometimes LiDAR will not work. Always think that remote sensing is a tool, sometimes it will work. Then you have to do some allometric equation to generate a biomass. That is a deep forest, it will not work. Drone-based LiDAR, it will not work.
The second question is, you mentioned something about AI using its pixels to understand this fire incidents. Can you elaborate on that?
Yeah, this again depends. When you're using any technique, you need a data for validation as well as calibration, right? You are getting an output. You want to know whether your output is correct or not. You have to go for a sensitivity analysis. So about sir question, can you repeat that question? Oh, okay, I understood. So you can do here what we have done. We done 11 models. It is a pixel. You can done different way. It is a vector and raster. It's a pixel based, or you can do a vector based. Here we will do all the pixels. We will assign some values. So the same pixel will exhibit. It will answer all machine learning. Select this pixel as a more prone to fire. That pixel will be select as a high vulnerable. So you need a fire data, right? To whether that pixel will be, because I think those who are working on AI, there is a diagram called FOR diagram, right? FOR diagram, what it will say? True, it is a true, false, true, positive, and true, negative. Suppose there is a landslide is happened. The system is predicting the same area as landslide. I am saying it is a true positive. Sometimes it will not predict. That time I am saying it is a true false. That means there is a FOR diagram is there. That diagram will give you whether your model is correct or not. That is why nowadays there is a new branch of AI is called explainable AI, right? Some of the mistakes you can again correct it from an explainable AI, right? So how you work on data? Data is very important, right? You simply put some data, but data should be a structured data, right? The based on data set only you are getting data. The pixel is based on your type of experience, whether you need a pixel base or it is a vector base. So in this project we are taken entire this one Wayanad, we are classifying into 30 meter by 30 meter pixels. So we will assign some values for 30 meter, right? Whether the event is there, we will assign some values like that. That is our concept for that paper.
Okay. Thank you, sir.
It's clear?
Yes, yes, almost.
So, any more questions from the audience side? Yes.
Hi sir, my name is Krishnaja. I'm from molecular biology background, so I'm very basic to this GIS and all this advanced tools. So I'm just out of curiosity, I'm asking, like if you're into a forest and if you want to count the number of the trees, we are using the drone images. So the how this AI is analyzing those drone images, I mean, if I'm not wrong, we are actually looking into the canopies. So overlapping canopies can be there. So how accurately we can predict the number of the trees in a forest area using this?
Now for this project, we are not using AI, right? For a
for analyzing the drone images.
No, for NISR. Okay. We are using one software only there. In algorithm, it will give you. Here we are not using AI. It is 100 meter by 100 meter only, right? We are not using any AI. The system will give you what is the height. That is again based on some allometric equation, right? Sometimes we will not get it. It will not penetrate, right?
Yeah.
That time we have to use some because Forest Survey of India already given for each species for allometric equation. So sometimes we'll take that one if it is missing. So it is a combined in different things. Now we are not looking for all the things because when you see this Nilambur, it is a teak plantation only, right? Because usually it will, the phenology when you see it will shed the leaf. That time the biomass mapping is easy. We will see the season also. Usually we will do the biomass mapping. Usually for Kerala we are using April, May and June, right? That's a season like that. So if it is a very big canopy is there, you will not get a result from a LiDAR. So yeah, you can add this point.
Yeah, so we are practically working on this domain of LiDAR also. So here it is not like a greenness, okay? It is working like X-ray. It's like it's seeing the structure, like the skeleton. So once you remove, like there are some techniques called like filtering. So by using filtering, you will remove all branches. Then only one trunk will be there, okay? Yeah, so if you use some kind of steps, like evergreen forest, you have the multi-storied arrangement. So based on some kind of vertical and horizontal analysis, you can be seeing the horizontal cover, like a canopy cover percentage, and also if you use some kind of techniques, you have the like understory vegetation, middle story, top story. Okay, it is like a not greenness. There is no any kind of avoidance. So like it is like almost like a millimeter level of accuracy. So here there is no much problem. Only thing is there are more steps involved in the processing of the LiDAR data. Yeah.
So any more questions from the audience side? Yeah.
So very nice presentation, sir. So just like from forest title background, I want to know about the soil moisture in the forest, like using remote sensing. What is the progress?
Actually, we are taking a field measurement only.
Remote sensing?
No. We are not using. For NISR, you will get a soil moisture. There's another American satellite called what is it? SWAT or something. In that, you will get a soil moisture, right?
Any depth?
Yeah.
Sir,
microwave will give you soil moisture.
Till what depth, sir? Depth?
For dry soil, it is 50 centimeter. As a theory, I don't know exactly. As a theory, for a dry soil, you will get a 50 centimeter only. 50 centimeter, it can penetrate. But I think NISR will give you soil moisture. I think so. It's a biomass. Lot of other applications are there. But our NISR will give you soil moisture.
Okay. Thank you, sir.
Yeah. Any more? So just tell me about the scope of hyperspectral data using, acquiring hyperspectral data using drones, or especially in forestry. So what are the advancements going on in this field?
Okay. It is very expensive. First, you have to keep it in mind because sensor is very costly, right? Even LiDAR also last time what happened in Nilambur, it lost something in the lens or something in the forest. We cannot retrieve. Hyperspectral, even the land hyperspectral, we paid almost 80 lakhs with all the infrastructure. There's a new hyperspectral imager is there. It is more than 100 lakhs. But there is a beautiful application you can from a hyperspectral remote sensing. You can easily classify the species, right? Suppose you once you develop a spectral library for different type of forest or species, you can do as a one of the benchmark for a forest because nobody has developed for India what are the spectral library for the forest species. Once you are having a library, you can easily classify unknown things. Now we recently we published a paper called soil spectra can be replaced by wet chemistry. No need of doing all wet chemistry in your soil. What is a sodium, potassium, something? This soil spectra will give you for what is the type of your concentration of these chemicals. That's the recently it is published, and I think last week this is the Times of India also they published. And also we developed some spectral library for Uma rice. Uma is one of the rice in Kerala. So we developed the first time for the spectral library. For forest, if you develop a library, it is very useful, right? Somebody will develop some library for the spectra. It is, I think maybe NRC, maybe they are, I don't know exactly. Maybe that is very useful for your classification. Because once you know the unknown samples, it will act as a trainer. It will train the system and it will say what type of this thing. So maybe future, because when you go to USGS, they are having all spectral library. When you see any type of vegetation, they are having a spectral library for them. So for them, it is very easy to classify their land use. Suppose the land use is changing every time. It is this spectral library act as a trainer. So you can get an unknown values, right? If you get an unknown, you can make as a known like that. That's the importance of hyperspectral. It's clear.
So thank you very much, sir, for a wonderful presentation. So the two presentations have been done. So let's go for our third and the last presenter of the day is Dr. Annie Elias. She's a Ramalingaswamy postdoctoral fellow at 𝐓𝐑𝐿𝐵, Coimbatore, and founder of 𝐿𝐑𝐄𝐒𝐋 Analytics, who will join us online. She's an online. So Dr. Annie Elias is a strategic consultant and scientific innovator with over 12 years of postdoctoral experience applying artificial intelligence, genomic analytics, and predictive modeling to agriculture, forestry, and biotechnology. Founder of 𝐿𝐑𝐄𝐒𝐋 Analytics LLP, she leads cross-domain initiatives integrating omics, machine learning, and sustainability analytics to deliver scalable solutions for government, industry, and academia. She has held key roles at Risk Management Solutions, Monsanto, and Cornell University, with a proven track record of delivering high-value global funded projects that translates artificial intelligence and genomic innovations into real-world impact across forestry, agriculture, and climate resilient systems. So let me welcome Dr. Annie Elias online.
Thank you, sir. Thank you, sir, for the introduction. I just want to mention that this Ramalingaswamy Fellowship is not a postdoctoral fellowship. It's given for people who had done their PhD and postdoc outside India. This is a government scheme where government invites people who had done all the things and then come back here and develop and some technology, pioneer some technology here in India. So it is equivalent to Scientist D or in India. So yeah, but thank you for the introduction. I just want to share the screen. I hope you can all see my screen. I just want to start by reading the passage. Greetings, Earthlings, or as I prefer to think of you, a fascinating collection of blue and green. Let's see what wonders this view of your world holds. I am Gemma, and I'm here to observe, analyze, and perhaps occasionally offer a slightly unsettlingly insightful commentary. Let's begin. This is the message that was sent by the large language model from Google, which is Gemma, and this is the first message that was sent by them. This is a part of the space AI race, meaning like big tech companies are exploring space to establish their data centers, to use the solar power, continuously available solar power, the space cooling effect, and reduce the impact on due to environment. So here I just want to see, like in this time, like what we can do in forestry, this changing time, how we can use AI for reimagining decision making and management. So I just want to give an context of like how this AI is emerging. This is a large and a rapidly growing area where a lot of money is involved. As you can see, in 2025, the global market value for this AI services, software, and hardware is $390 billion US dollars, and it's expected to expand to 3,400 billion US dollars. For all the service, software, and hardware, and there is an expectation that 29.3 annual growth rate for just for the software part. And if this is the global scenario, I think India is not behind. India is also having the AI market projected to expand rapidly, where we are currently valued at 6.8 billion and is expected to grow to 27.7 billion by 2030. Government is actually encouraging global AI engagement in establishing data centers, research centers, and developing tools and skills. For example, in Ministry of Education has established a AI Center for Excellence in Healthcare at Indian Institute of Science, Bangalore. And a lot of big tech companies are investing. In this last quarter of this year, a lot of investment is happening from global tech companies. For example, Microsoft investing 17.5 billion, Google investing 15 billion for establishing a data center hub in Visakhapatnam, and Amazon announcing 35 billion. So all these, and plus NVIDIA collaborating with the Deep Tech Alliance for establishing processing units. So all these investments are coming, and India is considered as a big powerhouse. And so what is the big deal? Like why AI is taking this momentum? Like why it is so important? I just want to give an overview for the advantage of the students. What it does, like it transforms big complex data into fast, predictive, and automated decision making. So a model, an AI model, can take data which are big and diverse. It can take structured data, like a spreadsheet type of data or sensor data, or it can take unstructured data like text, images, and audio. And it can also take time series data like weather data or market fluctuation data. And all this can be taken together, also like simulated data. All this can be taken together and can be placed in the model. The model can learn the patterns and anomalies and find relationships. And with more data, with more variation added, an improvement can happen continuously. And this will happen in a fast pace. Like this processing of the data and identifying and learning the patterns happens in a fast pace. And with millions of data points being used, and real-time analysis is happening and reducing the time for analysis from weeks or months to seconds or hours. So based on this analysis and learning, a forecast will happen, and for example, like identifying, making a risk map, or identifying the risk early, or detect a problem early and make recommendations based on that. And this whole process can be automated, meaning like reduce the human interference and helps support the humans to make more informed decisions quickly. So this is the whole big deal about AI, why this is taking why this is gaining the momentum. This is the whole big logic behind it. So I just also want to give a theory of like what it is, just again, because I was told that more students are there, so I just want to give a little bit of theory, like I would say like a highly simplified version of the theory behind this AI. So when we think about AI, we are thinking about statistics and deterministic models. So that's what is happening behind. And what it does is it reduces in all these statistical models, what our aim is to reduce the difference between the truth and the estimation. That when we talk about, in statistics, when we talk about the truth, we just give the value for any value like this, and that same representation with a hat on it is the estimation. So here the loss function is that reduce the deviation, is the deviation between the truth and the estimation, and our aim as a modeler is to reduce that deviation. So just want to give an idea of like a convolutional neural network walks through the unstructured data such an image, where it takes overlapping kernels containing the features that it wants to scan through an image. And these kernels actually scan every grid of the image and see if it can identify the object it is intended to identify. So, and it will give a score value. A score value meaning like a probability value whether an object is present or not, and also if the object is present, whether it belongs to a particular class or not. A class meaning, like for example, if you are scanning the room, it can be the class can be like you can train the model to identify a chair, a table, or a laptop, like that. Those are the different classes. And so this this scanning will help to identify from each and every grid, like whether a particular object is present, whether the object is belonging to a particular class or not. And based on that, a probability value will be given for each of these grids. And as I mentioned, like this idea is to reduce the deviation between the truth value, the truth, which is the actual thing that is present in the real world, and the estimation that is coming from the model. So you can see that everywhere you can see the hat value and the non-hat value. So basically this is looking at how much is the deviation, and there are some regularization parameters because we are taking a lot of features from different types of data sets. So to prevent any overfitting, some regularization parameters are also being used. And based on all these things, we select a model, a loss function, which eventually being selecting a model that actually fits better to predict or classify and predict objects in an image. So this is the basic idea behind an AI, basic theory behind an AI image classification, or in any types like this. You can actually use the same logic in any type of probability distribution or distribution for classification. So given that, like in forestry, we can use that, there are some high value uses for AI. Like for enforcement needs, when human resource constraints are there, and then there are scalability is required. For example, in forest protection, like with the camera trap and also with the thermal sensors and with computer vision models, we can detect the presence of a poacher or an encroachment or an illegal logging. And with that, we can make optimization for the patrolling, how to route it, how to route the patrolling, or how to develop a risk map. This can also be used for conservation and restoration. For example, in identifying the plantation survival or canopy closure, as the previous speaker was mentioning, or to identify the fragmentation, or species identification, or invasive species detection. This can also be used in monitoring and performance reporting and better planning, for example, in inventory, in biomass, carbon measurement reporting verification, and also detecting the pest or pathogen presence. And early detection will help us make better informed decisions and make prevention, help us to make some prevention activities, which this can also help in policy making, like develop a land use change dashboard, as previously sir was mentioning, like land use patterns are being studied in other geographies, and this can also be used. And also we can find out like the risk map for a fire or drought or invasive species, identify places and incidents where where the human-wildlife conflict or fire incidents can happen. So when it comes to the human-wildlife conflict, that is that concerning the public safety, that is a much faster adopting place where AI is being used. For example, using thermal drones, cameras having thermal vision, night and day thermal vision, and then computer vision based detection using from sound based or image based sensors. The data can be collected real time through images or through videos, and this can be analyzed to identify the incident or to identify whether there's a high possibility of a human-animal conflict is there. And based on that information, and the response can be optimized, like dispatch an enforcement team or position the resources like that. This can also be used in wildlife mitigation by early detection and reducing the false alarms, and then identify fire in places and optimize the resource dispatching. So in India, as I mentioned, like human-wildlife conflict management is a major driver for AI adoption. And there in this last quarter of this year, a couple of Indian forest departments already adopted AI methods like thermal drones, mainly for human-elephant conflict, to mitigate human-elephant conflict in South Bengal and Jharkhand. What it does is like it has day and night vision cameras, and there will be a real-time tracking of elephants. And these forest departments are planning to expand these use of sensors to identify poachers or other wild animals, where geotagged images can be sent to the officials, and officials based on that images, the response can be given. So, and also like in our Agriculture University, Kerala Agriculture University, in collaboration with the Engineering College in Kochi, had already developed an elephant alert, early warning system for human-elephant conflict mitigation. So this is a developing area. And also another the AI tool that is being used in forest departments in India is for fire detection. for predicting, for detecting and mitigating the wildlife. So this was deployed in Telangana back in 2024. Testing was done, and what it does is like it reduces the response, reduces the detection time from 6 to 12 hours to less than 2 hours. So that actually also it reduces the false alarm. And what it does is that it uses carbon dioxide sensors, thermal cameras, and satellite imagery and land cover type to make a risk map and a risk management map. And that actually helps in quicker response from the forest department in reducing the wildfire or spreading and identification, in reducing the spread and detecting the wildfire to prevent it from happening. So at 𝐓𝐑𝐿𝐼𝐂𝐀𝐋 Analytics, my company, we are also motivated to develop some AI-powered pipelines for inventory, for biomass estimation, for categorization, for pest detection, and for due diligence. We also develop some tools for accelerating molecular breeding in diploid and polyploid organisms. And currently the polyploid work is in agriculture crop. Our work was been featured in one of the Malayalam dailies in this year. So I want to, I know that most of the people sitting in front of me, although I cannot see, but are students. So I just want to mention that, okay, all these tools that we have been developing were motivated by a lot of stories. Some of them are personal, some of them are professional. And these stories actually help us to sit day and night and think about a solution. So I am actually here stimulating the innovators. Think about something rather than thinking about like the jobs are going away. Think about how jobs can be evolved. Jobs are evolving, and think of being becoming an innovator. Like for example, some ideas are like develop a tool for animal language translation, or develop a computer vision, AI, computer vision, robotics tool for self-driven trucks. So with that, I just want to thank you for giving me the opportunity to present an overview about how.
So thank you very much, madam. So it's open for discussion. So any questions? So any questions from the audience? Okay, it's no questions. Thank you, madam, very much. Okay. So one question. Yeah, one question.
Thank you for the excellent presentation. I am Dr. Nandakumar. I was in KFRI and now I have an organization and currently working. And you have done an excellent presentation. In fact, all the speakers have done an excellent presentation. The question, I wonder if some forest officer or senior forest officer should have been here to hear the deliberations. Because nobody is knowing what is the potential in the real field. For example, wildlife problem, human wildlife problem, real-time analysis, real-time decision making. All these things are lacking. Problems are there. Again, we have working plans, management plans, panchayat level plans, block panchayat, district panchayat, all integration. These things can be done with using all these technologies putting together, not in isolation. Of course, you can, when you do it in isolation, you can prepare wonderful paper, you can have impact factor, but it will not reach to the ground. So the task involved is how to make it useful to the field, because AI market is $1 billion. So how to make the revenue of the government, revenue of the department, revenue of the state to that level. That should be the aim for, particularly some of the administrators would have been there. Okay, thank you. Excellent presentation.
Thank you, sir. Yeah, I totally agree. Unless we can actually translate these ideas in real world and optimize the resource use, then it's all this billion-dollar valuation is of no use. So my thinking is that a lot of private-public partnership and involvement of younger minds should be there, and also empowerment of conventional thinking. People who should also be there. So only with all these putting together, the implementation is possible.
Yes. Yeah. Thank you very much. So.
Thank you.
So there is a small announcement. We have Mr. Anuj Sharma, the Managing Director of Indian Subcontinent, Campbell Scientific India Private Limited. He'll be giving a 10-minute presentation about his company. So Campbell Scientific is a world-known company for data monitoring in different fields like meteorology and agriculture, etc. Yes, over to you, sir.
A very good evening to everyone, and we really thank KFRI for giving us this opportunity. I'll try to keep it very brief, a very quick presentation about what Campbell Scientific is and what Campbell Scientific India is. So Campbell Scientific is a global company known for data acquisition and meteorological sensors. All these dots that you see are the offices that we have worldwide. Our head office is in Logan, USA. It's a 50-year-old company started by Campbell brothers, known for the ruggedness of the system that we supply, known for the accuracy and reliability of the systems that we build. If I talk about Campbell Scientific India, we have our head office in New Delhi. We have our regional offices in Kolkata, in Bangalore, and in Trivandrum. I have my colleague 𝐓𝐑𝐿𝑒𝐿 Matthew with me, who is from Trivandrum. He's the one who handles technical and commercial activities for this region. And then we are also responsible for other countries like Nepal, Bangladesh, Bhutan, Sri Lanka, and Maldives from the Indian office. The system has always been known, Campbell, the name has been known for the accuracy and ruggedness that we offer. You can have your systems running in the most extreme environments, be it anywhere. I'll take you through the different applications also, and you can trust that you will get your data. So we have multiple applications, whether automatic weather stations for different purposes. It could be airports, it could be research, it could be agriculture. You can have different parameters plugged into Campbell Scientific data loggers. Like for water, you can have different water parameters, water quality, water level, water flow plugged into the data loggers that we offer, and use it in any of the applications. For soil, we have our own soil moisture, soil temperature sensors. We have the profilers, we have the single point sensors. We are also into gas flux monitoring, eddy covariance. We have our sensors for CO2 and H2O monitoring. We have our sensors for 3D wind sonic anemometers. For eddy covariance field, where the data is very fast. 10 Hz monitoring is done for gases, for carbon sequestration, being deployed in forest, being deployed in urban. We also supply our systems in energy, mainly in renewable energy. When we talk about solar and wind, it's very much required for them to give a very precise data at the end of the day about their generations, as they are penalized for not generating enough energy. We are also into structure monitoring because we make the world's best data logger. We have that technology built in to monitor all the structural sensors very accurately. So wherever the dams are being built, wherever big structures, the roads, the railway bridges, recently the biggest railway bridge that was built between that Kashmir path has all the Campbell scientific instrumentation done across the length. Even the Statue of Unity that we have multiple data loggers, multiple sensors from Campbell Scientific in it to study the impact these big structures might feel over the time. So if I talk about product line, we offer our data loggers, we offer different sensors, we offer all the structures that are required, being a 3-meter tripod or a 10-meter or a 32-meter met mast to do all the monitoring. So as a company, we supply the complete product line. Some notable projects, this is one of the pride for Campbell Scientific globally. We have our weather stations on the Mount Everest, starting from the base to the top, to the summit, now on both the sides, on the Nepal side also and on the China side also. So you can see it does not only have Campbell Scientific sensors, it has sensors from all the global leading manufacturers, RM Young, Gill, all those different companies. But the data logger that sits inside is Campbell Scientific. So this was done by National Geographic. I have given the link also. There's a complete documentary on it, how the people were trained in Campbell's office to install these. Our engineers were not able to go up till there. They were mountaineers from National Geographic who went and did the installations. We've put a lot of weather stations till date. If I talk about India, we have more than 400 Campbell Scientific weather stations, mainly for research purpose. A list of clients who trust our dependability and the accuracy and the reliability that we give to them. All those ISRO is a very big client for us. We have done systems for water level and water discharge. We have done for inquires for tsunami warning. So that shows the trust all these organizations have in us when it comes to casualties, when it comes to the real alarms that are needed. That's where they have put Campbell Scientific, even when we are on the expensive side. It was recently, there in the LinkedIn from NCCR. They have deployed our systems on the floating buoys in the deep seas. And there was a recent storm that came, the cyclone Ditwa. During that also, all the data was recorded and transmitted back to their station. Eddy covariance system, NRSC is one of the biggest clients that we have for eddy covariance. They've put in different forests. They've put in urban environment. Agriculture is also one of the major clients when they talk about the gas emissions from different crops. So eddy covariance is one of the applications. Glacier monitoring, as I showed for the Mount Everest. In India also, we do a lot of glacier monitoring for organizations like NIH, Wadia, and SASE in the Chandigarh. We have our systems on all the glaciers in India wherever researches are going on. Solar monitoring, we've installed more than 500 weather stations for solar power projects in India. NTPC has approved Campbell Scientific for all their projects. They need to know how much radiation was coming. They need to know how much the panels were heated. They need to know the impact of dust on the solar panels for generation. And that's where all these systems that we supply give them accurate data. That's all that I had for the company that we offer. If anybody has any questions, I'm happy to answer.
Thank you, Mr. Anuj. So, anybody interested in instrumentation? Dr. Pawan, you have a lot of questions regarding instrumentation.
Yeah, usually Campbell, I'm like fond of Campbell team, but just your instruments are very costly.
So the reason that you don't need to buy them again and again, I still have the time I started working for Campbell. Campbell in India is new, but we used to work for a dealer before. I started working in 2012. The stations which were installed during that time are still operational, giving quality data. So you know that it's a dependable system. You don't need to invest again and again in that project. You just leave it on us. We'll make sure you get a quality data once you install that. Yes.
So any more questions regarding instrumentation? Okay. Thank you, Mr. So let's conclude session 3, that is digital forestry. And we had three eminent speakers for this session, that is Sri Shafiq PM from 𝐓𝐑𝐿𝐃, Dr. Girish Gopinath from 𝐓𝐑𝐿𝐃, and Dr. Annie A. Elias from 𝐓𝐑𝐿𝐃 Analytics. So Dr. Shafiq PM, he is one of the senior manager for projects and technical coordinator of 𝐓𝐑𝐿𝐃, and he emphasized more on IoT services and different types of drone techniques available with them, and different case studies he's presented, which is carried out in different parts of Kerala. So you all know that IoT is one of the basic requirements for this digital forestry. Mostly it deals with the ground level of data where sensors have been, multiple sensors have been used. Okay, so that gives a form of input for acquiring this digital forestry output. Second, we have Dr. Girish Gopinath is more into remote sensing. So he has a wonderful experience in LiDAR, as well as he already have various projects from different funding agencies where remote sensing techniques have been widely used in forestry as well as in non-forested parts, especially in Wayanad. Yeah, Wayanad is an area. And he's also having an expertise in the field of landslides, especially in this last landslide happened in Wayanad district. So those areas he has been well known in those different areas of applying remote sensing part. Third speaker, we have Dr. Annie A. Elias. She is more into CNN, that is different types of neural networks. It's a convolution neural network. And she explained the science behind it, as well as different types of AI, that is artificial intelligence used and its applications in the different fields of forestry. So I sincerely thank all these presenters who have done a very significant and explanational presentation, so that all the young communities have seen that, as well as they will implement them in future. So the point is very clear. It circulates at one point, that is how well we can effectively manage our forest using technologies aided designs as well as equipments. So with this, I thank all these presenters. And lastly, I would like to thank the director of KFRI and the entire KFRI team for giving me an opportunity to act as a moderator for this session called Digital Forestry. Thank you.
So with this, we are coming to the end of this technical session on the third day of Tropical Forestry Conclave. So I would like to thank all three eminent speakers for their wonderful lectures, and Dr. Magesh for very efficiently moderating the session. So now I invite our director, Dr. Kannan C.S. Varya, to give away the mementos to the eminent speakers of the day. I first invite Sri Shafiq PK. He's a program and technical coordinator from IC4 Trivandrum, to accept our token of appreciation. Next, I hope Professor Girish Gopinath sir is around. Yeah, thank you, sir. Professor Girish Gopinath sir, he was one of the speakers, but he is not in the hall right now. So I would like to invite Dr. Magesh, scientist, CWRDM Kozhikode, to accept the memento. Next, I would like to invite Sri Anuj Sharma, who represented the 𝐓𝐑𝐿 scientific team and gave us a very technical presentation. So with this, thank you, sir. With this, we are concluding the technical session of the third day of the conclave. So I would like to thank all the eminent speakers. There were 9 speakers of the day, so I would like to thank all the eminent speakers, all the moderators for the successful conduct of the event. And I would also thank the active participants, the student participants from various colleges, their faculties for coming over here and actively participating in the session. And I would also like to thank all the staff, project staff. I hope, excuse me, sir, Director, sir, can you please come over? Sir, I would like to invite Professor Girish Gobinath, sir, at 𝐓𝐑𝐄𝐋, Cochin, to accept a token of appreciation from �KFRA. Thank you, sir. So with this, we close the session for today. So after this, the tea has been arranged just outside the auditorium, main auditorium, and we have a poster session, poster evaluation session, I think immediately after this. So those who have brought posters offline, physically present here, are supposed to stand near the posters. So in case if there is any justifications from the committee, they are in a position, they should be in a position to answer. So otherwise it is closed for the day. Thank you all for the active participation.